各位现场来宾还有线上来宾，那我们会议即将开始，将于9点正式开始，谢谢。
翻转新生，传承创新。台中荣总在岁月的淬炼下，蜕变成长。于时光的洪流里与时俱进，开启医疗新纪元。
融合新科技，让医疗有了无限的可能。你可曾想过，未来的医疗会以何种方式呈现？台中荣总引领智慧医疗新趋势，邀请您与我们一起迈向未来医疗的新境界，共同创造体验医疗新模式，兼具智慧与人性的就医经验，实现全人智慧医疗典范的美好愿景。
李副院长、徐院长、郭院士、邱院长，还有各位与会的嘉宾，还有线上的嘉宾，各位早安。那即将开始我们2022年精准医疗国际高峰会。那首先我们先请我们陈院长致辞。那院长因为他公务繁忙，所以他预先录制了影片。
各位先生、各位女士，非常荣幸的有这个机会，台中荣民总医院在今天举办精准医疗国际高峰会，非常的感谢在精准医疗相关的各个领域的专家学者，今天受邀来到台中荣总，或者是从线上发表演讲。那诚如在座的各位先生女士们，你们所了解的精准医疗一直是全世界还有我们台湾医疗业界大家非常重视的问题。自从美国的奥巴马总统在2015年极力的呼吁大数据以及精准医疗的重要性之后，在全球健康守护以及医疗的发展，大家对于精准医疗的各个面向都有所琢磨。那台湾医界以及台湾各个医疗相关的研究机构，比如我们的中研院、国卫院，对精准医疗也都是不遗余力的在推动。那台中荣总，那自从前任院长许岳衡院长参与 TPMI 的计划，那台中荣总至今在于这些基因检测标本的收集，一直是稳居全台湾医学中心的第1名。那在精准医学的各项研究，还有论文的发表，也都不遑让。那现在更是有多位的主治医师、教授们加入台中荣总的精准医疗的团队。那全院在精准医疗，在各个教授主任的领导之下，也是有蓬勃的发展。那今天很高兴有多位院外的教授、理事长们共襄盛举，来促成今天的精准医疗的国际高峰会。那非常感谢我们研究部的诸位同仁们，在今天会议的筹备上做了很多的尽心尽力的这些的准备的功课。那期望我们的今天的精准医疗的会议能够顺利的展开。那大家在这个周末假日也有一个不错的知识上的飨宴。那期望大家在今天的所得所学，能够在我们以后精准医疗的推动上能够更有助力。那当然最重要的是把我们的精准医疗所得到的这些研发的成果，或者是我们在临床上大家所互相交流的知识们，能够落地运用于更多病人的临床服务，能够提升我们病人的健康，守护我们国人的各项的健康福祉。那最后祝福各位身体健康愉快，再次的谢谢你们的参与，谢谢。
谢谢院长的致辞。再来我们请李正宏副院长也为我们致辞。
好，现在那个国院士，我们徐院长，我们中国副院邱院长，以及我们现场以及在座的各位嘉宾哈，大家早安。那真的欢迎各位来参加我们这个精准医疗的高峰会。 那大家知道，台中荣总在我们前院长、许院长以及我们陈院长的大力推动之下，成立这个精准医疗中心啊。那我们有的基因库有10万个啊，在8月的时候，这 TB 研究计划啊，中央研究的计划总共有53万个。那么这么多的基因库的话，我们希望将来借着这个大数据以及 AI 的一些分析啊，可以用来治疗在各种的疾病，可以用到最小的医疗耗能来治疗医疗的获益这样子。那我们知道精准治疗目前为什么方兴未艾呢？以前我们在一些癌症的分科，大部分都以器官的分科，那其实一个基因它可以导致很多的疾病，很多的癌症，所以将来来说的话，可能不是以器官来分科，而是以基因治疗的方向来分别这个不同的癌症。就像我昨天才看到的 Merck 的在去年6月有一个药Keytruda，那它又不只是可以治疗这个大肠癌，也可以治疗多种的癌症。这个可能对我们来讲，这是一个 promising 的一个 result。那这个精准治疗其实它不只可以治疗癌症，它可以治疗胃病，也就是一个预防医学。所以我们现在喊 precision medicine， 我们将来喊的是 precision health。那 precision health 的话，就是在慢性病的话，也许我们可以达到预防的效果。我本身是一个骨科医师，那我每天我已经开过6000多例的椎间盘突出。那椎间盘突出它本身它就是一个慢性病。那有些人负重他会得椎间盘突出，有些人负重他不会，有些人他怎么搬重他都不会，有些人稍微一搬他就会。那就是个人的基因不一样，个人的 DNA 不一样，个人的体质不一样。那就像椎间盘突出这个基因也慢慢被找到了，将来我们可以配合，假如你有这个基因的话，我们可以配合你生活的形态的改变，那你就可以不用你就不会造成椎间盘突出。像这样也可以做到预防医学。那第三个就是说，其实我们虽然可以做基因检测，可是基因检测现在在各地其实你问5个医师他的讲法不一样，问5家公司他的收费也不太一样，所以民众其实莫衷一是。那医师也各有各的说法，这说明了我们现在精准医疗正在发展当中，希望将来能够更聚焦，我们研究的更清楚，让民众有更大的一个卫教。那今天请到真的是各位的都是精准医疗，各位的也都是在这个领域的佼佼者了，我们很荣幸邀请到各位来 share 你的 knowledge。那我们也希望这个高峰会来把这个 knowledge 传递到各位，让各位能够收获满满。最后敬祝大家身体健康，谢谢。
谢谢李副院长对我们现在跟未来精准治疗的一个提点。那再来，我们要进入我们的正式会议的开始。那我们首先欢迎中国医药大学附设医院癌症医院邱昌芳院长来当我们的座长，然后 Dr. Ansoni 是我们的 speaker。那请。
大家早安。今天大会的主席陈副院长，还有郭院士，还有许院长，还有各位嘉宾，我很荣幸能够担任第一场的介绍人。It is my great honor to have the opportunity to introduce you the first invited speaker, Dr. Anthony Magliocco from the United States. Dr. Anthony Magliocco is a professor of pathology at the University of Central Florida. He has over 20 years' experience in cancer pathology and precision oncology, along with the molecular diagnostics and translation cancer research. He has founded and led multiple clinical laboratories and has overseen the deployment of numerous new laboratory tests in his career in both Canada and the States. He is also the president and the CEO of Protein Biodiagnostics, which is a precision cancer diagnostic company. Dr. Magliocco also published over 300 research articles in many leading journals. So today, he's going to give us a talk online on the topic of implementing molecular tumor board to improve delivering precision oncology to physicians and their patients. So let's welcome Dr. Magliocco.
Thank you so much for that introduction. I hope you can hear me clearly and also see the screen. That was a very kind introduction. Thank you so much for inviting me to your session, to your symposium. I hope you're having a wonderful symposium. I'm sorry I can't be there in person. I'm coming to you, it's Friday evening in Florida, and I'm actually in Fort Lauderdale attending an ESMO meeting at Fort Lauderdale. So it's my pleasure to give you an overview of implementing molecular tumor boards to improve precision oncology delivery to physicians and their patients. The information here is primarily for educational purposes, and some of the products we refer to may not be available in Taiwan. I'd also like to thank Roche for helping sponsor this event with me, and they've been a partner of mine over the years. You heard my background, so I won't really go over it again. My most recent experience is at the Moffitt Cancer Center, where I was head of pathology for about 8 years. Moffitt is distinguished by being one of the foremost cancer centers in the United States and starting a precision medicine program that went back to the early 2000s. This program was called Total Cancer Care, and they've now recruited over 400,000 patients to this program, where they've banked blood samples, tissue samples, and collected molecular data from the patients. So Moffitt has been very much a leader in precision medicine over the years, and I've been very fortunate to work at some very leading centers and work with other collaborators that I've learned a lot from about implementing precision medicine. I left Moffitt 3 years ago to form Protean Biodiagnostics. Protean is an early stage company that's located in Orlando. You're welcome to come to visit us. We're very near the airport in Orlando. If you're coming to go to Disney World or to vacations in Florida, it's a great place to visit. The laboratory is a CAP-CLIA laboratory, and its function is to really implement and deploy new precision medicine tests. The lab is located, there's a special part of Orlando called Medical City that's been developed to house many innovative medical facilities. It contains a new medical school, a Florida Cancer Specialist Phase 1 unit, a Veterans Hospital, and other new innovative centers. Another thing that's somewhat innovative about this part of Florida, it's where the space program is implemented from. And many of the astronauts go through Lake Nona and Orlando prior to their launch into space, and there's opportunities to study the impact of microgravity on humans. Now, over the last number of years, medicine has changed dramatically. We've gone from traditional medicine to precision medicine. I've been fortunate to live through this time and really see the onset of precision medicine. I was trained in Canada over 20 years ago, and at that time, when we treated cancer, we were really limited to treating with surgery, chemotherapy, and radiation. Over the last 20 years, there have been many new advances, most remarkably in lung cancer, where we now understand the molecular basis of cancer and have developed numerous therapies that target the molecular drivers of cancer in lung cancer and other similar cancers. Also, more recently, we've discovered the immune system may play a critical role in cancer, and novel therapies have come about to really modulate the immune system. And finally, we're learning more and more about host genetics and how host genetics drive cancer and how therapeutics might be used in concert with the host genetic system to treat cancers. And the best example of that are PARP inhibitors that are used in people who have inherited homologous recombination deficiency problems. The problem, it's an exciting time, and if we look at these charts, the chart on the left shows the number of FDA approvals of cancer therapies. You can't really see the numbers here, but this is 1950 through to the year 2000, and generally there's only 1 or 2 approvals a year. Since 2000, the number of approvals have gone up dramatically, and in 2021, there were 56 new approvals of new cancer therapies. More than half of them with targeted therapies and require the use of companion diagnostics. This chart in the middle shows how many new companion diagnostics are being published. The number are absolutely remarkable. And the chart on the right shows the number of therapeutic choices for a doctor when they have a particular cancer. Each red dot represents a different type of cancer. The green dots represent the various therapies that can be used for that cancer. You can see that this is quickly becoming an overwhelming problem for cancer doctors to manage. There are too many drugs, there are too many choices, and it's becoming very, very difficult for cancer doctors to keep up. Within the United States, there's the National Comprehensive Cancer Network. These are the leading centers that do research and really define how cancer should best be treated. These are where the trials are done, and they publish guidelines. So every 6 months, the Comprehensive Cancer Network has updated guidelines. Unfortunately, only 5 to 10% of Americans can actually attend these cancer centers. The vast majority of Americans are treated in community cancer centers and do not necessarily get access to the latest therapies and clinical trials. One of the problems is that there's a growing disparity. The patients who are being seen and treated in community cancer centers are not benefiting from the advanced treatments that are now available. Now, if we look at the uptake of biomarker-driven testing, it's recommended that every patient with lung cancer undergo EGFR testing, ALK, ROS, and BRAF testing. What we can see is actually in the United States in the last few years, less than half of the patients have undergone the testing that is recommended. So there is a large gap that needs to be closed if patients are to receive the best therapy and recommended guidelines. This gap is growing, and there are many barriers that are causing it. One of the problems is that community-based doctors are having difficulty understanding what tests to use and also how to understand the results and what to do with the data. Many of them feel confused and isolated. How do we solve this? One of the solutions to close the gap are molecular tumor boards. In large academic centers, molecular tumor boards are frequent events. When I was at Moffitt, there were 2 or 3 tumor boards every day, and there was a molecular tumor board once a week. Molecular tumor boards allow doctors to talk to each other, to interact with other specialists, and to review data. So this is an important activity to allow doctors to interact. Now, the way that they currently occur is physically. So in a large academic center, the specialists can get together in a room and review the data. What about in the community where the specialists may be isolated or where there are no specialists? This presents real challenges in implementing molecular tumor boards. Now, do molecular tumor boards have value? Here is some data that shows that when patients go to molecular tumor boards and that there is a recommendation from the tumor board, they do significantly better when the recommendation is followed in both progression-free survival and overall survival. So there is evidence accumulating that molecular tumor boards can impact patient care by making treatment decisions that are both evidence-based and effective. Now, what are the challenges of implementing molecular tumor boards? One of the challenges are there are no quality requirements. We don't really know what is the best practice for a molecular tumor board. We're still learning. There's a gap in the knowledge and the genomic potential that we're trying to close. And in fact, 22% of physicians say their knowledge is inadequate and that they need further education. The other problem is that they're often, when a molecular tumor board makes a recommendation for treatment, payers may not actually implement that recommendation, and that's certainly a problem. There are also logistic hurdles in that it's difficult to get all of the doctors together in one room. And then there's complexities in trying to link all of the data together and make a treatment recommendation. So there are many, many barriers. But despite this, progress is being made. The other question is, are molecular tumor boards all run the same way? And actually, they vary quite widely. If we look at different medical centers, we may find that the composition of the molecular tumor board is different. There may be different types of specialists. Sometimes there's only medical oncologists. Sometimes they include PhD scientists, for example, biochemists, geneticists. Often there are doctors of pharmacy as well. The molecular tumor board at Moffitt included doctors of pharmacy, and that was a very effective addition to the tumor boards, as they could often make recommendations about specific therapies. In addition, the workflow of the tumor board may vary, and the tests that are being evaluated are also variable. Finally, there are different decision support tools available. So at the current time, there is a lack of standards in molecular tumor boards. We do know that molecular tumor boards are valuable, and how can we try to move this forward and close the gap? Here is another study where several different molecular tumor boards were presented with the same patient. So data was given to the molecular tumor boards about a certain molecular profile, and then the molecular tumor board was queried as to what the response was. Surprisingly, there was different recommendations for each patient. So this shows the lack of consistency in even between tumor boards when there are molecular tumor boards about interpreting the data and making a treatment decision. So there's definitely a lot of subjectivity, and I think what this really goes to show is that work needs to be done in both standardizing molecular tumor boards and learning how to interpret molecular data to supply the best and most effective treatment strategies for patients. Now, if we compare a molecular tumor board to a regular tumor board, there are several differences. Typically, a molecular tumor board will only focus on selected cases. These are particularly cases where a molecular profile might have been done. In the United States, typical molecular profiles might be the TSO 500 test, Foundation Medicine, Caris, or others. So when one of those complex genetic or molecular tests are done, this may trigger molecular tumor boards. Also, molecular tumor boards are generally currently only done at large academic centers. The memberships are, as I talked before, doctors of pharmacy, basic and translational scientists, and often the discussion revolves around biochemical pathways and theoretical considerations of treatment. So the molecular tumor boards tend to be quite different from the regular tumor boards. Now, if we look at regular tumor boards, they tend to look at all of the cases, but they're organized according to site. So there'll be a breast cancer tumor board, there'll be a brain cancer tumor board, etc. They'll talk about other aspects of the case. What about surgery, radiation oncology? Does the patient need access to a clinical trial? And so on. However, the molecular and the genetics are taking a bigger role in regular tumor boards, and I'd like to think in the future that molecular will be part of every tumor board and not really a separate thing. So as we consider all cancer cases, we're learning that a molecular analysis is very important to characterize the case and to select optimal therapy. So I think as the future goes on, we're going to see more and more molecular data incorporated into regular tumor boards. However, in the meantime, there's probably still a place for molecular tumor boards where expertise are really focusing on the innovative new molecular tests and what they actually mean for patient treatment. Here's an example of some of the people that will sit around the molecular tumor boards. So there may be a clinical oncologist, experts from molecular biology, pathology. Pathology is always very important. You have to know what is the histologic diagnosis. A pathologist is critical to stage a tumor and to determine if lymph nodes and margins are positive. Often, particularly in the United States, sometimes pathology is a problem, particularly if there's generalist pathology, and we strongly recommend that when a patient has a cancer, that the pathology be reviewed. That particularly in the generalist case, there are often gaps and errors in the pathology, and it's very valuable to review the pathology to ensure accuracy of diagnosis and to also select the correct case material to be used for molecular analysis. Because sometimes if a molecular analysis is done, the wrong block is selected. So pathology is very, very critical to that. Now, clinical genetics is becoming more important too, as we now know a greater percentage of patients with cancer have inherited cancer syndromes. And in fact, the more genes you test, the more likelihood you find that there are inherited genes that are driving those cancers. And there are also probably polygenetic factors that can also drive cancer. We are also learning that there may be pharmacogenomic factors as well that can determine what sort of medicine should be used and whether there's any sort of toxicity. So in the future, we're going to see a greater role for clinical genetics in the molecular tumor board, because the clinical genetics can also guide the therapeutic decisions. Further, if a patient has a predisposition to cancer, their future surgery might have to be different. For example, if they have breast cancer and they're from a high-risk family, they may want to consider bilateral mastectomies or even oophorectomy. So understanding the clinical genetics is very important. Also important is bioinformatics. As we learn more and more about the molecular basis of cancer, we're now dealing with issues with variants of uncertain significance and novel discoveries in altered DNA. Having a bioinformatics expert can help us keep up to date and determine if an alteration is important in a protein or biochemical function. And we're really becoming more and more now. I'm looking at cases, and often we're trying to determine, are there other examples in the literature? But as we do more and more cases, we find that there are truly ends of one, and we really almost have to consider each patient as an individual, as there may not necessarily be another example of that mutation in the world's literature, yet that mutation is still an important driver for the tumor. Finally, there needs to be a molecular tumor board coordinator. This person is critical in organizing the event, in placing in cases, and really recording what happens during the tumor boards. The molecular analysis is becoming more and more complicated. If we consider where the data is coming from a patient, there's often tumor tissue, a pathologic diagnosis leading to genetic testing, other types of molecular tests. We're also seeing more and more use of blood samples or liquid biopsy, and this is quite exciting because we can now look in real time what is happening to the tumor. Is the tumor spreading? Is it changing? Is it responding to treatment? So I believe we will see more and more liquid biopsies that will become critical to both diagnosing and managing cancer. All of this information now needs to be organized. So there may be multiple tests, so there needs a way to put it all together into a patient report that makes sense for the doctors that are really looking at those reports. The cases are then annotated, prioritized, and put together. These can then go to the molecular tumor boards. The molecular tumor boards can advise on molecularly guided treatments, or they can help assign patients to ongoing clinical trials. Finally, the patient report does need to report back out so that that information is available for other practitioners. Now, Roche is doing a lot of work on this, and Roche has created a commercial platform that has several products. One of them is called Navify Digital Pathology. So this is a digital pathology solution where slides are scanned through the DP200 and into the UPath digital path system. So those digital data can then be recorded and preserved so that the pathology image is now recorded and it can be shared with other pathologists. It can also be analyzed with computer algorithms, which can be useful as well. There are also tools for molecular genomics, something called Navify Mutational Profiler, and then there's also a Navify Tumor Board where all of this information can feed into the tumor board. Within the tumor board, specialists can log on, see the information, and network with each other to make decisions. Within the molecular profiler can take in raw data, can take in VCFs, can look at clinically relevant variants and actually make recommendations about treatment options and clinical trial matching. I'd encourage you to have a look at this. I find this to be a very interesting product that can be quite helpful in integrating different kinds of mutational data. So you can put together different sources for the molecular data and also put in immunohistochemical data and other kinds of data. The profiler will then organize it and make recommendations and clinical trial matching. So I'd encourage you to have a look at it. I think it's quite a good product. The other thing that Protean is working on is we've created something called an integrated summary report. And in the United States, the doctors generally don't like to read a lot of long reports. So what we've done is we've taken all of the key information and put it onto a very short report, and we put the recommendations on the top. We have a summary of the pathology, liquid biopsies, molecular analysis, and genetics to make it very, very easy. So this is very high-level data that the oncologist or the surgeon can have to quickly review when they're seeing a patient, and it really gives them the pertinent information about what they need. If they want to go into the details, they can then go and look and read these very complicated reports that are also attached. But the integrated report is a way to really present that data in a very easy to use way for them. Now, here's an example, or here is how the virtual tumor board works. So within the virtual tumor board, each doctor has their own account. When they log into it, it's in a HIPAA secure environment, and they click on, they get a list of their patients. When they open the patient, there's access to the digital pathology, there's access to the radiology, and all of the key reports are organized here for them chronologically. So this might be a genetic report, this might be a surgery report, this might be a path report. So they can just click on each one of these and see the report in here. At a high level, there's a summary that really explains what's happening with the patient. The TNM status is here. And then over here, there's some tools that are available. There's a guideline tool. If they click on that, that will allow the program to look at what are the NCCN guidelines. There's a PubMed literature tool, and there's a clinical trial matching tool. So within this environment, the doctor can explore the case, they can search the guidelines, and they can really plan their treatment. They can also invite in other doctors to review this or meet with other doctors to really go over a case. And having this environment allows doctors to organize their patients and really have a very nice environment to really study the case and the molecular findings. This is a deeper view of it, again, showing the tools that I talked about, the organization of the cases, and where the various clients are available to click into it. So I think that the Navified Tumor Board is a very interesting platform that can support tumor boards across the country, across the world. So people can join it at different times, at different places. So I think this virtual tumor board is a very, very powerful platform that allows physicians to keep up to date, but also to network and communicate with each other. I also use the tumor board to talk to patients. So when I'm reviewing a patient, I can invite a patient. We can go over the results together. We can show them information and so on. So this system is very, very powerful for communication. Now, here's an example of a program we're running in the United States called Delphi. It's called Delivering Precision Health Insights for Timely Treatment of Patients. And here's an example of a case that we treated at Protean. The case was actually a young man who lives in Australia. His mother contacted me and told me that he had advanced cancer and that he had difficulty getting testing in Australia, and could I help him? It turned out that he was visiting the United States. So when he was in the United States, we arranged to get his samples from Australia, also do a liquid biopsy, and do a genomic profiling. When we analyzed his tumor, we determined that he had NRAS activation and EGFR activation. We also utilized an innovative molecular profiling system called OncoSignals, where we're able to identify pathway activation. So here it measures 7 pathways: estrogen, androgen, PIK3CA. You can see PIK3CA is high, estrogen is low. The way you read this, red is high, blue is low, gray is in the normal range. If it's above into the red, that means this pathway is high. Here, this pathway is low. So when we read this, we can see that PIK3CA is high. We can look down here. We can see that hedgehog is high. So that this patient could potentially be treated with a PIK3CA inhibitor and also a hedgehog inhibitor as well. So it's a powerful method to look at signal activation. We then fed his data into Navify mutational data and into the clinical trial match, and it actually read out a clinical trial that was available in Australia. We told his doctor about that in Australia. The patient was put onto this clinical trial, and the patient had a complete response. So his tumor, he had a complete pathologic response, and now he's back at college and doing very well. So here's an example of how we can apply precision medicine and take care of a patient that's on the other side of the world. We can bring the information together and bring experts from around the world to really work and make a difference in the life of a patient. So in summary, I think the future of medicine is precision medicine, not just for cancer. You're going to see it for other conditions as well, neurological conditions, newborn conditions, and so on. And I think as we go forward, we're also going to see a movement towards precision health. How do we keep each patient healthy? How do we avoid them getting advanced disease? And each patient may have different risk factors and different ways to modify their risk. There are many challenges and barriers to overcome in terms of this, but I think we have some new tools that are becoming in place that really are making a difference. We see that precision medicine is not just for cancer. We're really going to be seeing it for precision health. Now, when we look at community cancer patients, particularly in the United States, they're at a disadvantage. This is because their doctors are now practicing in isolation. They have difficulty keeping up, and that they have difficulty networking. We think that the new digital tools like the Navify virtual tumor board, the profiler, and other tools will actually help close the gap. So we will now be able to easily deliver advanced diagnostics to community-based patients and doctors, and also the decision support systems that they need to understand how to use those advanced diagnostics, and also the network tools that they can communicate with their own peers and other experts from around the region, around the world, to really make a difference. So as we are talking today across the world, giving a lecture, I think the future is really about interaction, sharing knowledge, and really moving quickly. So I'm very excited about the opportunities in precision medicine. I think this is a really exciting time, and we're going to see many, many new innovations for both treatment of cancer patients and for other diseases as well. So I really look forward to the future and all of the remarkable advances that we will see. So I'd like to thank you for the opportunity to present, and it was, and if there's any time for questions, I can try to take those. Thank you very much.
Thanks, Dr. Magliocco. Very informative and very inspiring talk. Now we have, it's about 10 minutes open for the question or comment, and from both from the floor or online friends. Is there any comment or question? Yes.
Yeah.
Dr. Zeng.
Yes. Dr. Ansoni and Dr. Zeng and HEMA medical oncologist. And thanks your very wonderful talk. And I wonder if you told that because meaning why side and side variable in molecular tumor board treatment recommendation. So if you have different side, have the same mutation, have the different treatment station, how do you see this situation, and how do you recommend to improve this treatment difference? And yes.
Yeah, that's a very good question. I think you're, if I understand correctly, you're asking that there's a lot of variation in how doctors treat or select treatment. How do we minimize that variation? So one approach is by making evidence easily available. So the guidelines are being created by groups like the NCCN, ASCO, and other regional groups. I think these guidelines need to be easily available to doctors so that they have them at their fingertips. So then when those guidelines are available, those are the best available evidence. So the guidelines are created by experts who review the literature. They look at the level 1 and 2 and 3 evidence, and they base recommendations saying, if you see this mutation, you should use this drug. So using guidelines are the first way to make sure that high quality treatment is done. The second way is developing tools that will help us understand how mutations work. I think this is further down the road, and we are starting to see the emergence of artificial intelligence tools that can manage very complex information from molecular analysis and make predictions about what might be the best therapy for a particular patient. Using tools like this might be able to standardize how treatment is used. So that's another factor. The third is that we're seeing some exciting work with ex vivo analysis, so that if a patient has tumor, perhaps you can grow it like an avatar ex vivo, treat it with a type of drugs in an organoid system or so on, and really see what really works in a patient. Now that's obviously extremely experimental and a long ways away. Finally, there is the art of medicine, and every patient is different, and this is why we're doctors. So doctors really have to weigh up what's best for their patients. So does a patient have other conditions? Do they have mental illness? Do they have other debilitating conditions? And then it really comes back to the doctor and the patient to really determine what's best for that patient. So I think at the end of the day, there's still a human touch. There's still the art of medicine. But if the patient and the doctor are highly informed with the best available evidence, at least they know the best choices, and then they can make the decision together what's the right treatment for a particular patient. I hope that answers your question.
Any other question or comment? Yes.
Thank you, Professor, for your wonderful talk. I'm Dr. Chen, and I'm a rheumatologist. And my question may be not so direct to molecular tumor board. I'm wondering, is there any cases of non-tumor precision board in the United States? For example, in our hospital, we test patients with unknown fever condition, try to understand whether there is autoinflammatory diseases. So I'm wondering, is there any particular commercially available platform or organizations that has worked very well in non-cancer field? Thank you.
I think that's a really great question, in that these tumor boards don't have to be just molecular cancer tumor boards. They can be general tumor boards for other conditions like infectious diseases or rheumatology. And I think in many ways the same tools could be placed into them. I haven't seen an implementation, but I think you could use the same framework. You may have to have information from pathology, you may need imaging, you may need lab results and genetics and so on. So I think the basic idea could be used. I wouldn't be surprised if some centers have created their own based on something like a Teams platform or something. But it's a very, very good suggestion. I haven't seen one, but I think that you could modify an oncological one, or we could actually work with some of the vendors of these tumor boards. So for example, this one is a Roche product. We know that there are other vendors as well. So working with some of these vendors could modify the product for the other applications like rheumatology. I think that's a really excellent question, and really points us towards precision medicine in general.
Any other questions? Yes.
Thank you for your talk. I'm Pui Kwok from Academia Sinica. I have two questions. The first one has to do with the unevenness of the pathological and board recommendations or diagnosis, and this is a quality issue that we face all the time. And how do you envisage this going forward to have better consensus based on good scientific evidence? That's the first question. The second question is from the patient's point of view. We know that only a small fraction of people have effective target therapy right now. So for the people who have no hope after the molecular tumor board, it seems like a death sentence. How do you deal with the psychological effect of the people who don't have a good path forward? Thank you.
Right, so those are two good questions. So one is the quality of implementing evidence-based medicine. And I think that you can either encourage doctors to practice evidence-based medicine or force them to. And I think that most places or most doctors would agree that medicine should be evidence-based and that we need to make it easy to get the evidence and to implement it. So I think things like the tumor boards are there. In the United States, another way is to tie payment to it, and also promotions and other things to the use of evidence-based medicine. So it's basically the sticks and carrots to implement evidence-based medicine. But as you pointed out, patients often fall off of the evidence because after second, third line of treatment, there may be no evidence left. Now, we only talked about targeted therapies because there may be other types of therapy that are available as well. So patients may also be open to novel types of radiotherapy, or maybe there's novel types of general chemotherapy. But even if a patient has exhausted all hope, we still have to manage a patient with proper supportive measures. And in Canada, that's where I trained before. There was a lot of work put on really managing cancer as a chronic illness and also palliative care. And that proper palliative care can dramatically extend the life of a patient, that no patient should have to suffer. So proper pain control, proper spiritual support, proper physical support, and so on are also very important. So those are also issues that cancer is not just about targeted therapies, it's about holistic medicine. It's about really taking proper care of the patient and making sure that they're fully supported through their journey with the disease. So two excellent questions. Thank you.
Any other question? Actually, myself, I have two questions. The first one is, do you suggest that this molecular tumor board should also work on the cancer survivorship and the cancer genetic counseling? If yes, so how do you suggest to work it?
I think that's, you raise a great point. One is cancer survivorship, and also having patient advocates on a tumor board would actually be quite interesting. I haven't heard that suggestion before, and I think that might be quite interesting if we did have survivorship, patient support on there. In the United States, it's pretty common to also have, say, rehabilitation support, social support, and other types of support on there as well. So I think that can be incorporated onto a tumor board. Also in the United States, genetic counseling is quite common, a feature, and generally at the tumor boards, the genetic counselor will be there, because often it'll turn up that, oh, this patient didn't have a genetic test, they're young, it turns out that they may have an unusual tumor, or they had a couple of tumors, that there's some family history, and then you can refer that patient over to a genetic counselor. So in the United States, often a genetic counselor would be on the tumor board. There would also be a clinical trials coordinator on the tumor board, that they would be there as well to see if a patient might qualify for a clinical trial and get them onto the clinical trial. But I think those are really important things as well. The other things, when I work with patients directly, that often I have a number of patients that come to me, and I strongly suggest that they also go to their own foundations and societies, that there's also great support networks where patients can talk to each other, they can share their own information, and there's other support for both their physical and mental support that can be very helpful as well. So getting patients engaged into support systems in their own community, I think, is really important for holistic support and proper care. So those are really two good questions.
Thank you very much. Time's up, so I have to close this section. So I'd like to thank Dr. McGregor again for the wonderful talk and a very interesting discussion. And now I'd like to close the section. Thank you very much.
Thank you. Thank you. Bye-bye. Thank you. Bye.
谢谢大家的参与。我现在要把这个交给下一个 section 的主持。
那我们欢迎下一场座长许惠恒教授，还有讲者郭佩恩院士。
郭院士，还有李副院长、邱院长，还有在座的各位贵宾，还有线上的各位贵宾，大家早安。很高兴来介绍我们下一位 speaker， 目前任职在中央研究院生医所的郭佩恩郭所长郭院士。那郭院士他是在美国的University of Chicago 拿到他的MD degree， 然后在 University of Washington St. Louis 完成他的住院医师的训练。在回台湾之前，他是在 UC San Francisco 的 Distinguished Professor 在 Genetic 跟 Dermatology。那回台湾之后，他努力倡议这个 TPMI， 这是台湾也是全世界非常有名的很大的 Precision Medicine 的一个 cohort， 在几周前已经达到56万人哈。那这样的数字在欧美以外是 No.1 了哈，在全世界是前几名了哈。那很谢谢在座以及线上的很多贵宾跟医师一起参与 TPMI 的整个 activity。那目前我们持续收案，也希望能够 implement 到我们第一线照顾病人。那今天很荣幸邀请院士跟我们谈一下，也是他的专长哈，怎么去做这个 implementing 这个 precision medicine in Taiwan. So please join me to welcome Professor and Dr. Guo, please.
So thank you very much for the introduction, and thank you very much for having me here to speak to you. So I've been given an assignment, actually, to talk about how to implement precision medicine of common diseases in Taiwan. So when we think about precision medicine, we first have to think about how we have health and disease. So there are certain things that determine our health or disease. The first thing has to do with genetics. And then we also have to deal with the disease biology. And then we have to think about the environment that we find ourselves in. Unfortunately, this is a picture of Taichung. And then we have to worry about how we conduct our lives in terms of our lifestyle. One thing, though, that we always forget is that there is also an element of chance. Because if you are never exposed to COVID, you will not get COVID. But then if you are exposed to COVID, then all these other factors can affect whether you get it or not, whether you have a serious disease or not. So we cannot control the chance, but we can get information from the other elements. So precision medicine then is to try to incorporate as much information as we can into clinical practice. And the goal is to prevent disease, prolong the time that we are healthy, and to optimize medical care. So the goal is not just treating diseases when it's already there, but actually we want to delay the disease onset and to treat it in the best way. So when I think of precision medicine, we think of dividing the population into different risk groups. some people have heart disease in the family, some people have cancer and so forth. But then once we have this information, then we have to act on it. If we just know the information and do nothing, then it's the same thing. Even if we don't know the information, it's the same thing if we don't do something about it. So it has to be proactive, meaning we have to do things to lower the risk for the conditions that we are susceptible to. And then, lastly, everything has to be personalized, because the best way to keep ourselves healthy is different from person to person. So today, I want to emphasize one point about precision medicine. No matter precision medicine for cancer or for rare disease or anything, it's based on predictions. So we want to be able to use a lot of information to predict before we get sick whether we're at risk for that disease and whether we will have that disease young or old. If we know that, then we can take certain measures to lower the risk for the disease. However, sometimes we cannot prevent the disease from happening. Then in that case, we want to get at the very beginning of the disease, we want to predict whether we're going to have bad symptoms or not so bad symptoms, and then we can try to treat for cure. So this is what Dr. Magliocco is talking about when you have liquid biopsy. When you don't even know that the cancer is there, you can already find out that something is happening. If that's the case, then you can do the workup and try to treat it for cure. Now, for someone who already has a condition that is being treated, we want to predict whether the person will have a bad outcome or not so bad outcome. And with that, we can emphasize the treatment to prevent really bad complications for the disease. And finally, another branch is really to talk about getting information to predict how we're going to respond to medicine, whether the medicine is going to be effective or whether the medicine is going to cause severe side effects. Okay, so I also think of precision medicine having 3 different branches. So the first branch has to do with precision medicine to make molecular diagnosis for rare diseases. So someone is born with a genetic disease, how can we identify the molecular signature for that? And if we know that, then we can make the proper diagnosis and know what the prognosis is and how to manage the condition in the best way, and also to help the family plan for the future. And the second is the exciting area of precision oncology that we just heard about. And this is where we want to identify the cancer before it becomes a big problem, or we can identify the best way to treat the cancer based on the molecular mechanisms. But what I want to really talk about today is how to implement precision medicine for common diseases. We know that rare genetic diseases that are difficult is a small percentage of the population. Even for cancer, it's not a very high frequency for the population. But I can assure you that all of us will have trouble with common diseases. All of us will get it. And so I think it would be really good if we spend some time to determine how to manage this. And so this is why I think that we should do it in Taiwan, because Taiwan is the best place to develop precision medicine for common disease. It is the best place because we have very homogeneous genetic background. 96% of the people in Taiwan are Han Chinese. And then by extension, if we do anything in Taiwan, it will benefit all Han Chinese around the world, which is about 1/5 of the world's population. Just for that, we should do it, okay? And then the other advantages are that Taiwan has a very highly educated and health-conscious population. So I watch TV sometimes at night, and then I'll see many advertisements about how to keep yourself healthy. And so that means that there must be a big market in Taiwan. And so because of that, then we really ought to take advantage of this kind of highly educated, very health-conscious population to help develop this really amazing way to handle common diseases. And the other advantage is that Taiwan has a long history of national health insurance, which incorporates a lot of electronic medical data. And also Taiwan has really good doctors and really good medical system. And so this is important because there are other countries that may have a lot of Chinese. But if they don't have a good medical system, if they don't have electronic medical records that can be used to generate the data that we need, then it's still impossible to do. So until about 4 years ago, the only way we can decide whether someone has a genetic risk for a common disease is based on rare mutations in different genes. So based on those studies, in the United States, we can identify only 0.4% of the population with these rare mutations that give us high cholesterol or high blood pressure and so forth that will lead to heart disease. Same thing with type 2 diabetes, only 0.1% of the population, and then breast cancer, only 0.25% of the women in the United States, based on rare mutations alone. But 4 years ago, based on the UK Biobank data, researchers at MIT and Harvard came up with a way to look at common variants. And with that kind of AI-driven approach, we can actually identify many more people in the population with the same level of risk as the people with rare mutations. So if you look at this, we have 20 times more people that are at high risk for heart disease and so forth. So with this then, it can really help us implement a better way to manage these patients from a public health population level. So once we have these ideas and have these high-risk groups identified, we have ways to help them. So for example, if someone has a familial high cholesterol syndrome, then we can do early testing, control the cholesterol, and so forth. Okay, so this is based on UK tools. What about other populations? So based on the UK tools, we can tell that for many conditions, they can identify people with high risk. So 3 means this is 3 times the population average for risk. So you can see that for all these different conditions, using European tools on Europeans, we can identify the high-risk group pretty well. But if you use it for Africans, the same tool applied to the Africans, then you get very poor results, meaning that you really don't get that much help. What about Chinese? So we just looked at 100,000 people using the European-derived tools. So this is interesting. For some of the conditions, actually the European tools work just fine, right? For prostate cancer, diabetes, and so forth, it worked pretty well. But then for many other conditions, it doesn't work so well. So this is a very important point that we must remember. If we don't do it in Taiwan or China, then we will never get it because the European methods will not apply so well. Okay, so based on this background then, 3 or 4 years ago, we decided to do something in Taiwan. So this is the beginning of the Taiwan Precision Medicine Initiative. So in order to capture as many conditions as possible, we have decided that we have to do a big cohort. So 1 million is an arbitrary number. But to think about 1 million means that if a disease prevalence is only 1%, if you have 1 million people, that means that you have 10,000 cases. So we think that that's pretty good because all the major diseases will be covered, and even some of the subtype diseases within each ICD-10 code can be obtained with sufficient cases. So then when we recruit these people, we are partners with them. So they are not donors, they are participants because they give us access to their comprehensive clinical record. And then we also a DNA sample that we can generate their genetic profile. But we will return the results to them and give them guidelines as to how to keep themselves healthy. So it's a two-way partnership between the participants and the project. And so in the end, we want to be able to come up with disease risk predictions and also personalized health management for everyone who can come up with a genetic profile. Then we can match it properly. So this is a very different design. Many people think that the TPMI is a biobank. It's not a biobank because it's a very different design in that I just mentioned that it's a participatory study, meaning people don't just give us some sample and never hear from us again, but it's something that we do together. And it has both genetic testing and genetic profiling. So genetic testing is based on all the known literature. So BRCA1 mutations, APC mutations for breast cancer and colon cancer, and so forth. But then we also have genetic profiling to help us generate these polygenic risk scores. And so the other commitment of the participants is that they give us full clinical data access forever. Well, as long as the project goes. So they are going to give us this data, so which is going to be very interesting to follow patients when we give them this risk, whether they actually will have the disease in the future. And then we analyze the full data to develop what we call the polygenic risk scores. And this will then be returned to the patients over time. As the genetic testing results, we can give it to them right away. But then for the common disease risk results, it will come online as we have them. And then we want to give back to the participants guidelines for health management. Now this is, remember, we talk about proactive. So if we just give them the report, if we don't help them to manage the risk, then we are not really doing our job. So we want to give it back to them with guidelines to help them manage their health. And then finally, we want to create this ecosystem to encourage optimal health practices. So we give them their risk, we give them suggestions, but once again, they have to do something. So then this ecosystem is going to help encourage everyone to manage the risk based on the guidelines. Okay, so this is the Taiwan Precision Medicine array design. So we have 124,000 genetic tests on the array and 578,000 markers to cover the genetic profile. Now these profiling markers are actually from the data we obtained from the Taiwan Biobank and also some of the mainland Chinese sequencing data. So it's optimized for Han Chinese that will cover our genetics really well. And then the good thing about this test, unlike the dynamic tests like liquid biopsy and so forth, this is a static germline genetic profile, which has to be done only once in a person's lifetime. And the test is actually quite reasonably priced, and so it can be done for everyone in the world. Okay, so the consortium is with 16 medical centers, and as we heard earlier, Taichung Rongjong is the best partner in this project. You guys have already recruited over 10,000, over 10,000, 100,000. Yeah, over 100,000. My number is wrong. So over 100,000 participants to this program, which is very, very good. So we're actually working with your team to return the results and provide the best guidelines and see how it goes, because you are the example for all of the consortium. Okay, so to put things in perspective, so where are we? So if you think about all the big projects in the world, UK Biobank recruited 500,000 people, and it's already finished in terms of the data generation, although analysis is ongoing. The Chinese Kadoorie study, they also recruited 500,000 people, but it ended several years ago because they haven't followed up on that. And then there's other studies in Mexico that are mostly sequencing based, and then in the US, they have a Million Veterans program. They recruited 1 million people. So this is the biggest program in the world right now. They have genotyping data, and they have 1 million people. But once again, this is not a participatory program. They get the data, and they're thinking about how to get the data back to the patients now. And then the US All of Us program, which is the program that started right after President Obama made precision medicine an important part of healthcare in the US. And so they started in 2016, and their goal is to recruit 1 million people. But so far, they can only recruit 350,000. And they have samples, and they haven't actually sequenced them or finished the data collection. And then the UK has our Future Health program, which is the biggest program. They want to recruit 5 million people, but because of COVID, they actually just started in August, about 2 months ago. And we all know about the Taiwan Biobank. And then our project, we have already recruited 560,000 people. And so by that account, we are now the biggest project outside of US and Europe, and soon will be the biggest program in the world. All right, so now I want to tell you some science. So this is the curve that you have to remember for any kind of precision medicine for common disease. So this plot has 3 features that you have to remember. The first feature is that there is the highest possible level for genetic contribution for that disease. And so for this level is different from disease to disease. Some diseases have very, very high genetic contribution. Others have less. But no matter what, there is a maximum level. And then the second number is this axis here, about the number of cases. In order to achieve this level of prediction, you need a certain number of cases. Now, this number also is different from disease to disease. Some disease require fewer cases to get to the highest level, and some you need a lot more. And then the third feature has to do with the slope, how steep this is. Okay, so these 3 features determine how easily we can get to the number we want. And the number is what we call the AUC, area under the curve. And this tells you the signal for whatever feature you're talking about. So I'll give you an example. So for hypertension, the highest possible AUC is 0.66. And then if you, but hypertension is a really dynamic feature, so you actually need a lot of people to get to this point. So you need almost 300,000 people to get to the plateau. And then at this point in our project, even though we have so many enrollment, we looking at the first 120,000 people, we have only 32,000, which is a big number, right? But we're only at the beginning of the AUC. Okay, so I hope that you all remember this. This is really important. The maximal genetic level, the number of people you need, and how steep the rise is. Because the steeper the rise, the narrower the gap between the cases that you need to reach the highest level. Okay, so now I want to give you another set of results. And this is, remember we talked about using the UK Biobank polygenic risk score tool on Europeans. They get pretty good, right? So anything over like 0.6 is pretty good. But if we use the same tools for our patients, then it dropped significantly for these 4 conditions. And you have to remember that 0.5 is like no signal. So when you have 0.52, it's really, really bad. But the good thing is that when we use these samples, to train our own, on our own people, then suddenly the numbers get much better. So we have 39,000 cases for hyperlipidemia, and we get to 0.61 and so forth. So with this, I can assure you that TPMI will succeed because we will get the number of cases we need and we'll use it on the Han Chinese and we'll be just fine. Okay, so how do we move this from a project to healthcare then? So we need to first validate these scores so that the predictions are real. And then we have to develop really good profiling methods so that we can extend it to everyone. And then we have to get genetic profiles for as many people as possible. So it's not just 1 million. We want all 23 million in Taiwan, all 1.6 billion around the world. Because then we can build really best practice health management guidelines and pursue optimal strategy for risk mitigation. And then basically you divide people into the different risk groups. So it's like everyone has a QR code, and then a new person can just do this test and then match up with the QR code and know what their health is going to be like. So in order to do all this, we have built a return of results system for the hospitals, and then everyone can look at their risk profile and look at something that is in high risk, and then they can get some guidelines for that. Okay, so, and then we have done a bunch of educational things to explain to the public why genetics is important for their conditions. And for each gene, we have a story that is really easy to understand. And then recently, we have a very successful exhibition at the National Taiwan Science Education Center about precision health. Okay, so I'll quickly do 3 examples in like 1 minute. So if someone has a risk for stroke, then what do? We tell them to do all the things that mitigate stroke risk, and then hopefully it will lead to a healthy aging. And then if someone has lung cancer risk, we can put them through the low-dose CT scan monitoring program so that we can identify cancer before it spreads. And then finally, if someone has diabetes but has high risk for kidney failure, then we can do a very aggressive blood glucose control so that they will not have any dialysis needed in the future. So I'll end by saying that precision medicine is really something that we have to predict, be proactive and personalized. And then with the 3 different branches, Taiwan is actually perfect for the last branch, the common diseases. And then if we can identify people with high disease risk, we can use the best way to maintain their health and lead to long health span. And then finally, we want to profile everyone so that we can benefit all of them. So I'll end by showing our team, the leadership of the TPMI team. Thank you.
好，谢谢郭所长把这个 Precision Medicine 从开始到 TPMI 的建制到如何未来落地实施，造福我们的病人跟群众哦，做了很清楚的解释。我们还有一些时间，不管线上或者是在线上的各位贵宾专家有没有什么特别意见要提出来请教？好，请那个邱院长。
谢谢郭院士哈。您刚提到的，他会到了一个 plateau 哈。我的问题是，第一个，如果我们的环境的 environmental factor change，the population of immigrant changed，the contagious disease changed，and the lifestyle changed，do you need to renew the polygenic score system all the time, or you will just redo the TPMI?
No. So the genetic risk is static, but all the other things you talk about, the environment, lifestyle, and so forth, that's what can change. So what we actually end up with is not just the polygenic risk calculator. We have an integrated calculator that the person can say, oh, I exercise a lot. Oh, I never exercise. I eat potato chips, watch TV all day. So that will actually change their risk. So that's the promise of, if you can change your lifestyle, if you can change your environment, so instead of going out and breathe bad air, you wear your N95 mask all the time, and at home you have air filter, then you can lower your risk. So I think that's a really important point. But the last point you made, when we have a different population makeup because of immigration and things like that, now those people will not benefit from this score. Their population have to do their own reference PRS generation. So I think the method is actually straightforward. It's just having to do it. So, Vietnam has to do its own, Philippines has to do its own, and so forth. But I think we're lucky because Han Chinese is 1/5 of the world population. So we do it, and it's good for a lot of us.
好，不知道大家还有没有什么问题？我请教那个所长哈，就看起来 genetic profile， 你有想法是希望全台湾2300万民众有一天都可以出生的时候就 newborn 就可以做这个 profile 嘛哈，所以这个 newborn 出生以后，他就可以做一些 whole life 的一些 prediction。当然，这个 prediction genetic polygenic risk score 是不会改变，但是随着年龄 exposure， 它这个 risk 还是会做一些改变的哈。那我看到英国现在准备做500万人的 our future health。所以你从这一部分来看，我们台湾未来 genetic 除了2300万以外，还有没有其他 more forward 的这个 vision？
Right. So I believe that the first million will give us a lot already. So then everybody else can take advantage of that. But remember the ecosystem. So the ecosystem is that if more people can contribute their data to this central project later on, just like for our future health project, then we get more and more data, and it would be more and more refined. But I think the key is to remember not just the genetic risk, but also what we do with it, so that when someone is born with a certain risk, they can actually keep up a really healthy lifestyle, and that risk will come down. So I think that's why we need this integrated risk calculator, not just a genetic risk calculator alone.
还有一个问题，就刚刚你提到那个 TBMI 后来把 UK 的那个 polygen risk score 对几个 common disease 的 area under the curve 的 prediction 哈，不过那一部分好像还没有加进去，像 age 这些 exposure 的 factor， 如果这些加进去，会不会更容易到达它这个 area under curve 的 prediction？
Yeah, and that's exactly what we have to do. So we already know it. So if we have right now, when we don't consider age, it's about 0.6 or 0.65. But once you add the age, then it actually goes to 0.7 something. So I think it's pretty easy to add that. But the other stuff, the exposure and lifestyle, that is hard to add. So that will become an individual calculator that the patient can add that and see how it modifies. So it's just like the calculator for breast cancer risk right now that we can do. We can put in all these features and then it will come up with a risk number.
好，不知道大家有没有什么问题？好，来，请那位。
我想询问一下，一般的基因检测的报告，它有一些 homozygote 或者 heterozygote 的 DNA 的 mutation， 然后会造成不同样的 risk 的 profile。然后我想请问一下，如何去分析，比如说 high risk、medium risk 或是 low risk， 你如何去跟一些病人卫教？这是我想要知道的。
Yeah, so everything looks easy on paper, but we are sitting across from a patient. Then it's the challenge. So what we are talking about is not giving them a score, but we are talking about what is the relative risk compared to normal, right? So in the US, I think all women have a lifetime risk for breast cancer is something like, 8% or something like that. But if someone has a BRCA1 mutation, then it becomes much, much higher. So they have to make a decision what to do, right? So someone will do what Angelina Jolie does, and some people will not. And so we have to sit down and say, okay, based on this risk, this is our recommendation. So right now, with someone with a high risk, the recommendation is not to get, bilateral mastectomy, but screening at age 25. So then we can tell them, so based on your risk, this is our recommendation, but these are the other options. So I think this is something that we have to work together with the patients, and all the doctors have to, be willing to listen to the patient, because different people handle this very differently. But anyway, so that the key is to define, each medical society has to define at what risk then it's worth doing something different from the general healthcare guidelines, right? So the general healthcare guidelines is try to balance the cost and the benefit. But for some people with much higher risk, that shifts to a different point. And I think that's what we have to do.
我们李副院长，请。
OK， 我们的秦始皇2000年就在找那个长寿的人哈。那我现在也有一些长寿，我们在基因这边也有在做长寿的基因啊。可是这个 aging 到底是个 disease 还不是 disease？ 我看我们是做到 common disease， 可是这个 aging 来讲，我们是不是做了过火了？我们是真的可以把一个人把它做到200岁嘛？就用我们这个 genetic consulting 这样子。我想说，因为这个计划，台湾也有人在进行嘛，在全世界有人进行，不知道这个郭教授对这个看法如何
？Yeah, so I actually did some aging genetic research before, and the problem is that when you study people who are 100 years old now, all the stuff that we find will be when they were young. So the environment is so different now. So we may find genes that cause them to be 100 now, but for us, it would be a different set of genes that get us to 100. So I think that's something to remember. But the second thing is that there are some people who are very old, but also still very, very, with it and very healthy. And so that's all we want to achieve, right? So I think aging is kind of wear and tear is inevitable, but if we can prevent some of the disease that cause us to be unhealthy as we're aging, I think that's what we want to achieve.
我们虽然时间差不多了，不过有一个线上的问题要请问那个院士了哈。他就问到这个 TBMI 当然是 common disease 为主，那对 cancer 这方面如何去 extend？ 刚提到了一部分呢，是不是也简单的再提一下 TBMI 对于 cancer 这一部分 covered？
Yeah, so for TBMI, we cover cancer in two different ways. One has to do with the genetic testing that we are doing for some of the known cancer risk variants. So we do that. But the other is to predict someone who's going to have cancer or not. So those are the things that we can do to manage cancer. But we don't know anything once someone has cancer, what is the right target for them? Because that has to be done by sequencing the tumor. So sequencing is still very, very important. But in a population context, we are focusing on something else.
OK， 好，这个 TBMI 能够走到今天呢，很谢谢郭院士一路在后面重要的这个 leader 跟推手。其实 TBMI 也是面临非常多 difficulty 啊，跟 barrier， 那个大家的帮忙跟合作非常感谢。另外就是经费，哇，国家本来都说要出的，到最后通通收手不太出了。所以郭院士使出浑身解数，到处去募款，所以他口才也是非常好哦。帮我们募了非常多经费，不过我们要继续努力。我们用最热烈的掌声，谢谢院士的指
导。谢谢院士，谢谢许教授。那我们在就是 coffee break 时间，然后到总共有20分钟到10点50。那外面我们有备有餐点，还有一些饮品。那请大家休息一下，然后再记得10点50分再回到会场，谢谢。 各位现场的来宾，还有线上的来宾，那我们第2段的 section 要开始了。那我们现在先欢迎林口长庚医研部郭长福主任当我们的 moderator， 那还有线上的 Professor Okada， 然后当我们的 speaker。那郭教授请。
It's my pleasure to introduce Professor Yukinori Okada. Now he's in University of Tokyo, and Dr. Okada is specialized in statistical genetics, and he has published a large number of outstanding papers in the most prestigious journals, such as Nature Communication. And he started his career at the University of Tokyo Hospital and had a very good experience in Brigham Women's Hospital, Harvard, and also Broad Institute. And now he was appointed as a full professor in 2016, and making him one of the youngest full professors at the school. And Professor Okada is a pioneering researcher in the statistical genetics and has lots of outstanding papers and research results and contribute significantly to the field. And today he will share his research experience in statistical genetics, disease, and also disease biology and in population genetics. So let us welcome Professor Okada.
Okay, hello everyone. Thank you so much for inviting me and giving me opportunity to have a talk at this wonderful symposium. I truly hope to visit in person, but sorry, I have other conflicts of the meeting, so I'm sorry to say hello in mail, but hopefully definitely see you in person next time. So today I'm going to talk about my specialty of statistics genetics and also disease biology, drug discovery, and personalized medicine. So this is a theme of our research activity. So human genomes. So human genome sequence is a blueprint of humans. So if we can fully understand these blueprints, we can reconstruct human biology, life, everything. But currently, we only know the tips of these genome sequence. So we know genome sequence, but do not know what they're actually doing. So my thesis is, how can we decipher the blueprint and how can we extract the biology? And there is several strategies to extract information from human genetics. So using the latest omics technologies. So technologies are more rapidly developed than we always expect in both dry and wet technology. So implementation of such latest analytical methods is critical in data science. And also understanding the mechanism is necessary. So these figures show how the analysis system association study, DNA and RNA changes in the last 15 years. So everything is changing rapidly and keep changing. So we need to definitely catch up these technologies. Then, because advancement of technology, we are now in the era of human big genome data. So human genome project become bigger and bigger. And for example, UK Biobank has launched GBAT for exome 500,000 subjects. And lots of disease genetic study has been done for more than 1,000 traits. Let me introduce some of the achievements from our groups recently. So this shows how human genomic project became big. We have recently finished the largest and cross-population genome study with 5.4 million individuals from diverse populations. So this is a GBAS genetic association of adult height. And we finally show that around more than 3 million of samples, what we learn from genetics, or what genetics explained human height, became saturated. So the increment of increased sample size in the genetic study is now at some stages. So we are now showing saturated map of common genetic variants on GBAS SNPs. So we are now a little bit the final stage of GBAS and going to move on to the second stages. Another topic in human genetics is trans-biobank analysis. So this is cross-biobank studies of Biobank Japan, UK Biobank from England, FinGen from FinGen. By conducting GBAS meta-analysis of these three resources for more than 200 phenotypes, we identify so many genetic variants associated with these diverse phenotypes and underlying mechanism and etiology of diseases. So previously we have done GBAS for each of the disease separately, but now we are doing them at the same time and try to learn many things from the resources. And also we are doing some detailed in-depth study for some specific diseases. So this is our latest result from cross-operational GBAS for rheumatoid arthritis, one of the autoimmune disease with inflammation of joints. So we have collected more than 205,000 individuals from diverse ancestries. We identified so many things, but finally found that the majority of the genetic signal is shared between the populations. So anyway, what I want to say today is how to collect genome data and how to do GBAS. We already know how to do it. So this is not a challenging bottleneck of the science, but how to connect human genetics to biology, drug discovery, and personal medicine. This is just truly important, but yet unsolved questions. So to further these fundamental questions, we are doing several lines of humomics researches. So today, sorry, I think I missed to change the slide. I'm sorry, truly sorry. So this is the slide for the human height. You can see that the number of after increasing number of the phenotypes, the birth genetics can explain saturated. And this is a study for, Japan, England, and Finland, and we have identified so many things. We are now doing GBAS for massively parallel for many phenotypes. And this is our studies for rheumatoid arthritis. We have identified many things by diverse populations. So, but this is the important point. What is the important, how we should decipher biology and drug discovery and medicine? Okay, let me talk. I'm sorry, I have forgot the slides. So I'm going to talk on 3 parts. So COVID-19 omics analysis and human disease microbiome analysis and genomics-based drug discovery. And so this is a COVID-19 pandemic. This is in Japan in this month. So we have experienced COVID-19 pandemic around 2 years and find that everything happens in this pandemic is always unexpected. So it means that we still do not know what is exactly the COVID-19 is. I hope this will diminish soon, but still this is unsolved questions. But the human genetics has a power to tackle this kind of unknown thing. So because genetic study is a hypothesis-free approach, so even though we do not know COVID-19, we can discover something essential for COVID-19. So this is a GBAS for COVID-19 severity, mostly done by Europeans, which has been conducted around half a year later of the pandemic of initiation. So there was a strong signal at chromosome 3. So it means that we do not know COVID-19, but something related to the genes included in this region, like chemical receptors, have strong impacts on genetics of COVID-19. And you may heard that this locus is really famous, at least the last 1 week. So this GBAS region is derived from the archaic hominin-derived genome sequences like Neanderthals, and this is prevalent European but not absent in East Asian because of the different migration patterns of Homo sapiens and Neanderthal. And this is because famous Professor Pebo successfully got Nobel Prize this week. So, because this chromosome 3 variant is absent in East Asians, we need to make our own evidence for COVID-19. So we launched Japan COVID-19 Task Force which is a nationwide voluntary network for COVID-19. We have collected more than 6,000 individuals for DNA, RNA, and cellular, more than from 100 hospitals. And by conducting GBAS for these resources, we identify one genetic variant which has strong risk on COVID-19. And of course, we are doing so many parallels of doing massively different types of mixed resources. But anyway, we found one region here. This region was implicated in the DOC2 gene. So DOC2 is an immune cell-specific expression profile, so immune genes. And this regulates innate immune response in RAC activator and chemokine and lymphocyte activation, macrophage migration, and typhoid interferon production. So this is definitely involved in the core of COVID-19 severity. And the recessive DOC2 deficiency is a Mendelian disorder associated with combined immunodeficiency. And also DOC2 is decreased expressed in both in the COVID-19 patients. So DOC2 is really implicated in COVID-19 severity. So if we need to show two things. One is DOC2 is really expressed in the COVID-19 severe patients. And the other is when we perturb DOC2, when COVID-19 becomes severe, it's nice. So we need to show these two lines of evidences. And then to disentangle these new challenges, we use a new technology. So new technology always gives us new science. This is single cell sequence. So here's the difference between conventional bulk RNA sequence, including everything like vegetables smoothie, and single cell sequence, we can pick each vegetable separately before smoothing up. So anyway, single cell RNA sequence is a powerful technique to tell us new life sciences. So we use this technology to these doctor challenges in pandemic. And here's the result in the doctor single cell data. I'm sorry, it's still in some Japanese, but maybe your friends in Taiwan can somehow understand Chinese characters here. So anyway, when we focus on innate immune cells, DOC2 is centered on CD16 high non-classical monocytes. So they are involved in interferon responses. But their dosage is strongly decreased in severe patients. And if you have DOC2 risk genetic variants, the people who have variants get decreased DOC2 level in non-classical monocytes. So we show the several lines of evidence that DOC2 is really repressed in the severe form of COVID-19 patients. And we also then showed another line of evidence, so perturbation of DOC2 by using DOC2 inhibitor, CPIPP. And here's a Syrian hamster model, losing weight and recovering after SARS-CoV-2 infection. If you put CPIPP, DOC2 inhibitor, weights keep lowering, and the lung became really edema, weight loss, lung edema, enhanced viral loads, impaired macrophage recruitment, and dysregulated type 1 interferon responses. So anyway, DOC2 deficiency is strongly linked to causality of COVID-19 severity. So I think the situation with COVID-19 changing, but this type of approach to identify people who have higher risk in these challenging diseases should contribute to how to make a precision medicine in upcoming next phases of pandemics. Okay, let me go into the next talk of the human disease microbiome analysis. So many lines of humomics information, clinical information, genetics, abriomics information can contribute to personalized medicine. And I'm going to specifically want to talk on microbiome analysis. The microbiome is a genetic material of all the microbes that live on and inside the human body. And this has a strong biological interaction between host, and they are considered a new organ in our body. And they have played essential roles in a variety of human diseases, including metabolic, psychiatric, and autoimmune diseases. So we have so many microbiomes, they can change mass, fat, or lean. So they are strong risk and maybe a key player in personalized medicine. And here comes the new technology. So new technology always tells new science. Previously, we used 16 S ribosome RNA G sequence. This is really easy, simple way, but the resolution of the microbiome is really not so highly clear. So then here comes the metagenome shotgun sequencing technology. This sequences all the genomes in microbes. And this has a little bit difficult scales and computational analysis, but they provide the highest resolution, species level information, genes, pathways, viruses. So new technology is better than older ones. So we definitely started to use these technologies. And here is a result of the microbiome analysis between cases in this type, rheumatoid arthritis and controls. So this is the result of metagenome association study. So each circle represents each bacteria, and the outer layer shows a final resolution definition, and color represents the case control ratio, and circle size represents dosage. So you can see that L7 species level. Of course, each of the dosage is very small, but you can find several species which are strongly increased in RA gut microbiome RA patients. I found that majority of RF-associated clades belong to genus Phlebotera. And this finding was replicated in other cohorts. So we learned that RA is a disease of joint inflammation, but we have a new knowledge. This is, if we want to fully understand RA, we also need to understand microbiome. And microbiome is associated with whole body things. So this is the metagenome and the blood metabolome association test. So when we do metagenome association study, we found that streptococcus is increased in gut microbiome patients and SLE patients. Then we tried to decipher how these bacteria is doing. So what happens when bacteria increases? So we did metagenome association test and found that the people who have higher streptococcus, so SLE risk taxa, are associated with increased risk of dosage of acetylcarnitine. So what happened in our gut is also strongly linked to what is happening in our blood. Then we found that we need more detailed catalog of microbiome. So we constructed a Japanese, and I think this really shared with you, Taiwan friends, the population-specific microbiome genome. This is MAG. So Japanese metagenome and Japanese virome. And we identify 20,000 prokaryotics and 30,000 virus. Anyway, we need many things in the gut of the Japanese populations. So one interesting findings was, in our gut of Japanese, we found population-specific antigen, the Bacillus subdices. This is a natto, fermented soybeans. I'm not sure how you guys eat, but we Japanese really like to eat these fermented soybeans. And also nori, nori labor. So B phosphorus. So these population-specific dietary related bacteria and their product is also strongly linked to our guts. And also, because of our data, we can see more detailed analysis in viruses. We conducted class-like phage subfamily analysis. Class-like phage is one of the phages which affect bacteria. And we have now so many catalogs of these phages and found that this class-like phage is strongly associated with status of autoimmune diseases, rheumatoid arthritis, SLE, multiple sclerosis, ulcerative colitis, Crohn's disease, and also diversity in our gut. And they have a strong target bacterial connection. Also, they have a strong high-dimensional network in our body. So our bacteria has a strong diversity between individuals, and this should be a key of personalized medicine. So here comes the idea of personal medicine, especially using genetics. So we can calculate genetic risk of individuals for each disease separately based on the genetics of individuals. So this is solely called polygenic risk score, PRS. Especially for the diseases with high prevalence and age-dependent onset, something like type 2 diabetes. So stratification population subjects based on PRS at the younger age is effective for personal medicine prevention. And PRS is not only the things to predict our personal medicine. In addition to genetics, PRS, we can include microbiome or other things. So many lines of omics information can help us to finally identify and predict individual risk and life courses. So this is, I think, definitely important in personal medicine. And also by using these predictive things for our biomarkers, we can assess what is important for predict clinical outcome. So clinical final outcome is our most interest. And this PRS can be an instrumental variable to infer causality on clinical outcomes. So we conduct a transbiochemical PRS analysis and identify obesity and high blood pressure is a strongly but modifiable risk factor for longevity. So by improving blood pressure and obesity, you can extend your longevity. So maybe the personal medicine not only predict risk, but provide how to solve this problem is also important factor for them. So, and then how to predict humans. So here are so many genetic resources. Currently, we are doing one by one prediction from some type of genetics to some genetic output data. So this is so-called unimodal deep learning. But now we have so many lines of omics information and output data. So multimodal deep learning. So using multiple lines of input data, genetics, omics, and we can assess so many multiple output estimates. And then we can estimate what human life is being in person. And finally, this kind of approach should be done by trans-ethnic and trans-biobank initiative. So not only researchers, or not only one cohort researcher can do it, but some international coordination can promote these sciences. So by cooperation with many biobanks and many participants and feedback, we can now achieve personal medicine. I think this is one of the needs for some international society, something like International Commodity Alliance, which I'm now in the executive committee. If you have any questions or interest, please visit our website. Okay, let me finally show the drug discovery. So prediction is important, but another important is how we can do, or which we can do for after prediction. So this is drug discovery. So this is a famous figure, how drug discovery is difficult. So to launch one drug to the society, so many clinical trials and target hit failed. So we need some change in drug discovery. So I think it should be a genomics-driven genomic drug discovery. So for here, the disease, genome, and drug, from disease to drug. Here's the traditional drug discovery. It's totally fine, but currently not fully enough. We need more another way. So using disease sample to get disease genetics and directly find drugs from genetics. So this is genetics-driven genomic drug discovery. Now this concept is in total supported by worldwide. So it's an interesting paper. Among the 50 drugs approved in FDA last year, 2/3 of the newly approved drugs have the support of disease genotype-phenotype associations. So disease-human genetics is definitely a resource for drug discovery. And high proportion of the approved drugs with support from disease genetics, so common disease genetics. So it means that GBAS. So common disease genetics, GBAS is now the strongest resource for drug discovery. So we should definitely develop a way to extract drugs from GBAS. So this is one of my old studies. So showing disease risk genes are rich in drug targets for the disease cell. So disease risk genes is not namely a resource for drug discovery. So genomics-driven drug repositioning. So drugs directly targeted by GBAS and these risk genes are candidates for drug repositioning. So previously we are showing this figure by collecting line by hand, but by hand is not good. So now we are making new automated methods. So something like MyGBus. This is a microRNA gene network enrichment from GBus. So find biomarker from GBus. And GREP is this is an automated repositioning drugs. If you put GBus or any kinds of disease-related gene list, you can get the list of the candidate drugs and candidate repositioning phenotypes. So automation of these genomic-driven drug discovery is an essential part to accelerate new drug discovery. And we are now expanding our strategy more complicated and biological way. So previously we have just shown, we are seeing drug discovery based on simple overlap between risk genes and target. Here this target between overlap between risk genes and target, but we didn't consider directional concordance. We are now using in silico drug screening which concerns gene expression, dynamics, and directions. So something like negative correlation in gene expression profile in cases controls, and also gene expression changes by compound. If there is a pair with some negative correlation, this kind of compound can cure the diseases. So we use human genetics to infer gene expression changes between cases and controls. So we have recently developed a new software transfer, a next generation dynamic stream in silico drug discovery. So transfer utilizes GBus and EQTL data to infer gene expression profile, and then going to find the compound with negative expression change profiles. And this software surely can pick up some interesting candidates for drug discovery, something like a verapamil blocker for hepatopoietic cell expression for COVID-19 severity. Anyway, this type of gene expression profile-based method is also effective for genomics-driven drug discovery. So we have launched several lines of best practice how we should do genomics-driven drug discovery. So we have proposed several lines of drug discovery methods. So in other words, no single drug discovery method is the best, but complementary doing multiple lines is essential. So first is simple directly disease gene and target overlap. And second is endophenotype QTL and minimize the randomization analysis. And third is case control gene expression changes by TWAS. And these methods tells us similar but not completely different, not completely same result. But this should be a good resource for drug discovery. And what was nice for us, these methods, we have some collaboration request, and here is a stroke genomics-driven drug discovery. So here's a collaboration with stroke genetics, and our method successfully pinpointed several candidate compounds, which is hopefully good candidate for drug repositioning for strokes. Okay. But anyway, my next step is from in silico to in vivo and bedside. So it is easy to say, oh, this drug may be effective for this disease computationally, but computationally effective cannot cure patients. So we definitely move to in vivo and bedside. So we are now tuning up the methods and also doing in vivo experiments with collaboration, patient-derived iPS cell, these model mice, and finally, I want to provide some drug finally to the bedside and suffering patients. Okay, this is my last slide. So, and also, what is most important is develop a young scientist. So if talented, gifted young scientists join this field, actually everything goes well automatically. So developing young science is a really important factor. So I'm now having a summer school of statistics genetics in Osaka University. This is a 3 days course for the targeting the students and doctors who have not the experience computations. So we showed statistics, Linux programming, statistical tool, and genome data analysis with 3 day course. And We have recently started a 1-week course for computation, bioinformatics course for the Osaka University Medical School. And this year, I'm going to start the same one at the University of Tokyo. So anyway, I think the best analysis can be done when the people who own the data do the analysis by their own. So this summer school is online, so everyone in the world can join, and all the resources, slides and data and commands and scripts are publicly available at the website of our lab. So if you have some interest, please join it. Okay, finally, these works has been supported by so many collaborators and so many participants of the biobanks. So I would like to say thanks to all of the participants this collaborate. I'm sorry, I can't make names of all the collaborators in this slide, but I always feel great thanks to the people who contribute to our project and sciences. And thank you for your listening.
Okay, many thanks for your comprehensive review on statistical genomics and also the outstanding results, research results on the disease association and also on the drug discovery. So now we have 10 minutes to open to the crowd.
Thank you, Professor Yuki. I'm Dr. Chen, and thank you for accepting our invitation. I want to thank you in person. And for the talk, you mentioned a lot of transbiobank studies. And now in Taiwan, we have the Taiwan Precision Medicine Initiative. And the director of the Taiwan Precision Medicine Initiative is at the floor at the present now. So I think maybe in the future, we can establish a trans-country, trans-biobank collaboration between Taiwan and Japan. And I will write to you in detail. And I have one question for you. In your previous talk, you mentioned about the nori and the natto for rheumatoid arthritis. But we know in the study of metagenomics, there's always a causal relationship questions, whether this microorganism lies in the gut, they are causal organism or not. How do you deal with the potential bias of the diet and the genomics and the phenotype? Thank you.
Yeah, okay. First of all, thank you so much for giving me a chance. I truly feel sorry cannot be able to in person, but definitely I agree to discuss and meet and collaborate in person in the near future, and also in cross-population, cross-biobank scale. Truly thank you. And also, yes, my answer for your question, microbiome, yes, this is really important question because things in our gut is very contaminated. So when we do this type of analysis, actually one of the important points is to clearly extract the genomes or DNA sequences from fecus, not directly from what we eat. So this is one of the important step of experiment, but maybe can be do it somehow. And another important problem is contamination of human-derived genome and these bacteria-derived genomes. Actually, we think there should be more things we can do to it, but I think once we get a fecus sample with unified and qualified method, we can somehow now extract information more specifically connected to microbiomes. But of course, we are mostly seeing bacteria and part of the virus, but not currently seeing fungi. So different types of DNA extraction is requested if we want to much fully check the these microbiome or circumstances. Yes.
Any other question from the crowd? Okay. For Okada, I have one question because I'm a rheumatologist, but also doing lots of study on deep learning. And I know you have employed deep learning methods into genomic discovery. And I'd like to ask your opinion on applying deep learning to the genomic discovery in comparison to the traditional statistical method. Because we know that deep learning is a powerful tool, but sometimes it may detect very true detail of the signal. So that may have increase the noise. Also dictate the noise that probably will confound the final results. So what's your opinion on the application of deep learning in the genomics? Thank you.
Yeah, this is a truly important question. So wait a minute. First of all, I think deep learning is only one of the solutions. So not always best, but sometimes best. So what is important for us is which kind of problem the deep learning should do more than the conventional one. So we don't need to stick deep learning always, but I think there may be several situations when surely deep learning is better than the traditional. Wait a minute, I'm now trying to looking for my slide. I'm sorry if there is any. Yeah, here's the goal. So I think this is what I think how we should apply machine learning to human genetics. There should be 3 stages. The first is unsupervised machine learning for population genomics to just extract core information representing genomics to use unsupervised machine learning. I think which have been already done. The second point is supervised deep learning for genomic sequences. So deep learning is very effective for extract specific protein genomic sequences. This is because biologically, transcription factors, RNA polymerases, these things only recognize the limited sequence of human genomes. So it means that information from specific part of the human genome, if we know it, everything we can do it. So in this case, deep learning is very suitable. And the final stage is supervised deep learning for population genetics. Because our genomic sequence can be converted to something like figure of the genome sequences, we can apply several types of deep learning, something recursive or convolutional to human genetics. So there should be several lines of how we apply deep learning. But I don't think deep learning is solely the best method. Sometimes it does not do well. So maybe just think deep learning as one of the technology to utilize genomics. And then, anyway, try and error.
Thank you. Any question from the crowd? Another question is about the cause effect reasoning using the genomic data. And now we know that most of the current method is to classify association, but not the causal effect. And I noticed that you use a Mendelian randomization method in the drug discovery and in silico prediction for drug discovery. So do you think that is an essential part to infer this association and also the important drug discovery?
Yeah, definitely. Yeah, actually, distinguish between causality and correlation is really important, especially when we do something related to causality. So I agree, Mendelian randomization on several types of the statistical analysis utilizing summary statistics can be a good method to infer causality. But I think the final purpose of showing causality is to decipher what is going on in genomic sequences. So if we can understand exactly the causal roles or functional role of each genetic variant, I think this shows causality. So understanding of functional roles of each genetic variant is essentially to finally understand causality in biology. So in this case, we are now constructing a genotype phenotype catalog, not only with disease, but as a more omics resources, sometimes within cell or within blood, like metagenome, metabolome, non-coding RNA, single-cell sequence, editing, anyway. But if we fully understand what genetics is functionally doing in the phenotypes, this should help to understand the causality of diseases and the phenotypes.
Thank you, Professor Okada. Because of time limit, I have to close this session. And thank you, Professor Okada. Thank you very much.
Okay, thank you guys, and see friends in Taiwan. Hopefully to see you in person next time. Thank you.
继续下一场，那我们下一场欢迎台北医学大学袁云教授，那speaker， 然后 moderator 是欢迎陈立忠讲座教授，请。
首先谢谢大会的邀请，让我有这个机会来这里主持袁云教授有关于 Precision Oncology 的一个 talk。那我很高兴有这个机会可以介绍袁教授。袁教授应该是台湾最早期的 medical oncologist， 不过他整个 training 跟他的 career development 一开始都是在美国。他曾经担任美国 LA CD of Hope Comprehensive Cancer Center 的副院长。在2011年被台北医学大学邀请回台湾来担任校长。那他做了2任以后，目前仍然持续是台北医学大学的讲座教授。他同时也是中研院生化所的兼任研究员。那他在这个整个的 career 里面，因为他对药物的发展的熟悉，然后对于整个 oncology 的 course 的药物的发展也真的是非常非常的清楚。那所以今天我们很高興有他來跟我們介紹 Precision Oncology from the target therapy to cell therapy。那袁教授請。
Yeah, thanks Leo for your introduction, and also thanks Taichung General Veteran Hospitals this kindly invitation. And today, I just put it a little bit more like a review rather than data presentation. And so that should be very relaxing talk. And this is my biography, and that's pretty much cover all my career life. Yeah, and I have no conflict of interest, and this is my disclosure. Yeah, because lately I involved more biotech, so you can see something there. It's all new. Yeah. And today I'd like to present, just follow this outline from the Genome Project, and then target therapy, and then to the cell therapy. And this is pretty much a general slide to cover all past 50 years to 100 years about cancer medicine. And the first top half of the panel is a talk about around the before World War I, and then continue to discuss the mustard agent, and then a real small molecule chemotherapy agent, for example, such as hydroxyurea. And start about 1970, which is the second half of the panel we show here. The thing is changing. It's not just have a more of significant, powerful small molecule chemotherapy agent, and gradually in about near to late 1990 and to 2000 around, we have more monoclonal antibody, and then into the large molecule, then combined with the immunotherapy, and nowadays cell therapy. And everything accelerated very fast since 1970. That's all because the United States former President Richard Nixon signed the Cancer Act, and that facilitated the whole cancer research as well as the therapeutics research. And this is historical photography presented here. And the further jump is happened in the early 2000. That's Bill Clinton, again, another former president of the United States, and worked together with such as Francis Collins and others, and established the Human Genome Project. And this is very critical because the Human Genome Project is such a big program that's not just to cover the United States, and actually is all over the world. And another important point is, at the beginning, they tried to establish that for commercial use. But after the serious debate, they actually opened that to the public use. So through that, and the thing is changing and it moves very fast. And you can see this is a historiological milestone. And the real precision medicine is actually launched around 2015. So we have about 10 years to 15 years the time. We are wondering about how we used this genome study and how to align with a different genetic background together with ethnic background, and what is normal, what should be called disease. And that takes about 10 to 15 years. And around 2015, and we start to learn from the first disease, cystic fibrosis. This is in this whole world, humans start to using the Illumina to make the diagnosis in a single point mutation on this cystic fibrosis lung disease. This disease is common in North Europe. And from there, then start to moving from the single point mutation gradually to cancer. And cancer is not the first disease to been discovered by using these the genotyping and as well as the genetic diagnosis. That's because cancer is a complicated disease. Cancer usually involves multiple genetic mutation rather than single mutation. But the changes are ongoing, and breast cancer, for example, I'm using that to show you is we don't call it breast cancer no more. We call HER2 positive cancer, ERPR positive cancer, triple negative breast cancer. So we put the molecular marker first, then we talk about breast cancer, the name later. And that is primarily because the molecular marker is critical. And that makes the decision about how we should treat this patient. And if you know this molecular diagnosis, and then quickly you know more detail about how to use the treatment. For example, this slide to show you is a BRCA mutation, BRCA1 mutation. So the treatment for this case is olaparib. Olaparib is a PARP inhibitor because the BRCA mutate, the PARP increase elevated, so we use PARP inhibitor. And this slide just to show you the efficacy is significant and fast. And that is just one of example to link genome and therapy together. And 2015 is an important year. United States former President Obama announced that the precision medicine era is coming. And then there's a following several acts, such as All of Us, such as the Moonshot program, etc. So what they try to do is to using the whole United States as a power and the centralize lots of the ability together, and then gather together with the other countries. For example, in Taiwan, Academia Sinica is leading this project together with the United States. And not just the genome. We start to realize the genome itself is not enough. We need to put more. So we have a longitudinal cohort studies, and we try to collect 1 million or above participants. And now it's already past 1 million. And I saw Dr. Kuo Guopei here, and he's the leader to Taiwan as well, to do the very similar things. And electronic health record also need to be considered, biospecimen survey, and more important is that gradually we start to realize that genome itself is not enough. We need environmental. Environmental is not enough, and we need to add in more like a behavior data. So the data become larger and larger. So biobank become very critical. Lots of the biobank formed by nation, but gradually, for example, United Kingdom, they released that to the industry, but they work as a nonprofit. And so that's open the biobank and with all the data to help facilitation the overall collaboration. I think this is so critical to move this whole genome medicine go faster. But it's not always this smooth. In year 2017, and FDA said no, and because they are reluctant to let the genome study data being used in the commercial. They suggest to not to let the vendor to leading that. So directly from the test to the patient, that is something be concerned by FDA. They still prefer the fee for service, and you go to the doctor's office, you get a blood test, and then or the tumor, you get the mutation diagnostic marker identified, and then let the doctor to answer to you. But the problem is that most of the doctor at that time say, I have no idea about how to explain this data, because I've never been trained in genome medicine in our medical school, and I don't have an in-service training as well. So that become a difficulty. So reality is, in 2017, and FDA go a little bit backward to allow lab-directed test and also the test directed to the consumer. But this is supporting supplement data to the doctor. That means everybody work together, patient as well as a physician. But that's reality, because with that open-minded setup, things go actually faster. For example, under that kind of guidance, lung cancer become no longer be a lung cancer or no longer be a non-small cell lung cancer. They are based on the mutation. They find out KRAS, EGFR, EML4, ALK fusion DNA. That's also critical because fusion DNA is hard to identify at the beginning, and you need a special technology. But that fusion, the protein and the genome have been identified, is accelerated the treatment outcome. So that makes the lung cancer treatment go to another era. So nowadays, we use EGFR positive lung cancer, ALK fusion protein positive, or many other. But some markers we identify from non-small cell lung cancer, such as BRAF, HER2, still not that being understand well. What's the role in the lung cancer versus what the role in the breast cancer. And also the treatment, the therapeutic modality carry out different outcome when we use it as the target therapy in lung versus in breast. Yeah. For example, BRAF is one of example, and BRAF is critical in many cancer, but BRAF has designated treatment, vemurafenib. But this therapy in colon is not quite good. In lung is so-so, but in thyroid cancer is excellent. So this is not just a target. It's not just a target-driven disease or therapy. It's also related with the field effect, also related with the disease microenvironment, and that's been known. And gradually, because the target become critical and the genome study being accelerated so much, lots of druggable target exist. Before it's called non-druggable. RAS gene, always say, mutated RAS gene is not druggable. But now there is the Amgen drug, the Amgen 510, which is a specific target on RAS. RAS itself, physiologically, is a GTPase. So there is a function. But if there is a mutation, primarily G12C mutate site, and then that make the specific motif changes, and then bind together with the MGen510. With that, they become efficacy. But the question here is, how about G12D? And G12D is also a significant number as compared to G12C. The G12D, however, is less significant in responding to this specific drug. So there's much we need to learn from that. And lately, because this genome study, and also because lots of undruggable become druggable, so things moving very fast, and people start to looking at the target on E3 ligase. It's in the proteasome pathway, and E3 ligase has never been called druggable target, but now become druggable. So the target therapy and precision medicine leading to a new era because that. And the PROTAC, the proteolytic targeting chimeric technology, is blooming. In the past 2-3 years, you will see the publication become more and more. And that's because the new drug, the target on E3 ligase. But the E3 ligase is about like 600, 700. How can you determine you have a specific target to the E3 ligase? Reality is they don't need to let you have a very specific binding. They are actually pooling. They are not inhibitor. They are moderator. So they don't block this E3 ligase. They just want to E3 ligase go closer to target. And then they throw the ubiquitin to the target. So after target labeled by ubiquitin, and then the proteasome system turn on, and then start to degrade. So the degradation is not based on the inhibitor. It's just a modulator. They are pulling the E3 ligase and the target together. So such a unique way. So this is the future, and we have lots of the small molecule which has never been heard, never been tried, and they start to target on these E3. And of course, after the Nobel Prize winner being announced a couple of days ago, people start to look at the click protein, whether you can use that to the E3 as well. So this is PROTAC drugs, which is new to most of the people here. And this is blooming, some already into the phase 2. And the design is not traditional. Not traditional, go by the drug screening. They go by the E3, and also they not go by IC50. So you can look at all the detail, the literature, but I don't have time to talk about detail, but that's fascinating in the target therapy. So this all above are small molecule, but how about the large molecule? From 1997, the Rituxan started. They target on CD20 in B cell. And then start from there, they find out more and more Rituxan. That's such a powerful drug, can bind to the CD20 positive B cell. More than that is not just a binding. It's not just the antibody. They actually have the complement conjugated, the ADCC, CDCC, the effect all there. Year later, 1998, Herceptin started. The HER2 gene overexpression have been known at least about 20 years before 1998. However, dimerization of the receptor become a binding with the HER2 blockage become so powerful in control the breast cancer. So again, we and also understand this is not just antigen antibody binding, they also trigger ADCC, CDCC. So with all of that, people start to know that immunology actually played a role more important than just protein binding. And from there, there's more drugs to coming out, and not just a large molecule. It's almost all the small molecule and the large molecule conjugate together. For example, TDM1, and so they are using small molecule and the large molecule combined. That's what we call ADC design. And also there is it's a bispecific antibody design, so that become BiTE. So with all of that, it's all based on the large molecule and make a more design. And the key is to block this signal. So things are changing, and just about last week, the HER2 positive also divided to low positive and high positive. We use Herceptin to treat it with high positive. But how about on immunochemistry standing? There's a weakly positive. What should we do? There's a new drug to target on the low positive. That drug is a Herceptin combined with small molecule. So you will see that more and more and more. And colon cancer is similar. We have the cetuximab, and we have panitumumab, and that's EGFR, and then we have also been combined with the RAS gene targeted drug, the MGN510. So that is changing the whole treatment paradigm as well. But one thing cannot change it is we cannot make the survival be 100%. That's because apparently when we treat the patient, more specific targeted, we may not touch that the original the tumor heterogeneity. That tumor heterogeneity issue continue become our challenge, and then you suppress one clone, you actually help the other clone. So, and also there's a redundant signal, also a barren signal, and also the some rescue signal, all can come together. So that can make the resistance go on and on. And the NCI guided to lead everybody work together, that including Yale, MD Anderson, the MGH, and Mayo Clinic, and say, well, I think that all the variation between the target therapy and the outcome may be from the tissue. You are not prepared very well. So they use the standard protocol, collect everything the same, but the result is terrible. And most of the large cohort will end up with that because so many people and so many mouses talk back and forth and finger pointing. But reality is we learn a great deal from this unsuccessful clinical trial. That is, immunology really played a role in the microenvironment. Nowadays, after Nobel Prize winner from Dr. Hongzhou and Jim Anderson, people think, that's very clear. Then we have a PD-1, we have a PD-L1, so we have lots of inhibitor. But reality is there's more than that. It's more than PD-1. There's so many, for example, like a STING, etc. So they still need lots of study. Another challenge is these antibody target on PD-1 or PD-L1 so far vary the result quite a lot. Some are 20%, some are 40% benefit. We have a good long tail, but seems like over 40% is still very, very hard. So something we need to improve more. And this slide, every time I show this slide, I feel it's out of date because so many antibody come up. Yeah, so we need to learn, not just the target. We need to learn from the genome target. We also need to know epigenome target. We need to know pharmacogenomics, metabolome, etc. So we need to learn so much. But from 2017, things is changing again, to the CAR-T world. Large molecule let us lead to understand the immunology background. And the immunology drug help us to guide for further improvement. And the CAR-T is the one. And we know the B cells, leukemia or the the chimeric T cell will target on the leukemia cells with the CD19 positive. That has been known, and that's why we are using that CD19 as a guide to target to the tumor. But at the beginning, it's not very successful because it's not enough. So further, we chimerically antigen receptor to stimulate the T cell, and then using the T cell to be our treatment. So that's in the body. We give you the opportunity to grow your T cell, awake your T cell by several steps. We take the T cell harvest, and we use the lentivirus to carry in the CAR and reconjugate chimerically, and then go ex vivo expansion, and then go back to the patient. It sounds like so simple, but every step is very, very difficult because most of the patient, they don't have enough T cells, or they don't have enough functional T cell after so many chemotherapy they got previously. And second, the lentivirus, the yield still not that high. So people try to use the different methods. Some are still using retrovirus, even though they do have a concern, but the AAV is still not big size enough to carry the gene. And expansion itself is also very difficult because they don't grow very easily and very well. And overall, all of these need to be done under the GMP, and that's very costly. So that limit the CAR-T development. But a good outcome still is so strikingly moves the whole field forward. And this young lady, Emily, got a severe disease, leukemia, and then have a CAR-T treatment, and in 23rd day, it's already show completely cure based on the flow cytometry study. So the CD19 become most critical marker, since that is overwhelmingly expressed in the B cell malignant cells, but not in the normal B or the hematopoietic stem cell cells. So they become a great marker. But not just that. Nowadays, we also have using the BMCA, B cell membrane antigen, and we have many others, like even EpCAM is also being considered. So FDA also encouraged the cell therapy growth, since this complete new target therapy era, from the small molecule up to the large, up to the immune, and then now is a new chapter. The CAR-T therapy can also not just use in cancer, but also in many other, such as genetic disease, etc., and rheumatological disease is another way. But then we try to simplify this CAR-T to see whether we can make it simple. So the more and more simplification steps has been done using a single chain variant fragment to be a key, and then be using amplifier, and also reduce the adverse effect. So there's a 4th generation now. The 4th generation is the key, is to show strong efficacy, but not the adverse effect, the cytokine storm, as what we say here. And also, we can use a less number of cells, since this is still a hurdle. Yeah. All the detail I cannot explore that much in today's talk, but so far you can see, based on all the encouragement from the FDA and all the support from the industry as well as the government, we have 6 CAR-T drugs available now. But look at that, they're all in a hematological cancer. It's not on the solid tumor disease. That's because our T cells, the cell, the yield, the specific, is still not that enough. And also, you have a CD19, such as specific for B cells, but we really need a more tumor-specific antigen being identified. And so we need a more selection from the solid tumor versus the hematological cancer. Otherwise, you will stay in the autologous forever. And if you want to do cost down, you have to go by allogeneic. If you go by allogeneic, and you have to identify tumor-specific antigen. And with that, let's go back to target therapy, generally using the sequencing, identify all the changes on the tumor. So this is the final slide, and target therapy moving fast. In the past 30 years, just never been stopped. Now it's the cell therapy, and it looks like in the next 10 years to 20 years, we are going to dealing with the CAR T, but gradually we start to see they're moving from the hematological to the solid tumor. These are all on the clinical trial. And also we started using non-viral transfection, which is also very critical. The company called Proseda is working on that heavily on the phase 2, and it seems like have a relatively good result from the just 2 weeks ago, the Boston symposium, CAR T symposium there. They disclosed partially, yeah, but it's still very costly, yeah. So we have to reduce the cost and then make it really affordable, yeah. Thanks for your attention. Yeah, I try to move fast, but still not fast enough. Okay.
我们谢谢严云教授。当然他不用担心说他时间讲的比较长，因为我们配1份的 ticket， 那我们听到2份的演讲这样。所以不晓得大家有没有什么样的问题想要问严教授？这个是他不容易有这个时间跟这个机会，他现在生技公司越来越忙了。
对，那个先问老师，那个想请问一下，就是比如说现在因为越走向cell therapy， 那但是其实我们还是有很多 cancer 的知道，比如跟它相关的 driver mutation， 其实没有它的药物的发展。那到底目前来说，还有哪些可能的small molecular 的 target 是可能目前有机会被发展出来治疗我们的肿瘤的部分？
其实我们use Chinese Mandarin， yeah， 其实我们被大的药厂立的，把一些药做出来，有了 positive 的 clinical trial 以后，我们就停了。那实际上来讲，我们还有好多同样的药，但是它可以 target 在其他的 mutate gene。OK， 反过来说，有一些的 mutate gene， 事实上也有一些老的药，难道它不是 target 吗？它也是 target 在 DNA， 这个 replication， 它也是很 specific 的，也是可以把它带来。所以在我们花很多的时间跟金钱去寻找这些新的 mutation， 还不如先把老药也可以新用在这些 mutation 之上。另外就是 sequencing， 就是我们现在的 sequencing 只是把它identify 你的 mutation， 然后报告，然后寻找药。其实这个是大概只用了我们那一份 data 里的 5%。我们有好多的 mutation 都被报告里 cover， 但它是放在最后一页啊。那些也非常重要 ，it's very important， 因为你在那里可以找到基因跟基因的 interaction， 所以你的 target 也可以从别的基因上找过来，去打你现在的基因，这也是一样的。
谢谢校长我本身是糖尿病科的医师，所以 tumor 方面对我比较距离比较远一点。不过我们 common disease 最近都在谈用 polygenic risk 的方式，不管是预测疾病或是预测它后续的 complication。那 tumor 也是一样，它的 specific gene 可是也有 harbor 其他就跟您说的 unknown 或 unidentified 的 signal。所以有没有类似像 polygenic 的这个 score 的方式来 determine target therapy， 而不是 specific for 不管是HER2、KRAS 等等？
确实也有啊，在那个像很多人正在做的就是一些 non-structural 的 variant， 也就是它没有什么重大的改变到这些 mutate 的基因作为 drug target， 但是他们发现有很多 non-structural 的 variant， 也就是像我们从前说的 polymorphism 等等，其实它也是非常重要。在这点呢，已经给我们 hint 了，就是在这个 PD-1 的治疗上 ，HLA subtype 是 critical， 但它的 EO outcome 是有不同的。所以像 diabetes 还有一些很多的 common disease，including Alzheimer 等等也是一样。所以我们今天的这些 biobank 是一只 treasure， 我们一定要好好的去用它。但是你要用它的时候，就一定要大家一起，要公开，然后要大家能够共襄盛举啊。那这里面呢，我觉得我们医学界、生物学界的问题不大，大家都很愿意合作，倒还是法规面的问题了。也是一个很 critical 的 IRB 就可以把你折腾死了，或者一个法律的一个 hurdle 就大家都噤若寒蝉了，那这就是很大的问题。国卫院不就是这样吗？所以这个要到国卫院去解决这个问题也是很重要的呀。
谢谢严教授。那我想今天早上的 section， 因为时间的关系，我们就在这里 close。谢谢大会的邀请，谢谢。
那请各位那个 speaker 还有 moderator 还有到我们台前，那我们要一个合照，全部要在。
太错了，他不去打球，他说明天才开始，
不是昨天开始，
不是昨天开始。
现在没有 PPT 这个。如果可以的话，演讲就这样。
会叫。
各位来宾还有线上的来宾，大家午安。那我们下午场的精准医疗国际高峰会即将开始。那我们下午的第一场非常有荣幸，那邀请到郑安理教授当我们的座长，那杨木华副校长当我们的讲者。请。
没有声音。好，大家午安，还有我们线上的朋友我是台大医院郑安理医师，非常荣幸今天来担任我们这一场盛会下午的第一场演讲的座长。那我们下午第一场，我们邀请到阳明交通大学的杨木华副校长。那杨教授呢，他也是阳明交通大学的讲座教授，他是在国内非常知名的肿瘤医学的学者，他多年来一直专注在这个头颈癌，还有特别是很多非常高水准的基础研究。那今天我们很荣幸呢，能够邀请他来为我们讲解有关于头颈癌的精准医学。我们鼓掌欢迎杨副校长。
谢谢郑教授的介绍。今天非常荣幸能够获邀参加2022精准医疗国际高峰会。那非常荣幸能够由郑教授担任座长。那今天跟各位报告头颈癌的精准医疗。那头颈癌的精准医疗其实是在各癌别之中发展算是相对比较少一点。那今天就跟各位分享一点点有关头颈癌的精准医疗的一些初步的面貌。那这个是今天的演讲的大纲哦。那先跟各位再回顾一下精准医疗在癌症的一些现况，然后再跟各位报告头颈癌的精准医疗。那另外就是头颈癌里面的唾腺癌，它其实是比较多相关的驱动基因的突变，那它的精准医疗算是比较可以找得到所谓的 driver。那最后跟各位报告 circulating tumor cell， 这个现在发展有稍微遇到一点瓶颈的一个 biomarker 在头颈癌的一些现况。那这个应该大家都非常熟悉。那精准医疗就是从 evidence-based medicine 进展到 precision medicine。那 evidence-based medicine 大概就是 stratified medicine， 就是根据病人他的临床的级别，还有组织或者是病理的一些分歧，然后再根据临床试验的结果，那再给予病人治疗。那 precision medicine 就是把这个 stratified medicine 分得更细哦。那根据病人的不同的分子特性，那还有他的 clinical feature， 那甚至还会包含他的 medical history、environment， 还有他相关的 biomarker， 还有他的 behavior， 那做出对病人最适切的治疗。那现在其实 precision medicine 虽然是说在各个癌别都有广泛的应用，但是有一些癌别相对是非常的成功哦。像大家都知道乳癌或者是肺癌，还有一些血液肿瘤 precision medicine 就是非常的成功。但是有一些的癌别 precision medicine 的发展就相对的比较困难。那可能的原因就是因为这些癌别它比较缺乏所谓的 druggable 或者是 driver gene。那头颈癌就是这一类的癌别的其中的一个代表。那今天就跟各位报告有关头颈癌在这种缺乏 driver gene 的状况之下，那有没有一点点发展 precision medicine 的机会？那这个是肺癌当做一个最好的例子，大家都非常的了解。那在30年前，可能就是组织学的分类，包括鳞状细胞癌、还有腺癌、还有大细胞癌、小细胞癌，就是肺癌的主要的分类。 那现在当然就是用各种基因的 mutation 或者是 fusion， 这个像是 EGFR 的 mutation， 或者是 KRAS ALK 的 alteration， 这个应该大家都非常的熟悉，所以肺癌是一个最好的例子。那另外一个很好的例子就是比肺癌的发展或许还在更早一点的乳癌哦，乳癌在很早就发现它的 hormone receptor 跟 HER2 对于病患的治疗跟预后有非常重大的影响哦。那这个大概是 precision medicine 的最早期的先驱哦，就是我们在看病人治疗的时候，不只是只有看他的 clinical 跟 pathological staging。那这个 classification of breast cancer by gene expression signature 大概是在 Human Genome Project 之后，这些 high throughput sequencing 渐渐越来越方便之后，有这样的研究。那后来发现这些 gene expression 的 signature 跟临床上去分所谓的 triple negative HER2 跟原来传统的分法其实有相当大的 overlap。所以现在临床上还是比较少去用 gene expression signature 去做分类。那但是检测某一个 panel 的基因对于预测乳癌是不是需要接受化学治疗，其实是现在是已经有临床上的实用的例子哦，就是 Oncotype， 它是在病人接受治疗之后，在 hormone receptor positive 的病人，有一些病人或许不一定需要接受化学治疗。那他去检测一定数量的基因的表现，然后再根据他的 scoring， 然后再决定这个病人是不是可以不用去接受原本不太需要的化学治疗。那这个是 gene expression signature 能够应用在临床治疗一个少数的例子哦。因为我们现在大部分的 precision medicine 所依靠的大概还是基因的 mutation 为主。那在头颈癌方面的 precision medicine， 就像刚刚跟各位报告了，其实发展是相对的比较困难，因为头颈癌是比较缺乏所谓的 driver gene 的 mutation。那头颈癌大概可以分成2大类哦。那我们今天先 focus 在 squamous cell carcinoma， 它大概可以分成 HPV positive 跟 HPV negative。那 HPV positive 大概就是它大部分都是在 oropharynx， 那 HPV negative 大概就是从口腔到食道上2/3都是 HPV negative 的广义上面的 head and neck cancer。那它的 histology 有比较也有不同，就是 HPV negative 它可能就是keratinized， 那 HPV positive 可能就是 basaloid。那 HPV positive 大概年纪比较轻，那但是也是男性为主。那 HPV negative 可能是跟抽烟、喝酒、吃槟榔比较有关系。那 HPV positive 这些可能它还有可能合并有其他的 immunosuppressive 的一些状况。那最近这几年 HPV positive 的 head and neck cancer 逐渐受到重视。那大家也可以很容易了解到，就是 HPV positive 跟 HPV negative 它的 etiology 非常的不同。那它的 gene expression signature 显然一定是非常的不同。那这2个是不是在做 precision medicine 的时候，我们是可以分开来看有什么不同的 target？ 那这也是最近这几年头颈癌一个比较值得大家注意的一个课题。那头颈癌有没有像 breast cancer 这样的 molecular signature？ 那也是有研究者去做这样的探讨。那去做这些 gene sequencing， 这应该是在做 microarray 的时代，也还是可以把这些head and neck cancer 分成4大类哦，分成 atypical、classical、mesenchymal 跟 basal 这4大类。那这4大类是根据它的 gene expression 的 profile 来分哦。那它 gene expression profile 我们可以看，这个是 microarray data， 它基因的表现是有明显的不同。那这个分类在临床上是不是有应用的价值哦？那这个到后来就没有进一步的临床上面的探讨，原因是因为我今天就算是分了这4类，那我治疗方法也还是相同，那就不像乳癌去把病人分类，或者是把肺癌病人分类有分类的价值哦。所以这个 molecular classification of head and neck cancer 可能仅止于研究，那在临床上面的价值到目前还是不太清楚。那我们也尝试做类似的分类，其实去把 head and neck cancer 去做分类其实并不困难，就是我们也是可以分成4类。那我们去 跟随这个 cell biology 去看看这个病人的 sample， 他是不是在 epithelial mesenchymal state 有这种 intermediate state， 或者他是比较偏 epithelial state， 或者是偏比较 mesenchymal state， 是可以分的。但是这些跟临床上面治疗好像还是有一段距离。那 head and neck cancer 比较常见的 mutation， 那在各国的报道其实大概都还蛮类似的，大概 P53 的 mutation 大概可以到70%以上。那第二个常见大概就是 PIK3CA 的 mutation。那在接下来就是会有不一样的 mutation， 我们可以看到有像 P10 的 mutation 也是常常会被报道。那但是这一些 mutation 大家可以看得出来，除了 PIK3CA 或许有可能是 druggable， 那其他的 mutation 好像大部分都跟治疗其实没有太大的关系。那我们也在我们自己本土的病人呢，也做类似的分析，那结果也是还蛮类似的，就是我们也是 P53 mutation 大概可以到70%以上。那刚刚的那个 mutation 大家可以看到，就是说 P53 mutation 70%是在HPV negative， 那如果是 HPV positive 的话 ，P53 mutation 的比例就会稍微降一点，大概降到3-4% 左右。那台湾的 head and neck cancer 其实 HPV negative 的比例是比较高，那我们看到 P53 mutation 到74%。那另外是台湾的 head and neck cancer 还有一个是 CDKN2A 的 mutation 的比例是相当高，那跟国外也是 consistent。那另外是 Caspase-8 跟 FAT1 这两个的 mutation rate。那还有 Notch1， 这一些在国外的报道也都是有看过，那它的 mutation rate 也是算是前几名，所以我们分析的结果跟国外是 consistent， 但有一些不一样，大概就是 Caspase-8 在台湾是看起来是稍微比较高一点，那但是这一些看起来好像也都没有真的 druggable。那 CDKN2A 的 mutation 曾经有人探讨在 head and neck cancer 会不会对于这些CDK 的 inhibitor 会比较 sensitive， 那就我做的 pilot study 是没有影响。那所以对于头颈癌来说，我们通常是希望要找到这种所谓的 driver mutation， 可是 head and neck cancer 其实真的由国外的 data 跟我们自己的经验看起来是没有 driver mutation。那如果没有 driver mutation， 到底有没有哪一些或许有比较 low incidence 的去看看呢？那在国外是发现 HRAS 的 mutation 有可能是 head and neck cancer 的 driver mutation。那其实 HRAS mutation 在过去的报道，它比例不高，大概4%到8%。那刚刚的那一篇国外的文献跟我们自己的病患也没有发现，大概有超过5%的 HRAS mutation， 所以算是一个比较小的族群。那 HRAS 的 inhibitor， 那最近是有被开发，叫做 tipifarnib。那 tipifarnib 这个 inhibitor， 它是对于 targeting 这些 mutated head and neck cancer 是有还不错的 overall 的 response rate， 可以到55%。那这些看起来是将来是有潜力哦，只是说这个 tipifarnib 它在 head and neck cancer 因为比例 mutation 的比例是还比较低哦，所以是不是每一个 head and neck cancer 的病人都需要去做 HRAS 的 screening， 这将来还需要再验证。那另外的就是 tumor mutation burden， 那这好像没有换到之前是，不过这个也可以，没关系。那再来就是说，在头颈癌方面，最近的免疫治疗有相当的盛行，那大概有几个重要的点可以跟大家讨论的，就是 tumor mutation burden 跟它的 microsatellite instability， 那这一些可能对于治疗会有相当大的影响。那 tumor mutation burden 过去的研究是发现，在 HPV negative 的病人，HPV negative 的病人，他 tumor mutation burden 跟他的 treatment 的 response 是有相关。那 HPV positive 或者是 EBV positive， 我们知道 EBV positive 比较多是 NPC， 那这一方面的话，就是跟 tumor mutation burden 是比较没有相关。那另外就是过去有发现有一些 mutation 的 signature， 那跟 immunotherapy 的 response 是有关系。那可是这个 mutation signature， 刚刚我们看到这边是有 Notch1， 跟刚刚过去的跟 跟我们的发现跟国外的文献也是 consistent， 所以有某一些 mutation 或许是跟 immunotherapy 的 response 是有关系。那另外是 microsatellite instability， 那这个对于 head and neck cancer 来讲，可能也对于它的 immunotherapy 会有关系。那这个 Keynote 158是一个是一个 basically trial， 它里面是包含了除了 head and neck cancer 之外，有多种的癌症。那在 head and neck cancer 里面，它如果说是有 microsatellite instability 的话，它的 overall response rate 是可以到 34%。那 NCCN guideline 也把这些 MSI high 的 head and neck cancer 列为 pembrolizumab 的一个 alternative choice in head and neck cancer。那所以刚刚讲的大概对于头颈癌来说，能够使用它 precision medicine 的 target 其实并不多。大概第一个就是 HRAS 的 mutation 将来是有机会，第二个就是相关 IO 的一些 marker， 像 tumor mutation burden、MSI high 或者是 MMR deficient 这些跟其他的 cancer 所谓的 agnostic treatment 的道理是相通的。那再来就是说，我们看一下 salivary gland cancer 的 precision medicine。那 salivary gland cancer 它其实有非常复杂的病理形态哦，那包括 adenocystic carcinoma 或者是 adenocarcinoma 或者是 mucoepidermoid carcinoma 等等。那它能够被 target 的 driver 相对于 head and neck cancer 来说，这几年有比较多的发现，大概主要发现都是 HER2 跟 androgen receptor。那这个是 salivary gland cancer 的 molecular target， 包括 HER2 跟 androgen receptor。那在 salivary gland cancer 如果是 androgen receptor positive 的话，那可以用这些 androgen receptor blockade 跟 GnRH 的 inhibitor 来做治疗。那看起来它的 response rate 是看起来还是相当不错。那这是我们自己的病人哦，这个是一个63岁的男性，那他是 salivary gland cancer， 他有 metastasis， 他 HER2 是2价，那我们是并没有给他用 HER2 的治疗。那 androgen receptor positive， 那我们是给他用 Leupride 加上 bicalutamide。那病人后来是曾经有达到 response， 但是后来还是 progression。那第二个 salivary gland cancer， 它 HER2 positive， 它看起来是无论是用 Herceptin 再加 docetaxel， 或者是右半边的这个是 ADC 哦，就是 HER2 的 ADC， 看起来的 response rate 可以到7到9成，看起来是相当的 promising。那这个是我们自己的病人，他也是 HER2 positive 的 salivary duct carcinoma。其实这些会表现 HER2 或者是 androgen receptor， 可能是 ductal carcinoma 是比较多。那看起来是 HER2 amplification， 那我们也是给他用了 Herceptin， 再加上他也是 androgen receptor 也是 positive， 然后用 androgen blockade。那后来他是在 salivary gland cancer 是有 remission， 但是他后来又发生了 second primary colon cancer。那另外就是 salivary gland cancer 有一部分所谓的 secretory 的 salivary gland cancer， 它会有 N-track 的 fusion。那这个是我们的病人，其实在蛮短的时间之内，就去年就一下子收到这个本来都很少见的这种 secretory carcinoma。那一个是有 N-track 跟 ETV6 的 fusion， 那另外一个也是 N-track 3跟 ETV6 的 fusion， 两个的 fusion 是这种最常见的，在 salivary gland cancer 所secretory type 的 salivary gland cancer 的 N-track 的 fusion。那这两个就第二个是有吃到药，第一个是还没有来得及，他后来病人在拿到药当天就走了， progression 的非常快。那 salivary gland cancer 它在国外的报道是用 entrectinib 或者 larotrectinib， 其实 response 都算相当的好。那最后再跟各位再分享一下 circulating tumor cell 在头颈癌的现况。那 circulating tumor cell 大家都很清楚，那现在我们除了在 single 的 circulating tumor cell 这个 cluster 的 circulating tumor cell 现在也受到一些重视。那去分离的方法大家都很熟悉哦，就是有2个主要的平台，一个 cell search 是用磁珠去做分离，然后再做 immunostaining 去做 visualization。那第二个是微流道现在可能更可以说是一个主流，那是以 CellMax 平台为主。那它的好处就是说，它可以去看这些 cluster 的 CTC， 会比 Cell Search 用磁珠 sorting 要更便利。 那这个是有国外的报道，就是说，其实做 CTC 除了的 numeration 之外，它有一个很重要的功能，就是它能够去做 primary culture， 然后去做 drug testing。那这些 cluster CTC 的重要性，我最近也逐渐被了解。那 cluster CTC 是必须要用特别的 biomaterial 来做分离。那我们过去也和中医院的张一之教授合作，那这2年持续的有在使用 CellMax 的 microfluidic chip 去做 cluster CTC 的分离，那也去培养 tumoroid。那我们是可以分离，很明显的是可以看到这边是真的是有 cluster CTC 在头颈癌的病人的血液之中。那这些 cluster CTC 跟 single CTC 它的临床的意义看起来其实 single CTC 也是有临床的重要性，但是 cluster CTC 好像跟这些 lymph node 的 metastasis 更为相关。那我们都知道 head and neck cancer lymph node 的 metastasis 是比 distant 更多，所以这 cluster CTC 是有它临床的重要性。那我们可以看到这里我们有分享1-2个病人的病程。那第一个病人就是他在治疗之后，他 cluster CTC 很快的下降，但是后来他 cluster CTC 上升之后，很快的造成 distant meta， 发生是要比 distant meta 还要再早一些。那后来的3个病人，他治疗之后 cluster CTC 都下降，那病人后来就没有再复发。所以 cluster CTC 对于头颈癌来说，因为头颈癌是一个 lymph node metastasis 为主的 cancer， 或许是值得去观察。那今天就简单跟各位分享这几个在比较相对困难没有 driver gene 的一个 cancer 里面的一些 precision medicine 的一些现况。谢谢。
好，非常感谢我们今天听到杨副校长非常精辟的一个头颈癌精准医疗不知道大家有没有什么问题要请教他？我问一个非常简短的问题，因为在台湾我们的食道癌很多都是 squamous 嘛，那就一个 molecular subtyping 这个观念来看，我们的 squamous cell carcinoma esophagus 跟头颈癌的 squamous cell carcinoma HPV non-related 它的关系到什么程度？相似性？
谢谢郑教授。如果 molecular subtyping 其实突变就是 molecular signature， 其实上2/3的 ESCC 跟头颈癌 non-HPV 几乎是完全一样，所以这些应该它在国外有一些欧洲的国家，他会觉得这个是同一个 cancer。那有的少数的 clinical trial 也会在一起做。那突变的 signature 像 TP53 这些也是几乎都很像，所以我也是跟郑教授有类似的，我不晓得郑教授是不是这样想，就是这2个其实在治疗的时候，有很多治疗的方式是可以共用，或许做 clinical trial 的时候也可以考虑是不是可以互相去比较，甚至联合。谢谢。
因为我们在医院好像头颈癌的跟做这个上消化道就分开嘛，
是
其实这2个病其实是同样的，
是
所以将来我们医师的分类也许要稍微修正一下。好，不知道我们大家有没有其他问题？好，没有的话，我们再次谢谢这个杨副校长给我们这么精彩的演讲。
谢谢。
接下去我请那个杨宗仪杨主任来主持。
我们下一个 session 就是关于 lung cancer 的 precision medicine， 那还有何昭基教授，那欢迎当 speaker。
好，各位现场还有线上的专家同好，大家好。那接下来很荣幸我们要到何兆基何医师来跟我们做演讲。那从早上跟刚才大家都可以了解现在目前大概 precision medicine 发展的比较好的一块，应该是在 lung cancer 这一块。那所以下面这个 talk 呢，我们很荣幸邀请到何医师来跟我们分享这一方面的一些新的 update。那何医师是台大医学系毕业以后呢，就在台大的胸腔内科完整的全龄，然后之后拿到这个病理研究所的博士学位。那目前是台大医院的教授，也是这个他们内科部的副部主任。 那这个何医师在目前是在台湾在肺癌的不管是临床照护，还有在基础研究上面都算是这是算最顶尖的一个专家了哈。那我们今天就非常荣幸请到何医师来跟我们做这个 Lung Cancer Precision Medicine 这方面的演讲。那何教授请。
谢谢杨主任的介绍哈。那我们接下来谈的是 lung cancer 的这个 precision medicine。这是我 disclosure。那大家看到一个这种表，大家看有时候都觉得很烦哈。不过还是先解释一下，这是台湾的 consensus。那这里头下面有看到5个学会哈。这是我们最近这1-2年，就是5个学会就是专家在讨论，我们把 lung cancer 做一个 consensus， 在这个 late stage 哈。我们知道 lung cancer 在分 small 跟 non-small， non small 的话大概主要是分 squamous， 那跟 non-squamous 是 adenocarcinoma 为主。那大家都知道 lung cancer 在作为oncogenic driver 或 driver mutation 走得最快，所以大家看到在这个 non-squamous adeno 这地方我们有很多的 oncogenic driver。那这些蓝色的就是我们现在基本上应该要验的，因为这都有很好的 target therapy， 很好的 target therapy， 所以基本上应该都要去验这些 oncogenic driver。那在讲 precision medicine， 在这个没有 oncogenic driver 的这个所谓的 no oncogenic driver 这个族群，难道我们就不做 precision medicine 吗？答案也是错的哈。其实我们还是可以去做一些像这个 PD-L1 的染色。那这些 PD-L1 这种染色的话，可以去guide我们去做不同的 IO 的一些治疗。那我们5个学会在最近会有这个会最近会有 NANS， 如果大家有机会的话，可以在学会的网站看到我们这个比较完整的一个版本。那 oncogenic driver 在 lung cancer 里头，在东亚跟欧洲其实不太一样，我想是大家都知道哈，亚洲人 KRAS 的比例比较高，接下来才是 EGFR。那东亚的话，那在 EGFR 这个 mutation 的比例是1半以上，其次是 KRAS。至于这些比较少见的这些driver mutation oncogenic driver 的话，实际上东亚跟西方没有太大的差别哈，差别最大的是在 EGFR 跟 KRAS。那对于一个胸腔科医师，或者说对于做一个肺癌的专家哈，我们的心里的痛永远是在这里。我们要得到这个肺的这个组织呢，我们大概有这几种方式，从一个比较不 invasive 的抽胸水 fine needle aspiration、needle biopsy、bronchoscopy、open biopsy 叫 surgical resection。可是呢，你可以看得出来，这个是拿到的是 cytology， 这个拿到的是 small biopsy。只有这个地方我们才可以拿到那个比较大的这个肿瘤。那我相信刚才杨副校长的 head neck 还有 breast cancer， 你要拿组织都相对这个肺都是比较好做的。那怎么说呢？你可以看到，我们看到了一个 small biopsy， 或者甚至一个 cytology 的 cell block， 我们光是要去做一个histology 的一个分类，我们做了 IHC， 在这组织大概就损耗过半哈，就已经损耗过半了。那我们现在在讲这个 precision medicine， 我们要做这些 oncogenic driver， 那我们的组织呢，大概就不够。所以在做肺癌的这一群人里头，其实尤其是我们胸腔科的医师，我们在做肺癌的时候其实我们现在最麻烦的就是要拿到一个足够的组织。好，在一开始的时候就想办法能够做足够的组织，才能够往下做所谓的 molecular testing。那这是我们现在应该是最头大的地方。那现在我们的这些 driver、oncogenic driver 在台湾的健保，我们都是 single gene testing。在肺癌的话，我们都是先做 EGFR， 然后 ARC、ROS1、BRAF， 然后EGFR 是用KIT 或是 single gene sequencing， 那其他的话可以用 IGC。可是大家可以看到这个 study 其实相当的有名哈，大家可以看。就是说，如果你是一个基因一个基因去做的话，你的一个基因一个基因去做的话，这个上面这个数值是代表它所需要的 slice， 就是那个 pathology 切下来那个。其实我们大概到了第4个基因的时候，你就大概很多人都没办法做了，这组织的损耗非常大。然后基因的检测那个基因的这个准确度也会根据第1个基因、第2个基因、第3个基因，所以。实际上，在肺癌，我们刚才的 consensus 有讲到，我们要验至少8个 oncogenic driver。 那你如果要验的话，你就不能用 single gene testing 这样方式一个一个去做。为什么？因为组织会不够。当然这还考虑到这个所谓的钱的问题，然后这是我们讲的经济学的问题。那当然我们今天不谈这个。对我们这个临床的医师来讲，我们希望的就是 NGS 一口气就是把它全部验。那我们也知道说，在 tissue 的这 NGS 常常因为组织不够，所以我们会用什么？我们用 liquid biopsy。那在目前来讲 ，liquid biopsy 这 NGS 的确是会 improve 我们这个 target gene 的这个 identification。这我想这大家都很清楚。那在这两个最大的 trial， 这是目前在文献上可以看到这两个最大的 trial。那这两个最大的 trial 告诉我们说，这些病人大概300个病人左右引入进来，每个病人都去做 tissue 的基因 test 跟 liquid 的 NGS。第一个 trial 它的基因是一个一个做的，所以它有将近192个病人没有完成 tissue 的基因 testing。那你可以看得出来，在 liquid biopsy 在这个地方，你可以 rescue 非常多的病人，就是在 tissue 没有验到的时候，你用 liquid biopsy 可以验到。那在这个 UK 这个 study 呢，它这个 liquid biopsy 它验到的这个就是 tissue 没验到，它 liquid 验到的这个病人数也比较多。可是你要看到下面这句话，它有8%，将近40%的病人是没有办法验 tissue 的 NGS。那如果反过来讲，如果这个病人可以做 tissue 的 NGS， 也可以做 liquid 的 NGS， 那 tissue 的 NGS 它的 sensitivity 跟 specificity 都比 liquid 来得好。也就是说，在一开始诊断的时候，我们在强烈的建议应该要验 tissue 的 NGS， 而不是验 liquid 的 NGS。一开始诊断的时候，这是非常重要的。那因为 liquid 大家不要忘了，它只是 tumor shading 出来的。它有些不 shading 的话，它没办法验。那只是说，在肺癌里头，我们最大的问题就是组织不够，这是一直都是我们目前在 daily practice 的时候一个问题。所以 liquid biopsy 可以做一个 complementary， 但是我们还是希望足够的组织做组织的 NGS。那接下来我要秀几个 case 只是 demo 说，我们现在的 NGS 的确比我们这个 single gene 的 testing要来得好哈。那像我们知道 NGS， 我们现在台湾大概健保给付就是像 COBUS 或者是 Sanger sequencing。那这个COBUS 跟 Sanger sequencing 呢，它的 sensitivity 当然远远比不上这个 NGS。那 NGS 是更 sensitive。像这个是一个我的病人哈，34岁的女性，她一开始的诊断就是肺有一颗脑有一颗转移，所以她是 M1B Stage 4。那这个脑部的转移呢，在外面的医院已经被开刀拿下来。当时这个转移的这个COBUS， 它的 EGFR mutation 是 negative。那他来到台大医院，那我们就让他进了 clinical trial。那这个女性34岁女性，大家想说，怎么去打免疫？这也有点误打误撞了哈。她打了免疫之后，居然消了。那这个 trial 当然打2年嘛 ，Pembrol 第一线的 Pembrol。2年之后，就是停下来，再过1年 ，brain 就是新的 brain 的 metastasis 跑出来，那 primary site 是 under control。这个时候我们在想说，这么年轻的女性，她应该有 driver 哈，我们的想法是这样。那于是我们就建议她说去做那个她原来的医院去把那个组织调过来，我们做了一个 NGS， 这个 NGS 出来是一个 uncommon mutation 719。那这个 uncommon mutation 大家知道，这个在 COBUS 这个 kit 它应该是要验得到的，它应该要验得到的。可是在这个病人身上，他原来的 COBUS 却是 negative， 然后是我们用 NGS 的时候才发现到这个 mutation。那这个 case 呢，大家应该想说，因为有 brain match， 大概应该要考虑用 afatinib 或 osimertinib。不过因为这个病人他工作的关系，他有一些考量，所以他后来是 rechallenge Pembrol。那 rechallenge Pembrol 到现在2年半了 ，brain 没有再复发，所以好像 Pembrol 对他来讲好像也是有效。那大家可以看一下这 tumor mutation burden 9，相对 EGFR 来讲是算高的，所以这个病人目前还在打那个 pembrolizumab， 所以这 NGS 其实比 COBUS 要来的更 sensitive。另外就是 NGS， 我们知道 COBUS 它有固定的点位，它不是所有的 mutation 的点位它都会去验到。 那这个84岁的男性，他是一个 heavy smoker， 100个 pack year。那看到这个，大家会想说啊，这应该是没有 driver 哈，在我们以前传统的想法。那他biopsy的时候是 adenocarcinoma， 也是一个 single 的一个 brain 的 metastasis。因为他的这个 PD-L1 是0%，哦，那其他的我们常见的这些 driver 是没有的。那想想看，84岁，然后没有就是家人跟病人拒绝使用化疗的状况之下，我们就想说，不然就试看看做一个 NGS， 就 NGS 出来是一个 rare 的一个 mutation 哈，这个是很少见的一个 mutation。那这个 mutation 呢，对于 EGFR TKI 是有效的。那这个病人一开始因为年纪大，我们先用 gefitinib， 但因为 hepatotoxicity， 所以要改用 afatinib。那从2019年到现在，他仍然还在用 afatinib， 所以这是一个 long term 的一个response 的一个病人，所以对这个 rare mutation， 它 EGFR rare mutation 也是很有帮助的。那另外，这个 NGS 哈，这个是一个我很喜欢引用这个 study 的理由，是因为这个告诉我们一些事情蛮有趣的。这是日本做的 data， 他做了 EGFR 的single gene 的 testing， 如果是 negative 的话，1900多个人，他去做 NGS， 在这个 single gene EGFR mutation negative 的这群人，他做了 NGS 的时候，他发现这个 major EGFR mutation 还有 2.7%， 那在 rare 的 EGFR 的 mutation 有 5.5%。也就是在 EGFR 这个基因上面 2.7 加 5.5 有 8.2% 的病人在 EGFR 还是可以找到这些 mutation， 而且对 EGFR TKI 会是有效的，那就1/12哦，也就是在 EGFR single gene testing 是 negative 的情况之下，12个病人还可以找到一个病人，他 EGFR 是有药物的。这个其实这个比率其实相当的高，也就是说我们现在传统的 single gene testing其实有很大的问题。那另外这个 case 这个 case 是刚好杨校长刚好离开，这个 mucoepidermoid carcinoma 这个是一个蛮特别的一个 case， 这个不是我的 case 啊，但是因为它很特别，所以我想拿起来给大家看一下。80岁的男性 smoker， 他有 brain， 这是他的 primary tumor， 有 brain 有 liver 的 mets， 所以他是 stage 4B。那这个 brain 因为它有症状蛮大的，他把它开下来之后，他的这个 pathology 是 mucoepidermoid carcinoma。那这因为病理科李部长在现场，他应该知道这个是一个 salivary gland 的一个应该比较好发在 salivary gland 的一个 cancer。那当然病理科就看到这个 mutation， 他就直接给他做了一个这个 fish。那因为胸腔科我们对 MAML2 我们不了解，如果等一下有愿意 comment 的话，可以 comment 一下。它这个 fish 是 positive， 我要先讲这个 fish 是 positive， 所以它有没有可能是 salivary gland 的这个 metastasis？ 那因为它主要的 primary tumor 我们认为是在 lung， 所以我们给它做了一些就是常见的这 lung 的这些 driver 都是 negative。那 PD-L1 是 2%。那当然，他开完刀以后，这个 frontal tumor 就去做这个 RT 哈。那这个 case 当然我们还是给他做了一个 tumor 的 NGS。那你可以看到他的这个真正的这个报告，它是 exon 14 MET exon 14的 skipping， 这个是在 lung cancer 的一个 driver， 好，这是 lung cancer 的一个 driver， 好，这个是比较特别的。那当然它有一些 amplification 跟一些 homozygous 这个 deletion。那大家问说，这 NGS 拿的是哪一个组织？他拿的是这个 brain 的这个开下来的，跟这个 fish 是同一块组织。所以我们当时在讨论的时候，我们认为这个 fish 应该是假的。我们认为是假的，因为 NGS 完全没有看到这个，因为 NGS 应该要包括这个 rearrangement 嘛，我认为应该要包括。所以我们当时我们在那个 discussion 的时候，我们认为这个 fish 应该是假的。这就很有趣了哈，这是一个非常有趣的。那大家就会想说，那 MET exon 14 skipping， 那我们就给药嘛。那这个是一个它的这个NGS 的 data， MET exon 14的 skipping， 它这个 oncogenic driver 就是我们刚才讲那8个 driver 其中一个。那另外这个 amplification 跟这个 deletion 哈 homogeneous 的 deletion， 那我们给它的这个 quizo。 那就接下来就申请这个 tepotinib， 这都是 MET 的这个 inhibitor， 这是那 tepotinib 是用 EAP 的 program， 可以看到它有效，但是 liver 呢是变大，它是一个 heterogeneous 这个 response。那我们目前认为它应该是 lung cancer， 只是一个比较奇怪的一个表现。那这是 MET exon 14 skipping 的 alteration， 这个 MET exon 14的 skipping 是目前在 genetic testing 应该是最难被检测的因为它的genome的这个 background 是最复杂的。那我们目前的想法应该是要用 RNA， 用 RNA 的 NGS 可能是最好的一个方式，可以检测到这个 MET exon 14的 skipping。那这个是 MET exon 14 skipping 的好发的位置最常见是这个所谓的 splice donor site 的 single 基因的一个 mutation。那我们刚才那个病人就是这个最常见的这个 mutation 就是最常见的一个 mutation。那其实杨副校长刚才有提到，我们有些时候会用 gene expression 的 pattern 去看看这个 cancer 的这个表现。也就是说，我们用基因的 expression 这个 pattern， 有些时候可以回过头来去看看这个到底是什么样的一个 cancer。因为我们现在目前大概都是在做这个 driver gene 的这个 detection 嘛。那万一如果这个肿瘤它的 driver gene 跑出来一个很特殊的话，那有没有可能是我们一开始的诊断是有问题的？我下面举一个例子，是我最近的一个 case。这是一个47岁的一个女性non-smoker， 她有 breast cancer， 然后开过刀。她在追踪的过程中发现了这个 multiple 的这个 lung tumor， 然后还有一个 median 的有个 mass， 可以看到这个 PET 这都有亮起来。那她其实现在有院其实就是另外一家医院大医院，她做了这个 CT 钙的 biopsy， 她报告是解 squamous cell carcinoma。那做了一些那个 IHC 的 staining， 他的 final 报告，他认为是一个 extrapulmonary 的一个 origin， 认为有可能是 uterine 来的。那这个病人他非常的就 nervous， 所以跑来我们医院，我们就再给他做了一次的 biopsy。那这一次呢，我们用的是这个 EBUS endobronchial ultrasound 的 transbronchial biopsy。那出来一样是 squamous cell carcinoma， 掰的位置不一样这两边掰的位置不一样，一样是 squamous。那我们的病理科医师非常就是认真的做了一大堆的一些那个所谓的 IHC 一个 marker 的 staining， 这个marker 的 staining。那他最后给我们的一个诊断叫做 basaloid squamous cell carcinoma， 他认为是那个lung的 squamous cell carcinoma， 这是他的这个pathology 一个 finding。那我们就想说好吧，因为都年轻嘛。我们就给他做了一个 NGS， 那这 NGS 出来以后，这是非常意外了。我想这个李部长应该看到这个也应该非常意外，它是一个 KIP V560的 deletion， 在座应该大部分是 oncology 应该对我来对大家而言，这个 mutation 应该是大家更熟悉的，因为 lung cancer 没有这个东西 ，lung cancer 没有这个东西。这 KIT V5601的 deletion， 它主要是在 GIT， 我想大家都清楚，这应该主要是在 GIT 80%到90% GIT。那发生什么事情？这个病人我们本来以为是 lung cancer， 就我们做了 biopsy squamous， 怀疑是 lung 的 squam。那当然你会跟我解释说，那 median 的内科那个我们有掰 ，median 的内科大科我们有掰，它出来的时候是一个 benign 的 lesion。那当然，因为他只掰了那个2次，我们一般会掰6次，但他那天因为 hemopsoris， 所以我们只掰了2次。那因为 hemopsoris hemodynamic unstable， 所以我们没有办法再往下做。那我们想说那是benign的，我们就没有再往下做。结果没想到这个 NGS 出来的是 KIT 这个 mutation。那我们再请病理科再回去染色，结果它出来的是一个 thymic carcinoma。所以有些时候我们对这个 pathology 这个诊断，有些时候我们认为它是什么，就 NGS 出来的 data， 有些时候会给我们一个可能一个不太一样的一个 information， 这是一个比较特别的一个 case。那我们就给他用了 imatinib， 那当然这只用了4天啦，这肿瘤感觉上好像有点消。那因为这个病人他对这些药物反应太大，所以他只吃了4天以后就再也没吃了哈。那我现在尝试给他换另外一种那个 c-KIT 的药，看有没有机会能够有一个 response。那最后呢，我用这张就是这一样，就是有5个学会台湾胸腔暨重症加护医学会 中华民国癌症学会、台湾肺癌学会、台湾临床肿瘤医学会跟台湾免疫暨肿瘤学会，我们5个学会这个共同讨论，专家共同讨论，我们有一个所谓的 treatment consensus。那这个部分是 for 这个 oncogenic driver， 所谓的 oncogenic driver。然后我们有这个就是建议应该 detection， 然后我们有建议的药物，这是一个我们的那个 lung cancer 的 consensus。然后这个大概在应该在几个礼拜或是1-2个月之后，可以在这5个学会的网站可以看到这个出来。好，这我简单介绍到这边，谢谢。
好。谢谢何教授非常精彩的演讲，还有一些临床 case 的sharing哦。那我们还有一点时间，那不晓得，现场还有看线上有没有问题要请教何医师的？我先请教何医师一个问题哦，就是一般我们的 PCR base 做出来的啦，或者是单点基因做出来，大概不会有那个问题说，我做出来有问题，不晓得它代表什么意思哦。但是 NGS 就常常做出来。可能这个它代表的意思，比如说它就算是最常见的 EGFR mutation 的，你也常验到，也不是常常的，偶尔会遇到一个你没看过的 EGFR 的 mutation， 那也文献查不到说这样的 mutation 会不会 respond to 这个 TKI EGFR 的 TKI。那不晓得何教授如果遇到这样子类似的情况的话，会怎么处理啊？那这个问题其实也带到早上我们有讨论到的，就是所谓的 molecular tumor board 的这件事情哦。那不晓得在台大医院或者说这个在 molecular board 在这一方面扮演的角色，这个何教授有没有什么这样的特别的建议哦？
杨主任的题目这个问题蛮难回答的。李部长在现场，等一下应该可以顺便，我先尝试回答，因为李部长他是这个 NGS 的专家，我想等一下请他回答好了。我先解释一下，因为我们这个 EGFR TKI 基本上应该是在 kinase domain 啊，所以就是 exon 18到21，就是那附近了。如果你是在这个地方以外的一个 mutation 的话，我个人认为那个可能意义可能不太大。那以内的话，如果当然上网去查哈，现在很多的那个 database 都可以让你去看，那很多人都会把这个 data 会丢进去，那你可以去看它的那个 response 或什么。如果真的没有的话，我的建议也只能试看看了，这是我的建议啦哈。那第二件事情是刚才讲那个 molecular tumor board， 那目前其实这应该是国家的政策的一个就可能是为那个国民健康署他们希望，卫健保署他们希望这样推行，所以在台大其实我们已经都有一个固定的一个 molecular tumor board， 然后固定会来讨论这些比较奇怪的 NGS 的这些 data。然后我们会做一个讨论。那我刚才举的那个 Fish Party 那个exon 14 skipping， 其实就是在 molecular tumor board 里头被拿出来讨论的一个 case。它蛮有趣的是，它 Fish 是 Party， 那 Fish Party 为什么 NGS 做出来反而是 exon 14 skipping？ 那 Fish 那个基因，照理说它 NGS 应该在嘛，哈。他完全没看到，他就只有看到他的那 NGS 的 exon 14 skipping 跟其他的这个 amplification 跟那个 homozygous deletion， 对，这个蛮有趣的。那我们在现在台大目前就是每个月固定会举行一次这个lung cancer 的那个 molecular tumor board MTB， 那我们会讨论一些比较奇怪的这些 case， 那这样的话大家也会比较有经验。这是目前我们的现状。我不晓得那个李部长要不要帮我们回答一下刚才这个问题，因为这个我可能没有办法回答得很好。
不会，那个教授太客气了。就是我们一个基因它，如果是一个就算是一个 pathogenic mutation， 比方说刚刚教授讲的 EGFR， 我们其实常常看的就是在 kinase domain。那如果是在 kinase domain 之外，那仍然是 pathogenic 的，那它可能真的是对这个疾病的产生有意义。但是我们现在的 TKI， 针对 EGFR TKI 其实都没有 kinase domain 之外的 validation 嘛。所以以 clinical validation 来说，虽然这个基因是 pathogenic， 但用药没有人敢说它会有效。那没有选择的时候，还是可以试试看，因为只有这条路哦。那刚刚教授提了另外一个，我也很有兴趣的是那个 mucoepidermoid carcinoma。那它就是在一开始的时候 ，Fish 有看到 AMML2 的 fusion 是 split apart assay， 应该是拆开。那我后来看一下，它后来的 NGS 是用行动基因的 ATA-uncle。那其实也不是说行动基因的 ATA-uncle 而已，因为大部分用来做 routine 诊断的 NGS， 大概都不会把 MAML2 的这个 fusion 放进去，因为这 跟治疗其实也不太相关哦。所以我在想说，所以如果您也同意的话，我看或许跟那个台大病理科商量一下，因为我那里有比较大的 panel 是专门 for gene fusion 的，有 cover 这个，那就是厘清看说它的 split apart 是不是它粘到那个因虫其他的不表现的地方，还是它真的有一个这样的 fusion 产生，这样我觉得会是很有趣。谢谢您。
谢谢，这个非常好的一个建议。不过那个病人他的用药是有效的，对，所以我们现在还是把它当 lung cancer 在 treat 着。那因为那个不是我的 case， 我可以再跟那个我们的病理科医师再讨论一下。谢谢。
好，那陈教授。
对不起，刚刚提到的 molecular tumor board， 有时候我们会碰到一些 rare 的 mutation， 即使是在同一个 oncogen 上面有不同的 mutation。那我想这个其实应该是不是有机会可以大家把这些 data correct， 就像刚刚李部长讲的，跟何教授讲的一样，很多东西以前没有治疗过的，大家不知道它有没有效，但是很多人会 try 去治疗看看这样。那所以那些 data 只有把全国的资料通通把它收集在一起，我们以后才能够提供一些 evidence， 至少是 real world 的 evidence， 跟他讲说有人这样治疗过，那这个会不会是有机会是有效的？那大概，所以我还是建议是说，每个医院不管是在怎么样状况，是 through 学会或是 through TCAR， 能够把这些不同的治疗的状况能够把它收集。如果我们通通给了厂商，事实上厂商的 database 会越来越大，但是我们还是没有自己的东西，所以这个是一个。那第二个，你刚刚那个 KIT mutation 那个病人，对我来讲 imatinib 是所有的 TK 里面最安全的一个。那如果它还是会有副作用，不晓得你那时候尝试减剂量吗？
有减过，我后来给他一天一颗，
一天一颗还是有副作用，
还是有副作用。
哦，那就没有办法了。谢谢。
好，谢谢陈教授的 comment 哈。那我觉得陈教授刚才讲的那一点，其实我非常同意哈，就是说台湾应该就是把自己的 data 收起来哈。那其实在场有那个郑教授啦，那邱院长，这个是应该我觉得那因为何教授也是协会的一些重要，我们的几个协会的一个重要的一个 member 哈，可以其实往这方面去规划。那其实像刚才讲的 molecular tumor board， 其实，我也认为应该不是各个医院去做了，因为其实有时候我觉得，比如说这个医院假使我有问题，那在我们医院开的 molecular tumor board 通常大概也没有办法解决。那其实应该是看看有没有机会弄一个比较全国性的，或者是一个怎么样的比较，因为其实这上面牵涉到很多的专家、专长的人。其实我相信能够一个医院大到集合这些专家，然后来成立一个比较 functional 的 molecular tumor board 不是那么容易的事情啊，所以可能台大像这样的大医院可以。那其实大部分医院可能不大能做这样的事情，应该是可以看看这样的规划可能会对病人会有比较好处。
我是觉得可以用北中南3个区块，然后利用学会是用那个TOS。好像有一个那个 cancer 的 lung cancer consultant 那个地方，我是觉得可以就用这样的方式去做这个 molecular tumor board， 大家把一些非常就是无法解决的这种 case， 我们可以另外再拿出来再讨论。我觉得这是一个可以做的这个方向。
好，那线上没有特别的 comment 跟问题，那我们时间有点提早，那我们还是move到下一个 session。那我们再一次掌声谢谢这个何教授。那我们把下面时间交给戴主任。
我们下一个session 是邀请到奇美医院研究部李建峰主任来当我们的 speaker。那对于我们这些 molecular study 在 cancer 上的一些运用，那还欢迎就三军总医院血肿科戴明生主任当我们的modulator。
谢谢在座的师长今天很荣幸受邀来参加这个精准医疗的高峰会。那更荣幸的能够介绍下一位重量级的 speaker。那与其说介绍，那如果大家都认识，那介绍好像没有太多的意义。那不过我还是要这个能够让大家 highlight 一下。那李建峰李主任李教授呢，他在 高雄医学院毕业之后，在中山医大拿到 biochemical 的 PhD 之后，那就一直在奇美的这个病理部啊，从主治医师主任，然后到现在也是在奇美医学中心的临床精准医学中心精准医学核心实验室主任，以及奇美医学中心的医研部的部主任。那他的临床研究的 paper， 那跟北中南各地方 outstanding 的学者都有很好的这个研究合作。那所以在我们那个PubMed 上面有近300篇的这个 paper 的 publish。那他个人也获奖无数。那最近也在2020年台湾的癌症医学会有一个 outstanding 的 research award。那以及有很多的这个paper 的 review， 以及在这个临床的这个研究。那今天很高兴呢，能够请李建峰主任呢来帮我们接下来这个研究的这个讲题哈。那我们热烈欢迎李主任。
谢谢戴主任，还有感谢在座各位师长跟教授，大家午安。那时候要交讲题的时候，我一时没想太多，后来收到那个 agenda， 我才发现那个讲题真的是不太好定，因为从早上到现在，大概重要的讲题都被就是说我的师长们其实都应该会被诠释得很清楚。那所以我在这里其实跟各位分享一个，我觉得精准医疗未来可以走出一个不同方向的地方我常常跟我们的同仁讲说，有一天可能不会再多久，这些定序的东西会被走到一个很极致的境界。那这个是也不是说一个耸动的名词我们去想。在2022年，也就是今年5月20号的时候 ，Sloan Kettering 他就 announce 说，所有的未来到 Sloan Kettering 就诊的 pediatric 的 patient， 他们的 tumor， 那 Sloan Kettering 都会帮他们做 whole genome sequencing 加上 transcriptome。那在去年或者前年，我还都认为 whole genome 的 sequencing 大概没有办法很快的走到临床，但 Sloan Kettering 他们这么做的原因，是因为他们发现 pediatric tumor。他们的 gene 的 structure 的 change 其实比想象中的还要多，所以已经没有办法用或者是像现在我们 NGS 一般 fusion 大概做10几个，那大一点的像 Foundation 做200多个 gene 的 fusion， 那再大一点的呢 Illumina 像我们实验室现在用507个 gene 的 fusion 准备要开始使用，但是其实都不见得足以负担那样子的肿瘤的 structure rearrangement 的强度所以他们是打算在 pediatric tumor 就是一律就是 whole genome sequencing 加上expression profiling。 那当然这个是奠基在 Sloan Kettering 它的那个医疗的费用很高的这个状况下，但是也告诉我们这个基因的检测，单就基因这件事情，它终究会走到一个最 detail 就是 whole genome sequencing 都停在哪里，而且这个潮流应该会很快来到。那在那个情况下，精准医疗应该要做的其他事情，就是刚刚在这个何教授的 session 大家有提到的，可能就是 molecular tumor board 跟更多的经验的 sharing。那再来就是不同的 omics 之间的这个连结那在这里我就用 urosilic cancer 做一个 model。那其实 transomic 的 approach 它终究会是一个方向。但这个方向没有办法前进，对不起，是我那个方没有电那有没有人可以帮我下一张？我们现在在做个体化的这种细胞治疗的时候，我们通常会希望用 high throughput 的方式把一个癌症它的潜在的 epitope 给出现，对不起，把潜在的 epitope 把它找出来，好，对，那。这个潜在 epitope 从基因里面去看，跟从其他的 omics 里面去看，其实是不一样的风貌。我们想想看，我们从基因里面去看一个潜在的 epitope， 它要用的方式是我们把一个 sequencing 定出来，把 sequencing 里面可能不同的序列把它找出来，还要去预测 这个不同的序列，它在细胞膜表面有没有办法被呈现出来，所以一切都是预测的。那它后来会需要一些 validation， 但是到了 validation 的时候，可能就已经有一个 gap 了，如果是猜错的话。但我们如果从另外一个 omics 把它回来，就是从 proteomics 的话，它可能牵涉的就是我们把 tissue 里面的蛋白质抽出来，那再来就是我们实验室常常做的 IP， 只是这个 IP 是用 MHC 的 complex 的 antibody 去做 IP， 就是说如果在这个时候，那我们就会把这个 MHC 整个抓下来，那在下一步就是做 peptide sequencing， 所以任何在 MHC complex 上面的这个蛋白质，这些 epitope 其实都有机会被找出来，被鉴定出来。那与其我们从基因体里面去预测，再预测，再预测，不如我们直接从这个蛋白质体里面把 MHC complex 抓出来，再去做这样的分析哦。所以这是两种不同维度的碰撞，就是从 genome 跟proteomics 两种维度的碰撞去交结出最有可能的 neoantigen， 这是一个方式。所以这是一种概念哦，就不同的体学它之间可能会有互相援助的时候。那我在接下来15分钟，我要跟各位提的就是，那在 urological cancer， 我们讲这个 cancer 的 etiology。那这种 transdomic approach 可以给我们什么样的 information？ 那其实我们用自己的一个比较小倍去的有些 cancer 是尝试着回答的问题哦。那我们今天在讲的精准医疗，其实有乳癌、有肺癌、有头颈癌，但其实泌尿癌它的预后一直是不好的。如果在 TKI 之前的乳癌，它的 survival curve 是这样，这是很早期的乳癌了，那个时候甚至连 HER2 都没有的时候，所以我特地找这一群当做历史的 control 在我们医院。那其实有些 cancer 像urinary bladder cancer， 其实它预后是远比乳癌还要来得更不好。那 upper tract urothelial cancer 其实更是哦，这它预后大概就是这么不好。所以，但这个癌症它其实并没有受到太多的关注，因为在过去它并没有太多的 target therapy。再来就是它不是西方人主要的疾病，至少排在乳癌跟 prostate cancer 跟 lung cancer 之后很多的地方。那在台湾，它很早的时候的关注是来自于 epidemiology， 就是 epidemiology。像那时候陈建仁副总统可能就跟很多的科学家做田野调查，去定义出台湾的这个五角病地区，或者做了相关的研究。那这些人我们现在都还是可以看到哦。我们如果有时候去盐田的时候，那可以看到他们的皮肤就长这样，这个是很典型的，就是你看诊的时候会知道这个就是砷中毒的病人。但现在这种病人可能会少一点点，越来越少，因为慢慢的就不见了。那在最近urothelial cancer 它收获了比较多的关注，是因为它开始有了标靶治疗。那这个标靶治疗是针对 FGFR3 的 target therapy。那这个是我们自己的用我们自己 limited case 做出来的，在台湾的 urothelial cancer 的 profiling。那这样的 profiling 告诉我们，其实在 urothelial cancer 里面，在台湾 FGFR3 的 mutation 大概在10% 多一点点，但是真的是 missense 治疗有意义的只有深绿色的地方，那也就是只有10%。相较于西方国家的族群，我们大概少了5% 左右的这个 driving 的 FGFR3 的 fusion 或者是 mutation， 所以暗示应该是有其他的不同的 etiology。那当然，砷是一个，但是我们做了很多田野调查，但是对于五角病区的这些病人，他的 mutation profile 其实现在也还不知道。那更遑论有其他的，在台湾也是重要的这个疾病的 etiology。那在这样的肿瘤里面呢，这是我们自己 in-house 的 case， 整个看起来在不同的癌症里面 mutation 的 tumor mutation burden 的分布，那最多的是 endometrial cancer 跟 colorectal cancer。那这个有点 bias， 是因为在 colorectal cancer 跟 endometrial cancer， 那为了 assay 的开发的关系，我都 include 很多 MSI high 的 case， 所以可以看到它的 tumor mutation burden 其实是相较高很多。那这些都是 MSI high 的 case。那除了 MSI high 的 case 之外呢，那这个 bladder 或者是renal pelvis 这种 upper tract urothelial cancer， 它的 tumor mutation burden 在所有的 IBA 里面，其实相较来说都是还比较高一些的。那确实这些在 bladder 或者是 upper tract cancer， 他们有比较少的 mutation， 或者是比较少的 driving event 的比例，至少相较于肺癌哦。那在这一种urothelial cancer 这个 IBA 里面，它有一个跟upper airway digest 的 cancer 类似的地方，就是 field cancellation。那像刚刚郑院长提的，就是 口腔癌、食道癌的病人常常一起发生。那它的genome的 change 其实会是怎么样？我自己的在这方面的认知就很少。但是有些 cancer 是很有趣的，我们常常会看到在 upper tract 就 renal pelvis ureter 有，然后 urinary bladder 也有，这样一路下来。那在我当住院医师的时候，很多病人会接受开刀，他们把那个叫做 full house 就是他们把两边的 renal 把两边 kidney 跟两边的 ureter 全部拿下来，可能甚至连 bladder 都拿下来，这种整套拿下来的。那我那个时候就有一个疑问就是像这样的肿瘤这么多颗，他们的 mutation pattern 是不是一样的？所以大概在10年前，我就试着做了这样的事情其实到现在还没什么进步，不好意思。但这个资料显示，像这个在左边的这一些一个大的栏位，就是同一个病人的不同肿瘤。举例来说，像这是一个病人的1、2、3、4、5个肿瘤。那在同一个病人，我把它叫做 Group 14，就第14个病人。很有趣的是，在这5个不同的肿瘤，如果你用Cancer Panel 去看 mutation 的话，那像这个肿瘤都有一样的 FGFR3 的 mutation 在这里，那其他的基因的 mutation 也一样。那就整个这样看过来，在这些同一个病人的不同肿瘤，绝大多数它的 mutation profile 是类似的，只有很少数就是这一个跟这一个不同肿瘤之间的 mutation profile 有一点的不同。所以面对 urosal cancer 有时候是 multiple， 但是你会发现其实做其中一个 biopsy 做 genome profiling 其实出来结果是类似的，所以它应该就是一个 field 的 cancerization 的过程。那是什么东西造成它的 field 的这个 cancerization 的过程DNA mismatch repair 的 deficiency 是很多癌症发生的一个原因，就像是在腹癌里面 homologous recombination deficiency 就是卵巢癌产生的主要原因。那在消化系的癌症 MMR 的 deficiency 就是大肠癌跟很多消化系的癌症其实都有。那在 urosal cancer 其实它是一个很 complex 的这个 carcinogenesis 过程。从 urosal cancer 的 profiling 其实我们可以看到 MMR， 也可以看到像这种 Bucky 的 DNA duct 它的 impact。那 Bucky 的 DNA 这个 A duct 在台湾最多的应该也就是像是马多磷酸的这种 DNA duct。那过去在台湾没有办法特别去做这个马多磷酸 DNA duct 的时候，我们大部分都把 signature 22当做这个 Bucky DNA duct 存在肿瘤里面的一个证据但是要做一个 signature 的分析，恐怕这个肿瘤它必须要做的就是 whole exome 或 whole genome 的 sequencing， 就不是一个 panel 的 NGS 可以做到的。那就是在临床使用上，它有一个小小的 gap 存在。很快就被快转了。所以像这样的carcinogenesis， 如果我们要去定义它的话，那就是需要另外一个体学的介入，就是质谱的介入那在食物里面存在的这些马多磷酸的东西，在肝脏代谢以后，其实进到 DNA 之后，它会被 incorporate 在这个核苷酸上面，去氧核糖核酸上面，有像是它会被 incorporate 在 A 上面，变成底 A， 然后再加一个 AL， 变成它结构在这里。那如果它变成底 AL 的时候，这个 A 看起来不会再像 A 它会看起来像 T， 所以我们在复制的时候，另外一股会做成 A， 那把本来的这一股在复制之后变成 T。所以像这样的肿瘤会有很多的AT 的 transversion， 那这个是在 mutation signature 可以看得到的。那所以在 cosmic signature 22就定义了这一种 AT 的 transversion， 就叫马多磷酸专属的。那所以一样，如果我们今天只做一个cancer panel， 其实是看不到这种 AT transversion， 除非它的 tumor mutation burden 很大。那在病因学上面，大概我们也只能看到这种 mutation， 我们也只能说是一个 correlation， 倒没有办法说直接它一定是这样的状况那所以我们大概是把那个 DNA duct 技术建立以后，试着回答一些问题。那其中我们自己觉得很有趣的一个问题是，就是在我们南部很常发生的洗肾，然后肾移植之后又长 urosal cancer 这样的议题。那我们知道很多病人在中了肾脏以后，可以过一阵子还不错的日子，但是在过一段时间之后，那可能因为抗排斥的药的关系，抑制了免疫，那肿瘤是会继续长的，就是会长肿瘤出来，而且是 de novo 的。所以我们 collect 61个这样的病人，这61个病人本来没有 urothelial cancer， 但是在接受肾脏移植之后，那他产生了 urothelial cancer。那我们在想，这些病人他会在这么短的时间之内，那甚至最短可以到2-3个月哦，这么短的时间之内，在免疫抑制药物的状况下，那就长了这个肿瘤。显然他本来他的 urothelium 应该有 something wrong， 可能就是 DNA duct 在那里。所以就是在这样 study 就是 whole exome sequencing 加上一个 LC-MS 去把它的 DNA duct 把它打出来。那讲的是结论，就是这个是在质谱上面直接可以达到的结果，跟它出风的状况哦。那在接受 renal transplantation， 它的 urothelial cancer， 它的 mutation profile 其实是跟其他的没有接受 renal transplantation 长的 urothelial cancer 类似哦，大部分都是有 TP53 的 mutation 100%， 跟 MUC6 的 mutation 也是100%， 但是比较有意义的应该就是 TP53 的 mutation。那其实在它旁边的 non-tumor 的 tissue 也会有一些 mutation 存在。那其实大部分的时候是 MUC6 的 mutation， 但是这里没有 TP53 的 mutation 哦，所以 TP53 的 mutation 是在产生 cancer 之后才出现的。那比较特别的是，我会看到 tumor 跟旁边的 pair 的 normal， 其实 tumor 这里确实有比较多的 AT transversion， 但是 pair 的 normal 没有，所以在它的 tumor 的形成过程里面会逐渐累积 mutation， 从 normal 到 tumor 的过程。那这个累积的 mutation 其实绝大多数都是 AT transversion， 那因为 DNA duct 已经存在，所以它才会一直会产生这个 mutation 哦。所以下面是这些selected case 它的分布，可以看到这是 normal， 这是 tumor。那在 tumor 这里很多是这个 AT 的 transversion。那一个肿瘤的产生，它的mutagenesis 绝对不是 single 的，就是不是只有一种 mutagenesis。举例来说，像上面这第一个 case， 这是它的 normal， 这是它的 tumor。但在它的 normal 可以看到的是signature 01，也就是 agent 相关的 signature， 也就是说它的 mutation 有一部分是来自 agent。那再来就是 signature 05，也就是ERCC2 loss 相关的 mutation， 那还有一些是 signature 30哦。但是在它变成 tumor 的时候，千篇一律产生了 signature 22。所以在这一类的肿瘤，在肾脏不好接受洗肾，而且在中了肾脏之后，这些新产生的 urothelial cancer 其实绝大多数都是跟马兜铃酸有相关的，这可能是我们很特殊的背景哦。那跟 signature 22有点不一样的是 ，signature 22它不太容易被定量，但是这个病人的 normal 的 urothelium 里面的这些 carcinogen 是可以被定量的，而且这个是 carcinogen 的浓度，就是 A duct 的浓度。那这个是产生 secondary， 应该说 de novo 的 urothelial cancer 的时间。所以当这个浓度越高的时候，产生的时间会越短，越容易产生。那再来就是这些病人其实都会用一些免疫的抑制剂哦。那这个免疫的抑制剂在特定的情况下可能会对这病人的 outcome 造成影响。比方说，如果他是一个 upper tract urothelial cancer， 那他的 bladder recurrence free survival 其实就跟有没有用 sirolimus 这个药物相关哦。如果用 sirolimus， 其实他的 bladder recurrence free survival 就会变得相较短很多。那在这个 transomic 的 approach 里面有一个是必须被提到的，就是mutagenesis 的产生，它不见得是因为 DNA mismatch repair 或其他的东西造成的哦，因为在我们的身体里面mutation 跟oncometabolite 其实息息相关，所以 IDH2 的 mutation 会有 oncometabolite， 或者是SDH complex loss of expression 也会有 oncometabolite。但如果返回来，一个 metabolite imbalance 会不会造成 mutation？ 其实是会哦。那我们来看这个在2018年的 Cell 这篇 reference， 它告诉我们，如果身体里面有很多这个氮，那它会产生氮鱼，就是 N 这个氮哦，它会产生氮鱼。那这个氮鱼产生的结果是，它会把，如果真的太多的时候，它会把任何的东西都塞上一个氮。那比方说，如果是pyrimidine 跟 purine 的时候，它就会 favor purine 的 biosynthesis， 因为 purine 比 pyrimidine 多一个氮。那在 amino acid 的 biosynthesis 的时候，它也会增加，因为它会做很多 polyamine， 会做很多 amino acid 的，所以这个 carcinogenesis 过程会受到影响。那也就因此， 在一个 urosilical cancer 的 mutagenesis 的过程里面，其实比我们想象的复杂。我们最终看到的都只有这个 mutation 的 signature 的形成，这是我们每天在分析，也就用来治疗的病人的一个工具哦。但是其实在最终它的背后，其实有一些，像在台湾来说，它可能会有 DNA duct 的影响，那它可能会有 DNA repair 的 deficiency 的影响。那因为 A duct 造成mutation， 那 repair 又 deficiency， 当然会产生特定的 mutation 的 signature。那除此之外，这个 metabolic dysfunction 其实它可能造因于 genome 的 alteration 或 epigenetic change， 但这些 metabolic dysregulation 也会间接造成mutation 的 bias。所以画一条线。当我们在右边看到一个特定的 mutation 的 signature， 或者是 mutation 的 pattern 或 gene fusion 造成 aggressiveness 的过程，或是造成可以治疗的过程哦。但是后面我们其实还可以多看到的是这个特定的 mutation 它产生的原因。那要问这样的问题或者做这样的应用，那就是非得要让我们的这个除了 companion diagnosis 之外，那也要延伸精准医疗检测的范围。好的，那以上是我的报告，谢谢各位。
谢谢李主任啊，针对以这个 urosilical cancer， 那带我们更深入的去 explore 当中一些mutation 的这些signature， 以及怎么样对于未来我们的研究上的一些启发。那这边就开放各位先进以及师长线上各位同仁的提问。那我这边先提一个小问题哈，那也是一个就是我在行医当中也会想到这个一些困境，就是说，就是如同刚刚李教授您提到，我们今天去看，这个 tumor 是一个 urosilical tumor， 它的一些 mutation 的 profile， 它的一些，比如说它 DNA damage repair 的一些 deficiency， 或者是说它一些种种的变化，但是呢，它其实前面还有很多的过程，比如说它这个是别人的 kidney 给他的，那这个是从其他的地方进到一个宿主里面，那还有宿主它本身的一些metabolic 的问题，所以说今天我们就如同先前的何教授讲，那我们都会专注在 tumor somatic 的 mutation， 那针对germline 的部分以及 host 它本身对于比如说同样都是接受这种 immunosuppression， 那有的每个人当然他的metabolic 的能力 drug 的 metabolic 是完全不太一样的，所以这些东西怎么样去 impact 到后面这个 tumor 的这个 carcinogenesis？ 那您对未来的研究有什么样的这样子的一些指示？
对，确实，如果能够分析的话，资讯越多，我们治疗一个病人的武器就越多。那不是临床的，它现在不会是临床的常规，但是我们如果看到一些 approach， 像是如果一个肿瘤你去做 whole genome 的 transcriptome 的时候，那其实你会看到的是这个肿瘤的表现之外，里面潜在的 virus 的这个 impact 也会在里面嘛。那其实我们会知道它是不是有像对 urosilical cancer 来说啦，它有没有像是 BK virus 或其他东西？那其实对于在规划整个治疗可能又不一样哦。那如果这个 transcriptome 它是可以延伸到旁边的 surrounding 的 tissue 的话，那可能对免疫会有更多的了解。所以这些事情是如果一想，你就会觉得要做的很多，但是做多了以后，那恐怕也是要有更 solid 的这个 cohort， 那更 solid 的分析才能够得到临床上可能应用的 information。谢谢。
好，刚刚好像对是何教授。
我因为是外行，我刚才听到一个，我不知道有没有听错，就是说，您刚才在解释那个 transplantation 之后，他又在复发的这群病人，他那个复发的这个genome里头，他马兜铃酸的成分还是就基因爱导还常马兜铃酸的这个toxicity还会存在。那这些马兜铃酸的这个，它是从哪里来？还是从食物过来？还是他身上原来有的？这我是觉得比较好奇，我不知道我刚才有没有听错。
感谢教授提问。其实这个就是我们一般种肾脏的时候是直接把这个新的肾脏种进去嘛，本来肾脏还留着。那新的肾脏不见得会出问题，但是出问题的都是本来这个 recipient 他自己身体里面的肾脏。 所以这些它会这么快就可能有的时候真的2-3个月就长，有一个原因就是它的这个 DNA duct 已经存在在所有的看起来正常的 uracilium 里面，那它是等待一个机会，等待那个它的 immune 被抑制之后，那它里面的这些不正常的增生就可能被放大，所以它的过程大概会是这样。谢谢。
还有一个很重要的就是 epigenomic methylation 的部分。那在这种情况，这个 methylation 在这里有没有什么样的角色？
目前确实研究的大部分都只有 coding region 哦。那至于像这样的 ADUCT 会不会对structure 或者是 epigenomics 造成影响，其实这个有人在做，但是还没有结果。谢谢您。
就是说，过去我们常看到一些gene expression array 啊，那不晓得对这种 epigenomic 的这种的有一些 array 可以有这种平台让我们来参考。
过去有一些像Illumina 好像有过一个最多到450嘛，从 Illumina 27到450，但就是以做的密度来说，可能现在都不够高了，所以现在应该都是直接上 NGS， 就是用类似 methylation binding protein 或者是匕子把 methylation 的地方抓下来，然后上 NGS， 可能会是比较全方位的做法。
是。那刚刚在上一场有提到，就是说关于事实上呢，现在很多北中南都有很多的病人 tissue 血液都送去。比如说像行动基因啊，或者是 foundation， 那事实上，像在行动基因已经累积了蛮大量的这个资料库了。那我们不见得就是说能够其实现在学会也在这个杨校长这边理事长这边在 organize 说怎么样去跟他们合作啦，去把这些很大量的资料我们都能够拿出来，甚至一些 raw data 再做一些分析，这样我想对临床也会有很好的帮助。那这边这个李教授也可以提供一些您的这个经验吗？
我比较没有这么做，因为我也不太知道，我不太确定那个时候他们收这些简体，在同意书的范围有没有包含？应该会是有，对不对？就是包含某个程度的再分析嘛。那个再分析能不能够给我们做，就是可能就另外一回事哦。所以我们有时候会在 paper 上面看到，有的 data 是去跟像是 foundation 去 request 来的，然后再重新分析的嘛。那这个流程我就没有跑过，对，不好意思。
我相信那边有很多的资料都可以让我们得到很好的一些研究的一些结果，做一些 translation。是何教授。
抱歉，因为李部长在这里，我真的很想再问最后一个问题了。那个你刚才讲到呢，那个 multi 的 omic 哈，我们知道 gene RNA、DNA RNA， 我们还有个 protein。那你刚才提到的是那个 genome， 包括那个 transcriptome 应该是 RNA 嘛？那 protein 的那个叫做 proteomic， 你对这个部分或是那个 kinase array 的这个部分，我不知道你对它的这个评价怎么样？当然，我们知道 protein 的部分在分析上会比这个基因 DNA、RNA 又更麻烦，因为它没办法放大，它没有办法放大，它就是你就是拿多少组织，你就只能做多少事。那这个部分，你的想法是什么？你的对它的整个长远的发展是怎么样？因为你今天提到的那个 omic， 我就想再多请教这个问题。
是，如果教授记得，我的第一张 slide 讲的就是，其实是一种 proteomics， 就 peptide sequencing 的过程，就那个是，对，就是把 MHC complex 抓下来，然后打 mask 的过程，其实那个就是一个。那我觉得那个 proteomics， 对不起，应该是说，客观看起来 proteomics 应该未来会是一个方向，但它遭遇到一些困难，就是它的方法学其实 reproducibility。其实没有基因学这么高嘛，因为这 PCR 已经发展这么久了，那 NGS 这些东西又都 commercialize， 所以 proteomics 如果有一天它的操作流程也可以非常的 protocol， 甚至 fully automation， 那你要去定量里面的它的不同的位点，像 threonine 啊，或是 tyrosine 的 phosphorylation 也都可以精准定量，我觉得那个会是一个未来很好的方向，但它现在差的就是方法学没有办法一致化。造成它 reproducibility 并不好。谢谢。
好，那就如同刚刚李教授有说，事实上在美国他们有一些 center 啊，当然已经很自豪，他们可以说 we sequence every tumor。那我想可能还是 cost 的问题啊。那未来如果说价钱能够更便宜，能够少一个零啊，那我们可以更自豪的说 we sequence every metastasis。那我想从 early stage 啊到更 advanced stage 的时候，我们可以有更多的这些data 啦，可以给我们做一些治疗上的这个启发。那今天就感谢李教授今天的演讲，以及很知无不尽知无不言言无不尽的这样子的跟 Q&A 跟大家的启发啊，再一次感谢，谢谢。
那等一下是coffee break 时间，然后到3点20分的时候再请在场来宾还有线上来宾再回来，谢谢。 各位来宾还有线上来宾，那大家午安。那再来我们要coffee break 后的第一个 session。那我们很开心邀请到 Professor Daewoo 来帮我们讲所以遗传性癌症的 Precision Medicine。那我们邀请台大医院肿瘤医学部卢彦生教授当我们的座长。
This is Yanshen Lu from National Taiwan University Hospital. This is my great pleasure and honor to introduce next speaker, Professor Shahina Daewoo. Professor Shahina Daewoo is a consultant medical oncologist and professor in clinical oncology in the UAE. She trained in medical oncology at McGill University Hospital in Canada and subspecialized in breast cancer management at MD Anderson Center. She also serves as president of the Excellence in Oncology Care Summit that is held annually in Dubai and is co-president of the Asia Pacific Breast Cancer Summit and Asia Pacific Gastroenterology Cancer Summit. She has also recently launched the International BRCA Forum and Young Women's Forum. Currently, maybe a lot of facts we know this and that, but the funny thing is that indeed many of them are initially presented by Professor Shahina Daewoo. For example, she is the first one to show the data that with the available of the trastuzumab, HER2-positive metastatic breast cancer has changed, become from a poor prognosis cancer to become a disease with good outcome. Also, she provided evidence, tell us that de novo stage 4 breast cancer, the prognosis is different from the recurrent disease from early breast cancer, and etc. and etc. So you will be very amazing that she is capable to dissect many important clues from a simple or chaos situation than to several facts to condense it into a very important knowledge to us. So we just very happy that she was invited today to give us this talk. Let's welcome Professor Shahina Daewoo.
Good morning, and thank you for the invitation to the symposium. My name is Shahina Daewoo, and I'm a medical oncologist from the United Arab Emirates. I've been tasked with talking about precision medicine in hereditary cancers. These are my disclosures, and if anybody would like to get in touch with me, these are my social media accounts and a barcode that you can scan to access. So let's start off by asking the question about precision medicine in the management of cancers. What did we learn, in fact, from targets? We have learned that identifying one important somatic mutation can actually make a difference. And here you see an example of a patient with metastatic melanoma, where we know in the past a single agent chemotherapy such as dacarbazine was associated with a poor response. But if you identified a somatic mutation such as a BRAF V600 mutation and targeted it with drugs such as vemurafenib or any other anti-BRAF, you had a spectacular response. So one gene is in fact good. And we have in fact seen across a spectrum of different cancers that matching for these targets with therapeutic agents can in fact make a difference. You can see examples from non-small cell lung cancer, pancreatic cancer, as well as breast cancer, where patients with highly actionable alterations who received a molecularly matched therapy lived longer than patients who did not receive a molecularly matched therapy. We've now also moved into an era where there's a rapid adoption of tissue agnostic testing, which is really changing the way we think about targets, where you have a target that could be present on any tumor, and you have a particular medication for that particular target, and it will work. So one target, one drug, all tumors. And examples of this include MMR deficiency or MSI high tumors, where you can give pembrolizumab, TRK fusions where you can give leratractinib. We know that RET fusions have now become an agnostic marker where there are a plethora of different medications as well. And we know that tumor mutational burden has also now become an agnostic marker where you can give immunotherapy. So this is just a slide that shows you an example of studies that have looked at MMR deficient tumors where you can give single agent pembrolizumab and have really good response rates post progression on multiple lines of therapy. And here you have the waterfall plot of a study that has looked at patients across a variety of solid tumors that have a TRK fusion, where the agent larotrectinib was associated with an overall response rate of 76%, and 93% of the responding patients and 75% of all patients actually remain on treatment and underwent surgery with curative intent. So agnostic markers do work with their associated therapeutic agents. But what about precision medicine, specifically within the realm of hereditary cancers? Now, when we're testing for different mutations, there are two types of mutations that we need to be cognizant about. We have germline mutations, and I'll specifically focus on BRCA mutations because that's where a lot of the hereditary cancers actually come from. So you have germline BRCA mutations where we know it's inherited from a patient, it's found in all cells and can actually be passed on to children. Then you have somatic BRCA mutations, which are acquired mutations found only in tumor cells, and these are not passed on to children. We know that the incidence of BRCA mutations across a variety of different BRCA-associated tumors are different. Hormone receptor positive breast cancer associated with about 6% incidence of BRCA mutation, triple negative breast cancer about 17%, ovarian cancer about 25%, pancreatic cancer about 4 to 7%, and prostate cancer about 5 to 10%. Now, there's been a number of questions that has impacted our understanding of BRCA-associated tumors, and I think these questions are very important when we're trying to think about managing these patients. First is, why is it important to test for BRCA in affected as well as non-affected individuals prior to the era where we had PARP inhibitors that could potentially target BRCA mutations? And the simple answer to that question was, it was for risk management of patients and their relatives to reduce the risk of different cancers developing. And if you look at the example of trying to monitor and prevent breast cancer from in individuals who have a BRCA mutation, we do have several strategies such as screening. We know that in patients with BRCA mutations, there are different screening programs, including the use of mammograms and MRI breast. There's risk-reducing surgery, including bilateral mastectomies, as well as you know, there's a higher incidence of ovarian cancer associated with a BRCA mutation. So bilateral self-pinge oophorectomy is a way of reducing risk. And then, of course, there is risk-reducing therapy with the use of agents like tamoxifen and raloxifene that has been shown to reduce the risk of developing breast cancer in patients with BRCA2 mutations. The next question is, in the era of PARP inhibitor, why was it important to test for BRCA? And I think this, for us as medical oncologists, is probably the most important question that we're going to ask ourselves. And really the simple answer is, it's important because it's clinically important to inform on potential responses to treatment. And we have a number of important clinical trials that have looked at the use of PARP inhibitor therapy in patients who have a BRCA mutation across a variety of solid tumors. We have the Olympia-D trial that looked at the use of olaparib in patients who have germline BRCA mutation and metastatic breast cancer. You can see that olaparib was superior to physician choice of chemotherapy. You have the SOLO-1 trial that looked at the use of olaparib in patients with advanced ovarian cancer as maintenance therapy. And you can see olaparib again was superior to placebo in the maintenance setting. You have the POLO trial in patients with pancreatic cancer, where olaparib was used as maintenance therapy in patients with BRCA mutated pancreatic cancer. And again, olaparib trumped placebo. And then, of course, you've got the PROfound trial in patients with prostate cancer, where olaparib was used, comparing it to physician choice of anti-androgen therapy in patients with HRD deficient prostate cancer. But in this cohort, you can see specifically those patients with BRCA mutation positive disease. And again, olaparib trumps novel anti-androgen therapy. The next question is, who should be testing for BRCA? Should it be the oncologist or the geneticist? Well, we need to understand the problem. Traditional referral routes to the geneticist may not meet clinical needs. There has also been a shift in focus from risk management to treatment decisions where the oncologist plays an important role. Guidelines have expanded the clinical criteria for testing, where oncologists again play an important role, and treatment-focused genetic testing has triggered an increase in demand for genetic counseling services worldwide that has put a lot of pressure on a variety of hospitals. We also know that when a patient with an individual with a BRCA mutation, if that individual has a cancer, the MRP is the oncologist. But if that individual does not have a cancer and is just at high risk because of a BRCA mutation, the MRP is not the oncologist, and then a different route has to be taken in terms of the counseling and the testing. We also know that there is a lack of genetic counselors available, and you can see from this pictorial diagram published in 2019, if you take an example of Asia, more than 350 genetic counselors present compared to America, where you have almost 5,000 genetic counselors present. So again, per population, we have not many genetic counselors in our part of the world. The other problem is if a geneticist or a genetic counselor is tasked with actually testing for BRCA, you can see that the turnaround time would be very long in an individual with cancer versus if a medical oncologist tested for BRCA. So again, you want that rapid information so that you can make your therapeutic decision, and so it may be more appropriate for a medical oncologist to test. The other challenge that an oncologist faces is what test to in fact order. Should we be ordering tumor profiling so we can get both somatic mutations, and there may be germline mutations hidden, but we cannot distinguish between the two. Germline testing will give you germline information, but you may miss somatic testing. And then of course you've got liquid biopsy that gives you somatic information, but you may not get the biomarkers that you want to get if the patient is not shedding enough cancer cells into the system. So again, this is also a discussion that needs to be had in the molecular tumor board as to which is the most appropriate test to look at. Now, when should we be testing for a BRCA mutation? Well, I'll give you various examples highlighting the need for early testing. If you look at the example of patients with prostate cancer, who have a BRCA mutation and who have castrate-resistant disease, you can see from the PROfound trial that those patients who received olaparib with no prior taxanes given, they did much better than in those patients who had received a prior taxane, suggesting to us that if you test early and you have the information early, you can make those treatment decisions. And at least for me, when I'm in the clinic and I have a patient with the castrate-resistant prostate cancer and I find out that they have a BRCA mutation early on, and I will, for those patients with BRCA1, BRCA2 mutation, I will go on to give them a PARP inhibitor first before chemotherapy. And if they have any other mutations in that DDR mutation, in that DDR pathway, I may think about chemo first before PARP inhibitor first, because we know that the impact of PARP is not that high in patients who have prostate cancer who have other mutations in that DDR pathway. Now, let me give you some other examples. This is an example of a patient with a germline BRCA mutation and ovarian cancer. This is a young woman, she's 40 years old. In 2018 May, she presents with abdominal discomfort and is diagnosed with ovarian cancer. She has a sister who has breast cancer at the age of 35, and when we tested her, she does have a BRCA1 mutation. She does receive pre-op chemotherapy with Taxol and carboplatin, and then has surgery in November of 2018. At this point, even though she has a BRCA1 mutation, she does refuse olaparib and she refuses bevacizumab. And at that point, she also refuses referral to a genetic counselor. Now, unfortunately, in September 2019, she does have recurrence of disease. And we think about, again, whether we should be giving her a PARP inhibitor at that point. And we do have results from the SOLO2 trial that looked at patients with germline BRCA1, BRCA2 mutation in the recurrent platinum-sensitive setting. And you can see that giving olaparib in the maintenance setting, in this platinum-sensitive recurrent setting, there is a 12.9-month improvement in overall survival. So going back to my patient, at this point, she did agree to get chemotherapy with gemcitabine and carboplatin. And post that, she did agree to get olaparib as maintenance therapy. And she has ongoing, this is an older slide, but we started her off on olaparib in January 2020, and she actually is still on olaparib. Her main concern is, will the presence of a BRCA mutation actually impact her insurance, which is a very valid question. The second example is of a lady who also has ovarian cancer, but this time she has a somatic BRCA mutation. And we know that germline BRCA mutations in patients with ovarian cancer accounts for about 14 to 21%, but you could get another additional 12% of patients who have pure somatic BRCA mutations. Now, this is not only for ovarian cancer. We also know that in prostate cancer, if we were to test only germline, you can see, for example, BRCA2 mutations, the prevalence is about 3.5 to 5.3%. But when you combine germline and somatic mutations, you can see BRCA2 mutation incidence can increase to almost 8.7%. So let's go back to that ovarian cancer example. This is a young woman. She's 35 years old. She presents with ascites and omental caking. And she's diagnosed in December of 2020 with ovarian cancer. She gets upfront chemotherapy followed by surgery. She has no family history. And when we tested her for germline BRCA, she had no pathogenic BRCA mutation. We sent her tissue for tumor profiling, and we did find that she had a somatic BRCA1 mutation, and she was commenced on olaparib. Now, we know in this first line maintenance setting, we have a lot of data to support giving maintenance olaparib. Now, as I put this question, her main concern of this patient was, would it impact her children? So we have two data sets that support the use of olaparib in the maintenance setting. We have the SOLO-1 trial in the first line setting. These patients advanced ovarian cancer, post-platinum-based chemotherapy, randomized to receive either olaparib or placebo. You can see that a descriptive analysis of overall survival at 7 years shows a clear-cut improvement in overall survival in these patients when you're giving them maintenance olaparib. We also have the PAOLO-1 trial, which randomized patients in the same sort of a setting, maintenance, first line, they got either olaparib plus bevacizumab versus bevacizumab. And again, you're seeing in that HRD positive subgroup at 5 years, there is an overall survival advantage. You have the third case example is also another ovarian cancer example to just try and show how we can expand the cohort of patients that could benefit from PARP inhibitors. We know that patients with ovarian cancer, a significant proportion of them will have a BRCA mutation, either germline or somatic, but you will also have patients who don't have a BRCA mutation but have some mutation in their homologous recombination pathway. And these are called patients who have HRD positive BRCA wild type disease. Could they potentially benefit from a PARP inhibitor? And the simple answer is yes. They can potentially benefit from a PARP inhibitor in the maintenance setting, in the advanced ovarian cancer setting. Again, these are platinum sensitive patients. First line patients, we have data from the PRIMA that used niraparib, Athena that used rucaparib, and of course the PAOLO trial that used both olaparib in combination with bevacizumab. And you can see there's a clear-cut improvement in progression-free survival. So coming to this particular patient, this is a 30-year-old lady. She has no family history. And during a C-section for her baby, she was found to have ovarian masses and diagnosed with stage 3 ovarian cancer. This was a high-grade serous ovarian cancer. She had optimal debulking of her disease, and following which she had adjuvant chemotherapy. She had no germline BRCA mutation. We did send her tissue for profiling, and we found no somatic BRCA mutation. But we also sent her tissue for HRD testing, and she was found to have HRD positive disease, and we commenced her on olaparib and bevacizumab. Again, her main concern, because remember she was pregnant when she was diagnosed, was will this impact her baby? Now, the fourth example is another example of a patient with breast cancer who has a germline BRCA mutation. We know that in patients with metastatic breast cancer, if they have a germline BRCA mutation, we have two trials, the Olympia D trial and the EMBRCA trial, that took patients with platinum-sensitive disease, randomizing them to either physician choice of chemotherapy or a PARP inhibitor. You can see that using a PARP inhibitor does improve prognostic outcome in terms of improving progression-free survival. So here's a case of a 38-year-old woman whose mother had ovarian cancer at the age of 42, and this patient presents with a breast mass in October of 2018. It's a triple negative breast cancer. She has metastatic disease. She's found to have PD-L1 positive disease, and she's subsequently commenced on chemotherapy plus immunotherapy. Now, by August 2019, she develops brain metastases. And by then, we also find out that she has a BRCA1 mutation positive disease. She has progression of her disease by November 2019 in her lung and in her liver. And at that point, we commenced her on a PARP inhibitor with olaparib, and she stayed on olaparib for approximately 10 months until she had progression of disease in August of 2020. Now, the use of PARP inhibitor in breast cancer has moved from the metastatic setting to the early stage setting, and we know from the Olympia trial that giving adjuvant PARP inhibitor in the form of olaparib in patients with a germline BRCA1, BRCA2 mutation who have a HER2 negative disease and those high-risk group of patients, it does improve prognostic outcome. And you can see that it has been incorporated into the guidelines. But oncologists face a number of different questions in the early stage breast cancer setting, where patients will ask, should I be testing my children now? And of course, I always send my patients to the genetic counselors to be able to answer that question. Children under the age of 18 should not be tested routinely unless specified by the genetic counselor. Do I have to remove both my breasts? And this is a very important question that early stage breast cancer patients will ask if they have a BRCA1, BRCA2 mutation. The simple answer is no. We know from multiple studies that yes, when you have a breast conserving surgery versus a mastectomy in the presence of a BRCA1, BRCA2 mutation, there will be a higher local failure rate in those patients with breast conserving surgery. However, the overall survival is not different between the two groups. Do I have to remove my ovaries now? And that's another question, because we know patients with a BRCA mutation do have a higher risk of ovarian cancer. And as you can see, the cumulative ovarian cancer risk to the age of 80 years is about 44% for patients with a BRCA1 mutation, 17% for BRCA2 carriers. And that risk in BRCA1 carriers starts at about the age of 40, and BRCA2 carriers starts at about the age of 50. And again, you need to counsel your patients appropriately. And does this gene, that means a BRCA mutation, mean that my cancer will come back faster? And the simple answer is no. That's what you tell your patients. We have data from the POSH trial that clearly shows that whether you have BRCA mutation positive disease or negative disease, this is a prospective trial of 127 institutes in the UK, it shows that there is absolutely no difference in the overall survival of these patients. So what do I do in my clinic? In my clinic, if I have an affected patient with a BRCA-associated tumor, the MRP will be the oncologist, and the MRP, that means the oncologist, will do the counseling and the consenting for testing of patients with ovarian cancer, advanced metastatic breast cancer, HER2 negative, pancreatic cancer, prostate cancer, as well as early stage early breast cancer. Now, in patients with metastatic ovarian, metastatic breast, pancreatic, or prostate, we ideally send the tissue for profiling. And if the patient can afford it, we do simultaneous germline testing. And if they cannot afford it, we will do germline testing only if there is a germline, if there is a somatic mutation that's detected. And it's the same for early stage breast cancer setting. We do tend to not send the tumor for profiling. We send for germline testing upfront. Now, if a BRCA mutation is detected, or other mutations are detected, or a VUS is detected, all these patients are sent to the genetic counselor. If no mutation is detected and there's no VUS, but the patient has a strong family history, we still consider referral to the genetic counselor. So in my final slide, I think Precision oncology as a framework for patients with cancer in 2022, this is what it looks like. Patients with cancer should be biopsied, and then their tissues or their germline should be sent for sequencing so we can determine if they're biomarkers of interest. All information should be discussed in a molecular tumor board, which composes of a molecular pathologist, an oncologist, a geneticist, and a plethora of different specialities that will all look into the logistics, the cancer heterogeneity, and the magnitude of clinical benefit for any of the therapeutic agents that will allow us to give a comprehensive therapeutic plan that will best benefit our patients. So thank you very much.
So thank you, Professor Wu, for your comprehensive talk. So is there any questions from the floor or one from the web? You can type in your question in the conversation, and we will ask the question represented of you. So Professor Dawu, may I ask the first question for you that in the number 4 case, you mentioned that she received immunotherapy as a frontline treatment because she has PD-L1 positive, but she happened to have a BRCA1 mutation. We know that immunotherapy, the efficacy is most prominent in the first-line setting, but PARP inhibitor would not be considered as a chemotherapy, right? And on the vice versa, that using PARP inhibitor, it seems that overall survival can be represented in the first-line setting. So for this patient, if she has a PD-L1 positive and BRCA1/2 mutation, either both BRCA1 or BRCA2 mutation, what would your recommendation of the sequence of these 2 treatments?
That is an excellent question, and thank you for that question, because I'm a big proponent of using PARP inhibitors as early as possible in patients with BRCA1, BRCA2 mutations. Now, in this particular case, I actually had the information for her BRCA after I started her chemotherapy. And that was one of the reasons why I didn't start her PARP inhibitor up front. Now, the controversy, and I do have separate slides for this. I usually love this topic. I love discussing about it. If you see my algorithm for patients with triple negative breast cancer, everyone has to be tested for PD-L1. Everyone has to be tested for BRCA. In a patient who is PD-L1 positive and BRCA mutation positive, and they are not in visceral crisis, I think it is reasonable to use a PARP inhibitor rather than using immunotherapy plus chemotherapy, although that is still also a very good first option because there is an overall survival advantage. We've seen that from the data, but there's no head-to-head comparison. So when I have a patient like that, I give them both options, and I give them the data, and I tell them I don't have an overall survival advantage for PARP, but then again, those trials are different compared to this clinical scenario. But if a patient is in visceral crisis, I will give them, and they're PD-L1 positive, I will give them chemotherapy plus immunotherapy first, and then go on to a PARP inhibitor. Now, when you go back to the Olympia D trial and you look at those patients who did not receive chemotherapy first, as in the PARP inhibitor was the first line of therapy, there was an overall survival advantage in the post hoc analysis. So again, I think it's an area of controversy, and I think patients should be given that option, especially if they're not in visceral crisis.
Okay, so may I ask the last question that if a patient have a BRCA1/2 somatic mutation, that means breast cancer, but after the test, she do not have a germline mutation. In that situation, will you consider a PARP inhibitor will be also available if she already refractory to prior chemotherapy? And then why you can think that, what is the reason that so far breast cancer is different from the ovarian cancer that I know that there's a small trial also show that it still have an efficacy of PARP inhibitor for somatic mutation, but why initially we believe that the efficacy of the PARP inhibitor were not so good in somatic BRCA1/2 mutation in breast cancer?
So actually, the data does support use of PARP inhibitor in breast cancer when there's a somatic mutation. We have the TBCRC trial that shows that use of olaparib in patients with somatic BRCA, there's an overall response of 50%. We have the results of the opinion study that also show that in the presence of a somatic mutation, olaparib works very well in patients with breast cancer. So I think there is very good evidence for use of PARP inhibitor in patients with somatic BRCA breast cancer. The problem is that only 3% of patients with the breast cancer will have a somatic BRCA, and so the incidence is very low. So to try and get that one patient who has a somatic BRCA mutation is actually quite rare. I have actually now 3 cases in my clinic, and the reason being is that I profile every tumor that is metastatic HER2 negative at the very beginning, so I can get that information. And if the patient has a somatic BRCA, very reasonable to use a PARP inhibitor.
Thank you. Because time is up, I would like to conclude this section. And thank you, Professor Wu, for your wonderful talk. Thank you.
Thank you very much.
下一场，那我们邀请Dr. Martin 来帮我们讲解关于 comprehensive genomic profiling 在临床上的应用。那我们请林口长庚肿瘤科吴教恩教授来帮我们主持。
Good afternoon, everyone. It's John Wu from Chang'an Memorial Hospital. It's my great honor to introduce today's speaker, Dr. Martin. He is an international scientific lead in Roche Foundation Medicine Pharma International. And Dr. Martin was awarded bachelor degrees in biochemistry from the University in France in 2006, and went undergraduate with the master degrees from University of Edinburgh Medical School in 2007. She completed his PhD degrees in genetic epidemiology from the Queensland Institute of Medical Research and the University of Queensland in 2013. From 2003 until the complete of his PhD degrees, his study field, the research of his fields was focused on genetics of complex trials and disease, and his work has been published in several peer-reviewed journals such as Genome Research and Science. After he completed PhD degrees, he worked on several different pharmaceutical companies, and nowadays he is the international scientific lead in Roche Foundation Medicine. And we know that in cancer precision medicine, we always prescribe the genetic testing for the patient with advanced cancer to find out the suitable treatment for the cancer patient. So I consider she is the best, the most suitable speaker to introduce this topic today about the putting comprehensive genomic profiling in clinical practice. So let's welcome Dr. Martin. Okay, thank you.
Thank you very much for the kind introduction and for the invitation. And thank you also to Professor Dawood to setting up the scene beautifully to talk about testing in cancer. So today I'll talk to you about comprehensive genomic profiling and how it's a tool to open possibility in personalized healthcare in the field of oncology. So what are we talking first about this vision of personal healthcare? So it's really looking at different things. So it's bringing together the data, the technology, and the analytics behind it that will provide deeper insight into the disease to do 2 things. 1, accelerate research, and 2, accelerate development, of course. And on the second hand, also improve patient outcome. And you can see in that infinity feedback loop, that 2 sides of the coin for personalized healthcare. But if you look closely at that feedback loop, you can see like important things along it. So on the clinical trial side, you got discovery of novel biomarker to get deeper disease insight, or on the other side, early personalized diagnosis to get personalized treatment plans. And that's where like CGP is really important to bring this information. Now, to reach that approach, as you all know, it cannot be done alone. There's a lot of different stakeholders. Of course, the patient is at the center, physician like yourself, but you need to bring a lot of different other stakeholders like biopharma company, like Roche, where I am, tech company, policymakers, payers, and stakeholders we maybe haven't thought in the past as well. And then you also need to bring the right tools, and the other one is this inner circle, from EHR and clinical data, digital monitoring, etc. But as we see, genomic and molecular data become a critical part of this personalized healthcare environment. Now, we talked about genetic markers. Just to illustrate, why are we talking about comprehensive genomic profiling and its needs in today's clinical practice and its relevance? Look here, the number of genetic biomarker-driven drugs that have been approved in recent years. I think it's astonishing. Since 2019, the number of single biomarkers that we had approved. And if you look at that in a slightly different lens here, if you cluster it by different tumor type, just taking the now this concept of tumor agnostic biomarkers, that if you have a mutation, if you have a targeted drug, theoretically it should work across all different tumor type. That's something that we see more and more coming. And we already know, looking at from an FDA lens, that we already have NTRK markers, the MSI, DMR, TMB high, and also BRAF. However, if we look at what's coming, over the next 2 years, it's projected that it will increase to 15 different markers. And this is one CGP will really come into play as a strong candidate tool to interrogate if there any mutation alteration in those genes. How does it look like from a guideline side? As you can see, ESMO now recommends the use of genomic testing in an increasing number of metastatic cancer because of that body of data that is coming through. And similar timeline, it's keep increasing, and the recommendation change fairly quickly to incorporate some of this new data that is coming through. Similarly, if you look at the guideline coming from ASCO now recommend for patients with advanced or metastatic cancer should undergo genomic testing using multi-gene panel-based assays if more than one biomarker-linked therapy is approved in that patient disease. Again, sees the change that we've seen here with the different shade of blues by tumor type. It's astonishing. Now, as I say, it's been data-driven, and there's many clinical trials in precision oncology that have been done over the years. Picking on a couple here just to bring you the example. The first one is the DROP study, which is a study coming out of the Netherlands, where the aim was to identify efficacy signals for approved targeted therapy or immunotherapy outside of their label. So as you can see here, the patient went through NGS testing. In that case, it's whole genome sequencing. And depending on what they found, if there was a drug that existed at any oncology indication, the patient was access to it. And the study so far has shown, at least for the first 500 patients, that's already a 33% overall clinical benefit. And that was defined as a confirmed objective response or stable disease for more than 16 weeks. Similarly, at Roche, we are investing in this type of clinical trial to drive this concept of precision oncology. And one of the trials that we have ongoing at the moment is the Tapestry trial, so tumor agnostic setting, where people are identified via a local NGS testing. Typically, it's like being tissue only, and then it's confirmed centrally by tissue and liquid. And again, those patients will be, depending on the biomarker identified, go into one of the different arms. So, as I say, putting into context the use of comprehensive genomic profiling. So what are we talking here as a tool? So comprehensive genomic profiling at least as we see it within Roche and Foundation Medicine. So it has to use this next generation sequencing approach, the NGS. But it's how you use the tool. It has to detect both novel, but also non-variant, but across 4 main class of genomic alteration. And also we hear about those genomic signature, you know, MSI, TMB, LOH now. And all those class of different genomic alteration have to provide at the end either or prognostic, diagnostic, or predictive insight that inform either research, if in a research setting, or treatment decision for individual patient across all cancer type. So I wanted to be clear on the definition, because with that definition, a lot of those smaller panel falls out of what is genuine CGP. Now, as I say, here, just an illustration of what we do within the Foundation One assay, whether it's with liquid biopsy or tissue, looking at all those different class of alteration plus genomic signature like TMB and MSI. But what we learn now from the data coming through is when and why should I use genomic profiling? And you can see here a bit of like a timeline of a patient journey going from diagnosis to the first treatment, to surveillance, to going to a second treatment. CGP has been mainly used at past the diagnostics stage, and mainly on the what do you call it, on the tissue based here. But ideally, using tissue and liquid biopsy is the ultimate combination to do, because it really brings you a full picture of the heterogeneity of a metastatic disease. And also you can use CGP later on when there is recurrence to potentially identify some treatment recurrence and some mutation that are there to make that recurrence. But I'll talk to that a bit later as well. So what do we know at the moment about broad NGS-based genomic profiling? Here's some data that comes out of the Flatiron database, looking at outcome biomarker testing for EGFR, ALK, ROS, and BRAF in patients that were newly diagnosed with advanced non-small cell lung cancer. And we saw that if you use NGS testing, it may help to avoid potentially mistargeted therapy options. And it's partly due because it's improved test uptake for the recently approved biomarker. And the frequency of unsuccessful genotyping was lower for NGS testing compared to the non-NGS method, so your more traditional testing methods. Another trial that I wanted to mention also coming out of France, so the Profiler 2 trial, which is a large trial to assess the added value of broad genomic profiling in previously treated advanced solid tumor. So here a trial was they used the Foundation Medicine test, so a large panel of more than 300 genes. And as you can see, it was in adult patient and they are receiving first or second line therapy. The interesting things here, they did two one was receiving CGP panel with Foundation Medicine. The other were receiving a control panel, so one of those smaller gene panel. And you can see the design now, it's gone through. But what the trial showed here, if you look at primary endpoint, was met, and that the CGP panel was associated with a higher proportion of patients with one or greater than one recommended targeted therapy. Now, it's a recommended targeted therapy. Now, what does it look like further down the line with treatment? More patients that did receive the CGP panel did actually initiate that recommended therapy, as you can see here. And they were looking here also some data. The main targeted agent initiated were PARP inhibitors, some of the PI3K, AKT, mTOR inhibitors, and also immunotherapy. So overall, that was like 15% of the patient initiated therapy, so 51 out of this initial group. Now, as I said, there's different way along the patient journey to use CGP, whether it's tissue biopsy or liquid biopsy. And as we all know, liquid biopsy now is opening a range of new possibility because of the convenience of it. So I just wanted to talk a little bit more on that. So here, in terms of that part where it can help at the recurrent stage and treatment selection. So we have a study running at Roche called BFAST, which is using liquid biopsy as a clinical utility tool to inform decision-making in ROS1 positive non-small cell lung cancer. So you can see that were adults which were treatment naive with unresectable stage 3 or 4 non-small cell lung cancer. They did the biopsy with Foundation Medicine, and there's different cohort, but here are the results from the cohort D, which was an open label multi-cohort phase 2 and 3. So as you can see, and we can see that there's benefits of an NTRK and ROS1 inhibitor in ROS1 positive patient within that trial when it was selected with liquid biopsy. And it was the thing also wanted to check, is it consistent with the tissue-based trial to demonstrate it was as good for a clinical utility piece, and it was. Another trial here, PRISM, looking at liquid biopsy to complement, in that case, tissue match metastatic cancer patient to look for those targeted therapy. And you can see some of the number here, but again, that really helped get those patients at the discussion at the molecular tumor board to be funneled to recommended match therapy. So in that case, we're like 56% of the patient. In some case, there was some reason, of course, to not recommend treatment during the molecular tumor board, looking at the results. So it was cases when there was not necessarily clinical trial, no match treatment was already received in the past, and worsening performance of status. But here, that's a single center study separately show that liquid biopsy had a clinical impact in 65% of the patient compared to the 22% for standard NGS diagnostics. Now, liquid biopsy has many valuable applications and also can provide complementary information to tissue biopsy. Also within the PRISM trial, showing here that the potential to identify operation around acquired resistance to targeted therapy, which is one of the topic, and one of the clinical use for NGS that we see more and more, and we identify some new mechanism of resistance over the years. But we can see here from the data from PRISM, in terms of secondary resistance DNA operation, they were found in 31% of patients. So it's something to keep in mind when a patient relapse. Can resistance alteration arise in the last few months? So if I conclude and summarize what I say, I really wanted to show that there is broad genomic profiling as really have the potential to identify and open greater treatment option in advanced cancer patient. And as I showed in the profile too, because there's a lot of, I think it's a great trial to demonstrate and talk about the debate, or should I use a smaller panel versus a large panel that would give me, TMB and MSI. But Profiler 2 does provide some evidence that those bigger panels have an incremental value from a clinical standpoint. And as I showed you, it met its primary endpoint. In addition, that all patients were on the CGP panel was initiated to targeted therapy, because in a lot of trials we see, okay, they discussed, they matched them to therapy, but have they actually initiated therapy and getting benefit? It's not always well translated this way. And in terms of the use of liquid biopsy for ctDNA analysis, it's really opening multiple potential clinical application along that patient journey in terms of its complementary use to tissue analysis, helping patient match to targeted therapy as good as it was a tissue analysis. And it's really also helped now opening that journey in the patient window where there is a relapse in identifying those acquired resistance. So I'll stop on that note and open the floor to question if there's any.
Thank you for your very good presentation about the comprehensive genomic profiling. And if anyone has question about this talk, no question. And Dr. Martin, I have one question because I also treat a patient with non-small cell lung cancer, and I know that comprehensive genomic profiling is very important to identify the possible driver mutation because we can prescribe the correct target therapy to the patient. But for me, I consider sometimes we don't have enough tissue to get the good result from tissue biopsy, NGS. So sometimes we suggest patient to do liquid biopsy, and rarely the liquid biopsy actually is a very good alternative method to detect driver mutation. That's fine. But sometimes if we want to know if the patient is suitable for immune checkpoint inhibitor, from the NGS we know if the data had a higher TMB or MSI high, that we suggest patient with immune checkpoint inhibitor. However, how can we get this information from liquid biopsy? Or the foundation medicine can provide more information regarding the the predictive marker from liquid biopsy to predict response to immune checkpoint inhibitor? Thank you.
No, thank you, and that's a valid question. And when you go back even like 3 or 4 years ago, liquid biopsy, because you're looking for that ctDNA, which I take the analogy, it's looking the needle into the haystack. So you need to be much more sensitive with your NGS technology. And fortunately, the technology has moved in the right direction. So at Foundation Medicine, 3 or 4 years ago, our panel was only 70 gene. MSI was able to be called, not fully as it was called within the large panel, but in the last 2 years, we did launch our new Foundation One Liquid CDX, which match exactly the number of gene and a genome signature like MSI and TMB. So now you are able to get from that liquid biopsy, which is very convenient for the patient, for everybody in the clinic as well, to get that information in the cases when tissue is not available. And as you said, especially in lung cancer, sometimes it's tricky because I know it's a bit of a slogan, but it's like, tissue is the issue, but it's a true representation of clinical setting.
Thank you for your yeah. I have another question from Professor Lu.
Well, I think that Roche devote a lot of effort on how to help us to identify the targeted driver mutation to help us to use the drug. But on the other hand, in the future, probably a lot of new drug will be, including in before, will just focusing on protein or RNA. For example, ER expression is about endocrine therapy. And in the future, a lot of antibody drug conjugate will talking about the expression of maybe TROP2, HER2, but not gene mutation. So do the Roche or the Foundation Medicine have any development plan regarding to how to explore your spectrum service from gene mutation profiles to RNA to protein in the future?
Yeah, no, thank you for the question. From a Foundation Medicine side of things, I'm not aware of anything like developed in the pipeline from, if you think of a proteomic platform point of view. I know they've been using RNA for a long time now with Foundation One Heme, exploring how to bring RNA also past using RNA just to detect fusion, but also to do looking at the map of gene expression as well as a potential better biomarker to what is used in using protein on slide at the moment. But it's something that we are exploring, but nothing coming soon to the market. But I think most of the industry is going in that direction. And I would say it's not necessarily moving to proteomic alone, but how do you integrate this multi-omic approach with genomic, with gene expression, with bringing proteomic, and making sense of it with the right bioinformatics behind it. But that's really relevant. Thank you for the question.
Thank you. Can I have another question? Actually, I also joined the study of Be Faster study. We checked the liquid biopsy to predict to find out some driver mutation from newly diagnosed non-small cell lung cancer. And actually, around last year, I have one patient who diagnosed with exon 14 skipping mutation detected by liquid biopsy from Foundation Medicine. But however, unfortunately, this patient has no response to MEK inhibitor. I used carbometinib or tepotinib, but no response to both inhibitor, which was available, is approved by FDA. So I consider still some limitation for the, maybe not the limitation because maybe the drug or something else we don't know, because only metaphoric sequencing I detected and I cannot find out any other possible driver mutation. And after progression on MET inhibitors are also do the repeat biopsy, but only metaphoric skipping was found, no other possible driver or passenger mutations. So I consider sometimes we do the entry study to find out very rare mutation that we feel very exciting because we can give patient very good novel targeted therapy, but still something we to understand why the patient who is resistant to the targeted therapy, but maybe current panel is not enough to answer this question, or do you have any suggestion or comment about this situation? Thank you.
No, thank you. And that's the reality, right? From since every clinical trial have been done, you never get 100% of response if a patient entering it with the right, meeting the criteria. In that case, having a mutation will do that. What I think we've learned from the last few years as well is having an alteration in a gene linked to a therapy doesn't necessarily mean that it will respond, because that will depend where is that mutation and what will be that change in the protein, because the drug have been designed typically to in response or to counteract a specific part of the protein that has been altered. And sometimes we find alteration in other part of the gene that we're not sure what do they mean at the moment. Are they really a pathogenic mutation, or is it just one of those passenger mutation that is there but not really doing anything? So we're still learning a lot about that, especially, and that's why we look at with Foundation Medicine, when you sequence without bias the entire gene rather than hotspot, you discover those alterations that were unknown before, and we catalog them and try to characterize them.
Thank you for your answer. Is anyone has any question? Okay. So I can see, thank you for your comprehensive introduction and presentation about this talk about comprehensive genomic profiling and then in clinical practice in oncology treatment. I consider time's up, so we should close here. And thank you for your joining us this conference. Thank you, Dr. Martin. Thank you. Thank you very much.
Thank you for having me. Bye.
那我们进行到今天就是最后一个 section， 然后是由我来演那个给 talk， 然后是江荣山部长当 moderator。
好，谢谢大家哈，到这么晚了哈，还留在这里哈，来参加我们今天的这个精准医学的国际研讨会哈。那我们现在进行最后一个这个题目哈。我们这个今天请到这个曾慧恩医师来讲这个题目，讲的题目是主要是在我们台中荣总哈，特别是在最近这1年里面来做这个 precision oncology 的这个经验的哈。那曾医师是我们在这里以前的这个血液肿瘤科的医师哦，那后来表现非常不错，后来有短暂的时间哈，给台北医学大学哈挖角去那边从事相关的研究。那因为事实上我们中荣努力在发展这所有的精准医学，特别是少了 oncology 这一块，所以我们又把它挖角回来来担任我们在精准医学特别这一块的中心的一个执行长所以我们今天很这个荣幸的来这个请这个曾慧医师来谈一谈我们目前我们中荣里面，在他的这个努力之下，我们在精准肿瘤这一块的一个经验
郑医师啊，请。
那谢谢部长的介绍。那今天就是因为回来大概半年的时间，然后就是跟大家分享一下在这一块的目前在台中总的一些现况跟发展。那不过前面还是会稍微提一下，就是其他部分。那癌症治疗我们大概很清楚，大概主要就包含有所谓的手术、那药物、那这个放射线免疫细胞治疗的一个部分。那手术大概各个医院其实都发展，大概就是走向一些精准，就是所谓的达文西或是一些机器人手背的一个手术。尤其在一些比较困难就是 approach 的位置，比如说头颈癌啊，然后还有一些就是泌尿道系统，然后或者是一些比较深部的，比如大肠直肠癌的一个部分。那这是我们的精准手术的部分。那精准放射线部分呢，在各个一样就是所谓的立体定位的手术。但有些医院已经有发展所谓的质子治疗部分。那在本院也是目前已经是在计划一个新建中，所以这一个精准的放射线治疗。在对癌症来讲，传统一个治疗的策略，大概我们都会根据癌症的一个肿瘤原发部位，那可能它的病理特征，那级别，然后给予药物上的建议选择。那当药物第一线有效就继续治疗，没线就转第二线，没效再转第三线。但常常这又是我们在临床治疗上会觉得这是最好的治疗模式嘛？那对于一些少见的肿瘤或是复发治疗效果不好的肿瘤，那到底该怎么办呢？那所以今天听了一整天，就是大家慢慢趋向的就是所谓的个人化，所谓的 precision 的 medicine。那精准的药物来讲，我们除了当然有一些药物可能，尤其是手术后的一些辅助性治疗，可能会比较像标准的传统治疗。那因为根据大型的临床试验，但在转移性的癌症来讲，可能还是会有我说的，还是要根据有 evidence 的 base。但是刚才讲，如果一些特殊的情况，那到底怎么样？真的病人都要所谓经历这样子的一线一线的化血治疗嘛？所以现在慢慢趋向的，可能就给病人做一个所谓基因检测。那刚才讲的，包括很多的前面的就是教授们都有分享。那有时候我们本来以为他是lung cancer，然后做下去可能他菌可能觉得它是一个 systemic 来的。那有时候他不然如果以前没做，可能就照着标准的lung cancer去做这样的治疗。那再就是我们药物上可能去筛检相关的一些平台，所以根据这些不论是基因检测或一些筛检的平台，可能会有一些生物的标志告诉我们怎么选择这个药物标靶治疗会更好。那 TPNI 的部分包括也会告诉我一些药物上一些代谢，那什么样的药物可能在代谢上这病人可以选择或是不可以选择。那再就是基因的药物筛选平台，现在很多大概药物上的体外做一些敏感的一些测试，或者是我们知道这项的基因的一个突变，可能对这一类的药物可能就是一个治疗反应是不好的，所以就走向可以所谓的个人化的一个治疗。那在精准的免疫细胞治疗，大概是今天也会有提到，就是大概是一个越来越夯的一个题目。那这所谓的CAR-T，大概今年在台大医院那发表的第一个就是诺华公司做的那一个非常成功的 AL 的案例。所以我们知道这现在的细胞治疗也是在我们的精准在癌症上非常举足轻重的一块，而且应该在10年、20年大概都是会越来越重要。那它就改造我们的一样的免疫细胞，不论是 T 细胞或 NK 细胞，那这样提升一个辨识与攻击肿瘤的效果。那感觉就大家应该都有看过美国队长，就是本来一个很弱的人，然后经过某些改造，他变成了美国队长。所以对我们的免疫细胞也是将来的癌症治疗也是非常重要的一块。那我们知道以前这种急性的淋巴性白血病负荷后预后相的差，就算它有一个就是亲属间的异体移植，长期的存活可能剩下2成，如果没有话，单纯化学治疗也只有小于10%，所以这样的治疗非常的一个不好。所以在一个所谓的 CAR-T 细胞来讲，那在转移性 B 型淋巴的白血病来讲，就可以整体的反愈率这个非常提升到8成。 那5年的一个长期存活率大概有50%的一个机会。所以跟整个传统来比较，不论是急性淋巴性白血病，或是一个弥漫性 B 型淋巴瘤，那做有 CAR-T 的治疗跟没有，就看到2条线，其实是天壤之别，就是31个月比上5.4个月的一个长期存活率。那所以在这里就是以本院来讲，目前我们已经在做就是跟诺华一样，就是申请所谓的 CAR-T 的一个国际认证，以后在这里可以做一个 CAR-T 的一个治疗。所以在今这个月的27号就预备进行它的 mock test。所以在这边对一个精准细胞治疗来讲，那在台中总也是在发展中，而且应该快要完成。那在我们这精准治疗医疗的目的来讲，那刚才其实有听到很多的老师讲过，就是其实诊断这件事情还蛮重要。以前都是靠病理去看它的 morphology， 但有时候 morphology 可能有时候是有困难诊断的，那有时候或是加上部位，所以有些真的确需要一些所谓的一个基因上的治疗检测来帮助我们，然后才可以有机会给到一个正确的治疗。那再就是基因上可能的检测，我们需要知道真正的风险，不是得到一个病人得到癌症就给他越多药物越来越好。那有时候多不一定好，有些慢慢的治疗是走上可能或许少一点，减少它的副作用，因为它可能它的预后可能有长期的存活，所以它不一定需要这么多的治疗，那可能反而造成后续的一些副作用。再是避免不必要的毒性，那我们知道刚才有一些药物的代谢，每个人其实还是会有一些不同，所以有些药物上，它可能或许要少一点的药物，它就可以达到一样的效果。当然，现在最常谈的其实还是希望正中所谓的癌症的一个驱动的一个基因，那有希望找到还重要的驱动基因，达到有效的肿瘤的一个抑制。那当然，现在上市的药物大概从2018年慢慢都是以一个基因的特性为标的。那其实在这个在很多就是刚才有看到，就是例如说像 NTRK 基因，那有这样的基因，那去做它的相对应的一个 NTRK 的抑制剂，那可以就算在一些比较罕见肿瘤上，它有很好的一个治疗的反应。那跟如果不知道，然后传统做化学治疗，就是一个天壤之别的预后。那当然就是困难肿瘤的胆管癌也是一个，包括知道有 FGFR 的一个 E2 的 mutation 有这样抑制剂，那对病人也是另外一个治疗的一个曙光。所以现在到一个未来治疗，但已经从知道它是肿瘤什么部位，我们大概已经今天听了一整天的大概用什么基因，那可能从肿瘤本身 tissue 去做，或是从它的血液去做所谓的 liquid biopsy。那再就是大家就一直在做，不止还有药物敏感测试。我们知道这些基因可能相对应用这个药，可是可能对Chanel来讲，可能可以选这个药，可以选这个药，可以选这个药，但是谁要先用比较好？对，所以有时候已经走向再来就是要做一些所谓的体外药物的敏感性测试的部分。那所以在一个精准药物治疗上，那我们的病人需要做什么样的检测才是适合，然后可以提供给我们的医疗团队去提供这个医疗的想法跟选择。那可能现在来讲，因为大家都在学习，那包括从第一场，那就是大特的时候，他就开始提，就是这包括美国这些他怎么去做这些 molecular tumor board 的一个部分。那其实在全世界大家都在进行这件事，因为现在的一些基因检测越来越多，到底这些报告我们怎么去解释，怎么去运用？那大家也一样在累积这样子的经验。所以现在的流程来讲，不过是欧美各国大概就是从我们拿到肿瘤的去 sample， 不论是血或组织，然后去做 sequence NGS， 那再就 data analysis。那现在其实还是很重要的，要进入所谓的 molecular tumor board 去讨论，因为它跟我们传统上的治疗还是很多的不同。一样的 mutation， 它在不同的肿瘤上可能有不同的解释。那不同的它的一样的基因，但在不同位点的 mutation， 可能对药物的反应也有所不同。那这是我们的精准中心的团队这样子，那包括陈一鸣医师在对面场这样子，那还有我们的实验室的博士跟研究员跟医检师。 那在这边，因为从今年3月份开始，中龙大概就是每2周在院长的支持下，每2周就是开一次 molecular tumor board。那原则上是，如果说是很 typical 的，比如说 EGFR 常见的 mutation， 比如赖兰坑什么，就不会提出来做一个讨论。那或一些很 typical 的表现就不提出来。那通常所以大家看到的大概就是里面虽然蛮多 lung cancer， 但是大概就是比较 rare 的一些 mutation 会提出来。还有比如说比较难治疗少见，比如 pancreatic cancer、cholangiocarcinoma， 我刚才有比较特别的，比如 RCC 这些困难难治，还有小颗的肿瘤，那我们才会进入到我们的 molecular tumor board 去讨论，然后给予一个建议。那目前来讲，大概已经讨论9个人。那其中的案例大概如果大家有参加过，大概就是比如这是一个胰脏癌的病人术后复发，那有肺部跟腹腔淋巴结的转移。我们知道胰脏癌是一个非常困难治疗的一个病人，那我们照现在的健保跟的指引，大概就是第一线就是卷塞塔饼加所谓的 nab-paclitaxel， 那3个月病人就恶化。那第二线呢，因为健保给付，所以的liposomal irinotecan， 所以就加4个月就恶化，所以病人其实是非常的一个 critical 状况。那第三线的时候，你就打我们常用的 oxaliplatin 加 5FU， 那就2个月恶化。那我们知道说，现在对这种比较困难难治的癌症，其实应该一开始就要做 NGS， 不过因为它还是一个自费项目，所以病人其实在第3线的时候才做。那发现它是有 BRAF mutation， 不过它位点不是常见的V600F 的位置，它是一个比较少见位置。不过经过一些讨论跟 literature 的 review， 那它开始就是除了化学药物应该还是有点，不过这些化学是他之前用过化学药，那加上一个 BRAF mutation， 那其实病人使用8个月后疾病缩小，而且是一个稳定的情况。所以你可以看到，这个特殊可以有机会可以帮医治病人的一个就是 driver mutation 的时候，病人就有机会可能肿瘤可以获得一个比较好的控制。那这是另外一个案例，那也是一个非常年轻的所谓的一个 RCC， 那它是就是我血尿发现的。那后来发现它是一个蛮特别的一个 RCC， 是一个 MIT family 的 translocation renal cell carcinoma。那当然里面就是一样，就是照我们的形式，就会大家一起讨论病科一些回复啦，包括我们还有一些我们的就是实验室的研究员的一些讨论，然后还有临床上医师讨论。那这病人是没有转移，所以他直接就是手术切除。不过对于这个病人后续如果万一他转移之后，因为相对它是一个比较 poor prognosis， 到底怎么去建议再怎么治疗、怎么追踪，还有这个可能是一个跟 families 的一个所以遗传性的癌症有相关，所以这大概就是在 molecular tumor board 讨论，就是除了治疗后续的追踪 prognosis， 那有一些需要一些癌症就是基因上的遗传咨询这部分，大概都会列入我们的讨论当中。那有些癌症的病人，那可能是一个遗传性癌症，那当然这些就是需要就是其实在这边我是在中龙目前还没有除了妇产科那边就比较以前的幼儿为主的一些遗传咨询师之外，那真正后来做癌症系遗传的咨询的遗传师在这边是没有。那所以在这边其实有些案例真的需要的时候，当然就会请就是以前北医的同事，然后帮我们上线，同时帮我们做一些意见跟分享这样子。但是这还是我们现在molecular tumor board需要的一个部分。所以在这边人员的组成主要还是一个肿瘤科的医师，那当然我们都会希望是比较相关的都会当成核心成员。那一样以比较多全癌别照顾，比如说血肿放肿科医师是一个比较里面的比较多的核心成员。那因为他跨的癌别比较多，因为我们知道有时候常常在这样讨论中是会用 off-label use 的药物，那因为医师看的癌别有时候多一点的时候，他就会跨到很多，就会知道很多不同的作用机转的药物，所以相对比较多。那当比如说有讨论到肺癌案例或是尿道癌案例的时候，那再把这样子的相关科的治疗癌症医师在一起邀请进来。 那当然很重要的就是病理科医师，那还有实验室的一些研究人员，那还有你的药师。那遗传咨询师的话，就是请线上就是之前北医的同事协助。那当然就是一些可能会需要一些所谓的协调师的一个部分，因为有些还是需要他帮忙做一些 coordinator。那Molecular Tumor Board 的一个目的来讲，其实当然一部分是很重要，对于临床咨询。那临床心理可能是给我们的临床医师，因为在是做很多这样的报告，到底是每个人都熟悉这样。像今天早上 Dr. Sony 有提到，其实并不是所有的医师现在做这么多都有这么熟悉对这个报告，所以有时候我们希望就是在这样Molecular Board提供这样的一个建议跟咨询。那当然再就会牵扯到治疗。大概也会提供一些药物上建议的选择，那大家也是共同来讨论。那当然有没有适合指导的临床试验可以转介？那我说，因为其实大家都还在学习，其实它对于我们医疗人员转，就是它还是有一个教育的意义在。所以整个 Molecular Tumor Board 它其实是 engage 在这个 cancer 的照顾的体系里面。那我们可能本来就很习惯有所谓的病人，那我们的主责医师跟癌症各管事。那有需要的时候，当然比较特殊的状况，就做这些基因检测，然后它出来的报告可能不是一个很 typical 的，不是像在该奈上有的时候，那可能就会进入到所谓的 Molecular Tumor Board， 那可能会需要协调师来帮忙做这些 coordinator 的事情。那如果有需要，它可能是一个 genetic mutation 可能有相关的时候，那可能还要再需要牵扯所谓的遗传咨询师进来。那最好在这边是还没做到所谓的存活者一个健康的管理的部分，不过这对于一些癌症来讲，其实这也是需要的。那所以目前在这边治疗流程来讲，那其实在 sequence 的时候，我想很多医院都一样，一定有所谓当外送平台，比如 Foundation One 或 ACT 或 Garden 360，那所谓的外送平台。那在这边精准实验室自己也有做，然后就目前是自己在做一些包括一些 actionable 就 Illumina 的系统，或者还有做目前有开始在做所谓的 TSO500 的一个部分。那所以因为院长在这边希望我们把一些资讯可以多一点留在自己的医院，你可以做 research。那之后再做 data analysis， 这边大概除了有使用 Navify 的 mutation profile 之外，还有使用到 Parian 的就是 diagnosis 的一个 system。那最重要是就像说，有些你在看，它告诉我们的主要的可能你看到第一页的一些主要的基因的 mutation 之外，那有些东西的 pathway 在思考的时候，病人真的没有什么药时候，可能在他藏在后面的有一些就是 mutation， 可能就要整个思考在他的 pathway 里面去选择药物。但这些全部还是要进到 Molecular Tumor 去大家共同的讨论，因为说其实大家都目前大家因为这种基因的报告越来越多，其实大家都还在一个共同的学习中，所以大家共同来做这样讨论。不同专科的专家间也会提供，就是治疗上的建议跟可能要使用上的注意事项。那所以在这边来讲，在台中总目前建立目前就是一个检测的一个平台，不论是自己做或是外送做。那再就是已经有建立的所以某一颗分子肿瘤的一个团队，那包含各个不同的医师人员跟研究人员。那再来就是希望之后院长给人的建议，就是要建构医院的基因资料平台，就像今天也很多老师跟建议的专家就说，其实应该共同的合作来建立这些资料平台。那其实非常的觉得这是应该是一定要做的，因为其实大家都在学习中，那大家可以互相交流，跟知道这样的怎么样的 treatment 对病人比较好，或是怎么样 treatment 是应该要避免的，根本可能会没有效。再就是一个药物敏感的测试，因为这件事情，当你有几个想法跟选择，它可能都是一个合理的 rationale， 但第一谁应该先用，因为有些病人不一定有时间让你这样测试下去，那所以可能这是我们现在正在建立的。那右边的部分是我们目前一些正在做的，有些是现有的平台，那有一些是医院自己开发的一些 panel 这样子。那再就是遗传性癌症部分，在今年是我们开始把它纳入，因为其实还是会遇到这些病人是他本来是 tumor 做 somatic 的 mutation， 那你会发现它其实可能是一个 genetic mutation， 或在 history 上可能需要做一些厘清。那我们现在知道基因可能不是 检测不是万灵丹，因为说不一定可以找，你找到也不一定真的是 driver mutation。但是还是非常需要在现今这个年代，是应该要在很多是一开始就应该介入这个基因的检测，那敏感测试，经过这些 tumor board 讨论，而且要经过它追踪的 outcome， 因为其实我们还是要继续检讨、跟努力、跟进步，避免不要再犯的错误。那其实这一个期待啊，就是这些检测有没有一天就像那个特斯拉就是自动车一样，只要输入这些目的资料都输进去之后，它就可以那个正确的到达，而且要安全的到达目的地这样子。那我们也希望一天这建立临床资料，那有病理，还有自己医院这些有 data 机，那还有我们之前的一些经验，那还有大家经验跟建议选择，那还有guideline，那可以跑出一个连接性的一个治疗的选项。那这是报告完毕，谢谢。
谢谢这个曾主任哈。事实上是很精简的报告。目前台中荣总精准医学中心特别是在 oncology 方面特别是做的目前的一些工作项目，看起来当然我们还是在初步的这个发展中了还在增加这个，希望能够学到更多的这个经验。那现在开放这个 Q&A， 不晓得下面的这些专家学者和我们的老师们有没有什么问题或是一些 comment 要给我们一点点这个经验的分享，有没有？我们请教一下吴副教授，我看你们林口长庚不晓得有没有在这方面做molecular tumor board， 你们的经验不晓得？
我们大概每个月也是类似啦，就开大概2次的会议，那也是大家一起来讨论，就是可能肿瘤科医师为主，那配合病理科医师。那因为有时候我们会外送到其他的检测的公司，有时候会请他们就是 medical 来一起来讨论这部分，所以也是类似。但其实大家也都在学习中啦，因为其实我觉得有时候那些基因检测结果并不是那么的就是well known的一个情况，那我们到底用什么药物，其实真的不确定到底用了有没有效。那的确很多时候就是在试试看，那也许有些病人效果很好，但有些病人效果就不是那么理想。那的确是要把这些 data 大家收集起来，然后有效没效我觉得要报道，因为我们过去的文献看到都是有效的 case 啊，其实最重要是很多没效的 case 都不会被 report 出来。我觉得没效 case 要 report， 但大家还知道说这样 case 不要再用这样的药物治疗，把没有 report 其实大家也不知道，所以我觉得这是蛮重要的。那其实我觉得我们就是大家都还在学习中啦，所以希望可以跟大家来多交流，谢谢。
谢谢这个吴教授的这个 comment 不晓得卢教授有没有什么要谈您台大的经验？
我们是觉得应该目前的证据还没有到这里了哈，所以并不是大家都还在学习中，而是说全世界都还在学习中。所以特别是乳癌，那个乳癌 NGS 能够帮的忙非常有限。untrack 或者是看到那个 tumor mutation burden， 那其他的 mutation 其实没有 role， 所以在我们的 team 里面就压根就不会有那个 molecular tumor board， 除非是讨论的是那个 gene expression profile 或者是 protein 的 expression。所以癌症很有趣，就是在光谱的极端，另外一边是 lung 的 adenocarcinoma， 他们日子很好过，就只要做那个 mutation。那个测试之后就知道怎么用药。那另外一个极端 ，breast cancer 是个代表，就因为它古早以前30年前就第一个发表说那个 RNA expression 可以盖的分 subtype， 就这个方法弄了半天，到现在没人可以 repeat， 因为其他 cancer 并没有这个特色。那最近当然那个那个 bladder cancer 或者是 urothelial carcinoma， 他们也开始分出有这样的类似的 subtype。那就大部分的 cancer 是 in between， 而且是非常在中间，而不是说靠近 lung cancer 或者靠近 breast cancer。那非常中间的这一组呢，就变成今天其实大家开会时候有讨论到，可能不是只有靠 NGS， 还要 omic， 还有很多东西的加入之后，我们对这个疾病的了解才会更深入。那这个时代是在来临中，只是说在还没来临之前，怎么应用 NGS 到我们的病人上面，确实也真的就是需要 类molecular level， 大家一起讨论，才能来猜测说它应该怎么应用。以上。
谢谢两位教授的 comment 哦。那不晓得其他大家还有没有什么问题？有没有要提问的？有没有？好，那我想我还是有一个问题要请问，刚好我们有2位专家在这里了哈，他们看看其他贵院的经验。我们事实上像我是一个耳鼻喉医师哈，我们当然不是比较属于内科系的医师哈。那可以看得到，不管是头颈癌啦，好像我们 ENT 现在处理很多头颈癌病人，那在对这些所谓的精准的医疗哦，特别是不管是 tumor 的这个 molecular board 哈，就是当然主科就像刚刚郑主任讲的，主其实还是肿瘤科的医师。那怎么样？贵院不知道怎么样来把这个临床科的，这是医生这个拉进来哦，甚至改变他们的想法哦。因为以我们外科医师的想法，通常我们都是就开刀把它拿干净，就觉得我们会场的工作我们完成了。但是后续好像就兴趣缺了哈，就是说后面哦，这个病人如果这个复发嘛，还是在开，要了不起开没办法开了就开才转，但是事实上他对这一些的我们的都不太有兴趣。我看我的同仁他们也都不太有兴趣。那事实上当然以精准医疗来看，我们现在推这个精准医疗里面，我在想，事实上我想我们医院一样，我们会发现，事实上临床医师他的病人在先去找他，但是甚至很多情况会跟他接触，但是他们都好像我们都好像不太有什么兴趣。那这样好像会让这个精准医疗里面会缺了一个很重要的一块。那我不晓得贵院你们怎么样要怎么样去想办法让这些外科医师或者说其他科的医师这样来救大家来一起参与所谓的 tumor molecular board 或者是精准医疗这一块，那让这个病人的治疗能更所谓的个人化治疗，所谓的 personal medicine。不晓得你们的做法或者是你们的建议怎么样？卢教授，
我想他的这个 molecular tumor board 往前一步，我最往前是说它的前身应该是先建立好一个多科团队。那如果有个好的多科团队，它的下一步只是说把它的那个expert 的那个范围更精英化，去讨论那个 molecular 那个 diagnosis 来协助怎么往下做。所以刚才主任所提到的议题，可能就包含了说，到底一个多科团队里面，大家合作的意愿跟合作的热情够不够？那我们运气是比较好，是因为人多，所以大家分一分，总是会有一些团队。那在很多医院里面，如果在主力上面并不是所有的 cancer 都有这么多人的情况下，其实剩下还是有机会把它。有些人当然会一人多饰几角哈，就是他可能会 cover 好几个 cancer subtype。那在那个能够定义好几个好的一个合作治疗的一个多科治疗团队，当这个团队形成之后，自然而然大家就会开始会想要往前再 move， 说那有些困难治疗或者是简单治疗，那到底 de-escalation 或者是那个 escalation， 各种的讨论就会开始变得丰富。那讨论变得丰富，大家就会觉得这个东西是值得的，就是这种互相join 的那种 discussion 或者是 care 互相帮忙，就会很有那个价值。所以我想第一步可能是就是多科治疗团队大家的向心力如何建立。那这个向心力建立，坦白讲，在台大也不是每个科都很成功。那我们也确实有几个科是相当的成功。那相当的成功，这很有趣，就是当大家捐弃说那个好像立场啊，或者团队成见，那就是变成一起的时候，其实真的会感受到就是1+1大于4，然后就是你的那个 productivity， 还有不管是在 paper 的 production 等等那些东西都会变好。那有很多团队本来一开始会觉得就是好像我为什么要怎么样可是大家还很难去 enjoy 说那最后1+1大于4的那种感觉。那如果大家已经开始真正体会到这个的话，我相信 就会比较有意愿往前走，那就也自然而然那个 molecular tumor board 的那种讨论的那种气氛，应该也就会慢慢的建立。像那个刚刚曾主任有提到，大家在讨论的时候，有时候不是自己没有精进团队，就曾主任是 call out 嘛，您是 call out 到北医的团队去回答某一些你们刚好缺那个 expert 的事情。那对于任何一个团队来说，其实大家如果有那个向心力，我相信可能就可以让那个事情做得更好。
谢谢卢教授的 comment 了，不晓得吴教授没有要补充的，或者你们的经验怎么样？
谢谢谢谢部长的提问，因为怎么讲，这样子的一个，我觉得这样 tumor board 应该是算是独立在其他团队的之外的啦，因为不会是在原来的团队。那其实我觉得就看有兴趣的人可以加入啊，因为我觉得以我自己来说啦，就在原来的团队中，像一般外科，我也不太会在意外科怎么开刀。外科怎么开刀，只要重要是他开不开得下来。那他们有时候外科一直在讨论说啊，这个血管要怎么保留啊，那刀怎么开？对我来说，好像就是能开刀跟不能开刀，能开刀交给他们，不能开刀交给我。那 ENT 可能也是一样 ，ENT 他们可能会在意说这种头颈癌的部分到底开不开得干净。那大家讨论很热烈，可对我来说就是能不能开得下来。所以我觉得可能不同的领域，大家在着重在不同的专业。那可能 molecular tumor board 的确是以外科医生来说，可能不是那么有兴趣，因为后来的药物治疗也比较少去 involve， 所以我相信每一个人发挥他在团队里面的角色，把他的最有兴趣、那最重要的部分做好，那把这些全部拼凑起来，那才是对病人最好的一个处置。大概是我的意见，谢谢。
谢谢两位教授的一个 comment。我们时间到了，不过这个线上还有一个题目，可能要请教一下曾主任呢。我这样，然后请教 NGS 临床使用的时机，还有每一位癌症病人出诊断的时候就需要 NGS， 或者是晚期治疗的时候才需要用 NGS 来选择适当的药物治疗。曾主任。
我觉得要看癌别，比如说像今天讲到比较多有肺癌的部分，肺癌大概一开始就得需要做。那刚才有讲到，如果你就用比较小的一个 panel 这些 HSPA 跟 NGS 去做的时候，其实就会发现还是会有一些重要的 information loss， 那减少了一些用 target therapy 成功的机会。那再就看像 breast cancer 可能就不是一个一开始就需要做 NGS 的 cancer。那 pancreas 跟 conjugate carcinoma 就会建议有一些困难难治，我们不知道它一开始它其实可能效果不是很好 ，chemo 效果可能不会维持很久，那有可能很快就要 move 到下一个治疗的时候，那你可能先一开始诊断，先做了这个 NGS， 你很快心里有个底，然后如果他需要，你就是在困难难治病人癌症上就直接 move 到下一线的治疗。那再就其他，比如说包括一些，比如说 ovarian 或者是 ovarian 的话，大家它现在比较多的，可能还是跟所谓的一个BRCA 或 HRD 的deficient 的patient 有关系，因为你知道它治疗不同。那不过它跟不太就是它的，如果是在它的 somatic 不是 somatic GNI， 再用所谓的 PARP inhibitor 可能都有效。所以有些人在说这 ovarian cancer 这一类的病人，或许还有他可能一开始或许就要做这些所谓的NGS 这些相关性的检测部分。所以这个问题其实很难回答，就是其实还是 by cancer 的，就是治疗的一些特殊性，比如说对标准这些治疗效果有没有效，它有没有一个很多其实还有很有效的标准治疗，那还是它是一个困难难治的治疗。那当然有时候就是，所以决定在它一开始是就要做这样 NGS 的检测。那再来就是看转移的时候，那如果经过多线治疗，有时候真的没有效，其实有时候的确就会看看有没有机会，就算是在 breast cancer 上面，它已经算是一个很多药物可能有效的病人，你还是会真的没有药物用的时候，还是会看一下，就是有没有在什么样的机会可以让病人有其他的药物选择。所以我觉得每个癌症的其实不同的。
谢谢曾主任的这个回答哈，我想这个时间也到了哈，那最后哈，我代表我们医院哈，台东总哈，感谢也是真的是谢谢各位 专家了哈，还有甚至我们参与的同仁哈，来参加我们今年举办的这个精准医疗的这个国际精准医疗的国际的这个高峰会哦。从早上可以看到很多的这个教授，我看到题目不知道好，讲者也都非常有名，可见我们这个精准医疗中心呢，花了很多的这个精神哦，能够把那么多的这个专家哈，国内外的专家请到这里来讲那么好的题目。那也可以看得到精准医疗，就像我们刚才我们讲这个讨论，真的是还是非常有趣的题目。虽然很多东西看起来都还有努力，或者是将来还有很有前景，不过的确是非常有趣的哈。那将来相信一定可以帮忙更多的这个病人哈，不管是在 tumor 或者是非 tumor 哦这一方面的哈。我相信在我们这个曾主任的这个努力之下哈，我们台中总统这个精准医疗中心，我想应该是会一步一步往前走啊。也看每年，我想应该会定期来举办这种高峰会哈，来邀请各位专家哈来一起参与，那给我们指导，那给我们一些建议哈。那最后还是谢谢大家今天整天的这个参与哈，谢谢大家。